perspective. We welcome you, sir, in our mindset and hope you will have an enjoyable time with us. Thank you. Over to you, Varun.
Thank you, Prachot. Now, I would like to request Mr. Mahmood Thakkar, sir, to start the session. We have been very keen to hear you, sir. Thank you.
Good morning to everybody. I'm Bhaumik Thacker. Thank you very much for inviting me. Special thanks to all the fraternity of IPA and Yash Institute, who is Yash Parivar to me, and Dr. Angadi sir. Dr. Vaishnav sir, I was so excited, the moment he approached me, for presenting and sharing my knowledge, whatever I've been doing for last 9 years in America, working as a pharmacist. I can share with my college from where I graduated, and share my knowledge to them and inspire them probably that they can move here and probably help help me out up to certain extent to make my job easy. So hopefully this introduction whichever I'm going to be giving you all, it will help you all up to certain extent. I know you majority of a lot of people I found out was appearing for GPAT. I was researching about that test. I have not taken that test in India, so I don't have much knowledge about it, but I hope whoever has appeared, they all clear the test, they all get the admission in M-Pharm, and they get all the success, whatever dreams they have, whatever dreams they have seen in their life, all the dreams comes true. That's all the wishes I wanted to provide to each individual, watching, listening to me. I wanted to just share a small sloka just to thank all the professors and teachers, and also my lecture is going to be in two languages, one would be English and Hindi, because it's been years I have not talked in Marathi, so mistake व्हायचे chances भरपूर आहेत. And I'm not going to be taking chance for sure, that will make people laugh at me. So again, thank you very much for inviting. And here is the small sloka for all the teachers and gurus, and that one is Guru Brahma, Guru Vishnu, Guru Devo, Maheshwara, Guru Shakshat, Parabrahma, Tasmai Sri, Guruve Namah. Well, thank you. With this notes, I'm going to be sharing all my knowledge, whichever I've gained here in the United States, with every one of you. And I hope, it's going to help everybody of you, in future. So here's a small presentation for everybody, I worked on. Okay, hopefully everybody can see this slide. Let me know if it's not visible or whatever. Just let me know.
Visible,
Is it
sir.
visible to everyone?
Yes, yes, visible.
Okay. I just want to make sure, because I'm not used this WebEx app ever, and I'm trying this for the first time. So apologies in advance. So my topic today is going to be pharmacy practices in America. I'm going to be sharing all the knowledge, all small details which all of you should know, how I work, what my responsibility is, what I do working as a pharmacist every day on day basis. I take care of at least 500 customers. They're all my patients, so I see at least 500 patients every day. And it's completely different, whatever pharmacy thing we have back in India, I've practiced back there up to certain time, but then I decided to move here in States. It's completely opposite. And what amazes me, it's gonna be in this slide. So again, this is gonna be pharmacy practices in America. End of the session, whatever questions you guys have, do not hesitate. I'm gonna be sharing my email, my LinkedIn. Please don't text me, no WhatsApp. because like most of the time like I'm working like morning, probably my schedule starts at like 6 o'clock and I work till almost like 9 in the evening. I'll be tired. I might be late to reply you. It's faster like if you just email me or whatever. So here is the presentation. It's about pharmacy practices in US. And today's my agenda is gonna be like discuss my journey, like to become pharmacist. Everybody is keen to know like okay कैसे करते हैं हम लोग like के ड्रीम देखा है हम लोगों ने कि like लाइफ में कुछ करना है, कुछ पाना है, but like certain time like we don't know, like we are all young and we don't know like what path like which route कौन सा रास्ता लेना है और क्या करना है. So like I will be discussing like this is what I my thoughts were like since childhood. I had a lot of relatives, friends living in states. I was never interested in GPAT or masters back in India because my intentions were clear, like to move US and like practice either as a doctor or probably pharmacist. I'm a pharmacist, proudly I can say that. The second thing I'll be talking about would be like pharmacy in America, like how which kind of work we do like behind the counter, like it's completely different. Uh, Back in India, like person comes to you, like they will give you the medication cheat, small cheat, and it's gonna have like the name of the medication. You dispense it. It's completely different here. Like a lot of things are involved. I would say insurances are there, medication therapy management are there. I have to make sure like a patient, elderly patient, I have like a lot of patients, they are into their 90s, 90 years old. They will be coming to my pharmacy. They'll be like, hey, Dr. Thacker, how are you, this that, like 90 years, and they're driving the car. So like I have to make sure like they might be taking a lot of medications like blood pressure medications, anti-diabetic, antidepressants. So I have to make sure, like, that before I dispense the medication to them, it's not gonna interact because, like, my life could be in danger. Like, if something happens to him, I'll be in the jail next day. My license will be gone forever. I cannot practice ever in America. The third topic I'll be discussing is different areas, like if you are coming here to America tomorrow or whenever, like different areas like where an individual can work. I'm sure this is going to be the important topic, so just in case, like if any of you are planning, are studying, or has any relatives back in US, they're planning to move here, like they can know like which areas, because it took me a while like to decide like what I want to do. It took me like after like I graduated, I came here back in 2013, and it took me like 4 years. Definitely like exams and everything was there, but again like there is a lot of like pharmacist is not a small profession, like it's a vast profession. So it's very important to know like which area you want to make a career in. And I'm not going to be spending a lot of time, probably like 15 minutes each, uh, just because I don't want to make like this a boring session, because I know like when I was a student, whenever the lectures will be going on forever, I'll be like tired, sleepy. I'll be like, okay, gossiping with my friends, and I'll be like, what are they talking about? That's all rubbish. I don't want to listen to them. I wish I can just go home, talk to my girlfriend or whatever. So I will try my best, like to keep it short, sweet, where like I will try my best to share all the knowledge. And if any question concerns, like end of this session, I will be more than happy to answer all your questions for sure. Okay, so as I was saying, the journey, it's not it's like it's tough. It's tough for sure. Uh, this is a very like it took me a while like to write this sentence. I was thinking like what to write, and I came up with this idea. I was like, okay, success is not obtained overnight. Okay, success is not obtained overnight. It comes in an installment. You get a little bit today, a little bit tomorrow, until the whole package is given to you. The day you procrastinate, you lose that day's success. मीनिंग बहुत सिंपल है इसका, लाइक के सक्सेस अगर कि आप बड़ी फैमिली में बॉर्न हुए हो, golden spoon is the term like we use here, then you might be getting the success like really easily. But that's like very 1 or 2% people. We are not born with Shah Rukh Khan's family or Amitabh Bachchan's family. We're all ordinary people. So like we have to work hard. Easily नहीं मिलता है सक्सेस, धीरे धीरे आपकी तरफ आता है। और जिस दिन आपने ट्राई करना छोड़ दिया, it's waste of time, waste of day. So let's not waste the time. Every day you have to work hard. You're gonna get the success for sure. Dr. Angari became like professor, then principal, then the IPA president. Like he became president. He had an achievement. He has worked hard. He has stayed awake the whole night to work, to climb the chairs, to climb the stairs. So yeah, just keep the goal, keep the goal. If you had a goal to clear GPAT, I'm sure a lot of people, you guys have studied hard. Don't discourage. That's what I will tell you all. Do not discourage. If you do not get success, that is not the end of life for sure. Pharmacy world is huge. If not pharmacy, it's not waste. Do not give up. But there is a lot of other fields. I know, everybody wants to travel, a lot of countries, make money. But again, just don't give up. If you fail for some reason, which I do not want, I want everybody to pass this exam for sure and achieve, whatever they want. But again, if you cannot make it, just stay positive. Okay? I'm sure everybody remembers this picture. It is from, I think, 2011, when India won the World Cup. MS Dhoni, last ball six, and look at the enjoyment Yuvraj had after 28 years. Okay, if it was Australia, winning year and year, World Cup, uh, people back in Australia, they don't care about it. But we all know, it was so much joy. After 28 years of wait, we know everybody was on the street honking, horn bajate the, India ka jhanda leke, everybody was in the street. Why? Because yeah, we waited that long. Journey was difficult, journey was difficult, but when you achieve something, yeah, we have to enjoy the success for sure. So my dream, once upon a time, my dream was to become just like this guy on the right hand side top, a doctor. Why? It guarantees secure financial future. I know doctors makes a lot of money and whatever, however they get money. uh it grants you high social status, respect, a lot of people treats doctor as if they are god. Like Bhagwan mante hai, whatever they say, yeah, they are 100% true. So I wanted that too for sure. You have a great opportunity to teach and share your knowledge, किसी पे अगर दिखाना है तो you'll be like, okay, people are going to listen to me because I am super smart. So that was when I was young. You can change people life. This is a positive point. If you become a doctor, if you want to become a really good doctor, you can change people life. People are sick, and then you all know what I mean. Then you can live anywhere in the world. Doctors are welcome, all over the world. You just have to clear some tests, some exams, and yeah, people will welcome you for sure. And it can give you more mindful, life for sure. So I had a dream. I'll be honest. I had a dream to become a doctor. But well, what happened? You see this glass on the right, left-hand side corner? Dreams broke. Dreams broke. किसी ने पत्थर डाला. Failed 12th grade. I wanted to become doctor, but okay, at that time, just scored 50%, which was really bad. I could not even get the admission anywhere. That's when Yash Institute, I was so lucky. I was so lucky. Okay, किसी ने मेरा हाथ पकड़ा for sure. Back in 2007. And that's why, like, those years, those 3-4 years I have spent at Yash, and the time I've spent with all the professors, oh man, I am very much thankful to them. Like, they have changed my entire life, where I was and where I am right now. Like, it's all thanks to all these people. Like, I know a lot of people have come, they have gone, but you guys are still here and, like, standing there like a pillar. All the students, I'm telling you, all the professors you guys have, you all are very fortunate. Like, these guys, like Dr. Angadi, Dr. Vaishnav, Shushma ma'am, Vandana Patil ma'am, Ashok Narute sir, he was my all-time favorite. And Girbane sir, like, all your professors, like, I still remember every single days, like, I've spent with you guys. It's been 13 years. 13 years, long time when I graduated, I would say. Like 2009 is when I graduated. But, like, every single time, like, whenever anybody discusses something, I'll be like, okay, I wish, like, I can go back to those days and I can get some time to spend like quality time with all these people who help me, like, nourish myself. And wherever I am right now, I have achieved a lot just because of these people, like, in short matter of time. So again, thanks to you. Thanks to you. I'm sure you all are watching me. If not, please convey my message. Like, whoever is still not there, I hope, like, to get in touch with them. So, like, then after Yash, like, I went to Nirma University. I scored like really good. Like, all these professors shared really good knowledge. They taught me you have to do something. You have to do something for sure. So they were really positive morale. I was from Pune. I moved to Aurangabad. I still remember, I think, like, first or second batch, and people were start staring at me. Okay, he's coming from some big city, and I was shocked. I'm like, why everybody's looking at me? But then, yeah, like, वैष्णव sir, like, उन्होंने बहुत comfortness दिया है. So all thanks. I still remember a lot of things. Like, but hopefully, like, we can talk more in future. And I want to share all those good memories. I want to nourish. I want to talk to you about that for sure. Okay, near my university, because, like, कि यहाँ पे program है कि, like, मेरे friends थे, मेरे I have a cousin who was practicing pharmacy. He told me, like, that if you go to Nirma, they have like 5th year. Like, it's basically just a clinical knowledge, 1 year of more, like, college. But there you just practice in a hospital setting, basically, because that's what we do here. Like, drug interactions and a lot of other things before the medications is dispensed to, like, the patient. Uh, that's what they teach you. So he was like, okay, you should apply there. So you saw, like, how much I scored in 12th grade, around like 50%. I don't remember right now. But then, like, when I was in Yash, uh, I was studying really hard. Like, I learned a lot with, like, staying with my, like, roommates who was from small town, small village, but very smart people, like, very intelligent. So I think they helped me as well. Like, I would say up to a certain extent, to focus on my study. They helped me a lot for sure. So, like, that's how, like, and then a day came finally from India to US. Now, question, why to become pharmacist in America? So I had a dream, as I showed you, as I shared with you all, like, I want to become a doctor. I could not for whatever reason. I graduated in pharmacy. Uh, Here's the answers. Why to become a pharmacist if not doctor? Basically, end of the day, I make same money, probably, like, more money than doctor would be making here in US for sure. It definitely, like, improves patient's outcome. I'm all about my patient. I just love all my patients. They all love me, like, to my death for sure. Like, but I still remember, like, like, एक patient था, like, she was 80 years old, 80 साल की थी, ब्रेस्ट ट्यूमर, ब्रेस्ट कैंसर, मेरे पास आई, लाइक, कि 2 साल से मेरे पास से लाइक दवाई लेती थी, कैंसर की ट्रीटमेंट चल रही थी, कीमोथेरेपी ये सब। तो लाइक कि मेरे पास आई एक दिन, उसको मैंने बताया लाइक आफ्टर सर्टेन टाइम, if you are taking care of your patient, after certain time they become your family. So she told me like I told her, in fact, like I approached her, I'm like, okay, I got a better opportunity and I will have to move, like I will have to switch this company and I will have to move to Walgreens Boost Alliance. And she started crying suddenly. She started crying. I'm like, what happened? I'm going somewhere, like, for my personal growth. What is that that's making you cry? And she's like, okay, Bamek, I considered you like my son, and I wanted to die in front of you. I wanted you to work here and take care for me, like, till the time I'm alive. So as I said, like, after a certain time, you and your patients, like, you all become like a family. So like, that's the first point, patient outcome. Like, you make a huge difference, like working as a pharmacist in America. Like people trust you just like, we trust doctors as a god back in India. They trust pharmacists like a god. So whatever I will be telling them, they'll be like, okay, they're way better than doctors for sure. Then community involvement, like, we also pandemic, COVID pandemic, community involvement इतना बढ़ जाता है, like कि I worked really hard, I will be honest, like I would be working like every day 15 16 hours in a day, like so company saw, like Walgreens saw, like even though he's coming from, like, he's brown, he's coming from different country. I've never ever faced like racism thing, like never. I'm proudly can say that. And they gave me the opportunity because they saw, like all my white people, all, like all my white patients, they're trusting me more than they're trusting their own doctors. So company realized that. Company was like, okay, we are gonna make you the head pharmacist. So head pharmacist for during all this pandemic season, where I was involved in immunizing all my patients. I was traveling, like every day, probably हर रोज के, like 200-300 mile, because I was the lead pharmacist and they gave me like, I don't know how many pharmacists and how many technicians they gave me. I don't remember at this point for sure. But yeah, community involvement, like इतने छोटे छोटे गांव, like इतने इतने बड़े बड़े गांव में मेरे को जाने का चांस मिला, जहाँ पे like के people, like people who are staying in that town, they are staying in that community, they have not seen any humans for like for so many days because it was locked down. I'm sure like India had a lockdown as well. I was visiting India a couple years back, and I remember like at 9 o'clock in the evening, like nighttime, you cannot leave out, otherwise people, no, police, danda marega. So like we had like similar situation back here in US, like complete shutdown. So they welcomed me so big time. They were so happy to see me. So community involvement, I got the opportunity for sure. Third point, diversity of career options. Huge diversity for sure. We'll go to that point. I'll show you like where, which area you can practice. Uh, there is huge change, like as far as working in India and working in US. We'll go through it for sure. The other part, like pharmacist in US would be like being part of healthcare team. You are talking with doctors every day. You can be rude with your doctors. Back in India, you cannot be rude with doctors. Whatever they say, they are correct. But here in US, you can tell them like, hey, no doc, you are wrong. You are trying to kill my patient. So I'm not gonna tolerate that. So like here, pharmacist, doctors, and nurse practitioner, they all work together in the same boat. And working as a pharmacist has huge growth potential. Pharmacy tech, pharmacist, pharmacy manager, and then like you can just climb the ladders. Like whatever I make, uh, it's not a lot, but like my president of my company, she's African American. Every year she makes 25, 25 lakh US dollar. No, no, 25 lakh US dollar per year, lot of money. You can convert that in Indian rupees. I don't know. So yeah, huge growth potential. Like, sky is the limit for sure for all the pharmacists. Okay, so as I was talking, like, that's me, a handsome looking guy on the rightmost side, the pharmacist. My duty, as I said, like, I'll be talking to doctors every day, whether it's oncologist or dermatologist or whatever, general physician, I will be talking to them every day. Uh, there is still a confusion, like, among the people, what is my role? What is doctor's role and nurse's role? It's completely different. It's difficult to make some people realize, yeah, like, what I do. I'm specialized in medications. The knowledge I have, like, pharmacotherapeutics, pharmacology, biochemistry, doctors does not have that knowledge for sure. Doctors diagnosis doctor's diagnosis, like, what the problem is in the body. Like, when you take your car to a mechanic, mechanic will check the car कि उसमें problem क्या है. But like, कि जब medication का part आता है, for sure it's my responsibility. Like, I'll be the one to decide which medication is good for which patient. So that's my role. That's a big difference for sure. And nurse practitioner, he or she is gonna be assisting doctors. Nurses are able to write prescriptions. Pharmacists can write the prescriptions whenever, like, we have to. A lot of other things, like, we as a pharmacist can do for sure. So here, this is the numbers, okay? Note down. And I'm sure, like, a lot of people, whoever is watching, they will think, yeah, they want to move to US because I think, yeah, there is a huge scope. Every year, okay, we have 52 states, 52 states आहेत अमेरिकेत. आणि every year there is like 20,000 people, graduates, 20,000 graduates, local people, and just 200 to 500 maximum foreign grads like me. Dr. Angadi sir was asking me, like, what is your title? Am I a doctor or what? It's completely different. Like, there is very few people who comes to US and they can work like, how the local people are working. So I can proudly say, like, yeah, like Yash Institute, uh, I belong to those 20,000 category now. What is the average salary? Okay, average salary for the pharmacist. I'm sure everybody wants to money, good bucks. Starting pay, it's all like, diversions, I would say. Like, it's always a range. Like, if right now I am hiring a lot of people at my place and a couple other places, I'm looking for pharmacists. Uh, I can offer from $45 per hour, every hour $45. I can pay them up to $120 per hour. So that's at 9,000 Indian rupee every hour, I would say. And yearly package, if you convert, a pharmacist makes like, anywhere between 95,000 a year up to, that's the, like, range I've said, probably new grads or like, somebody with the experience. But there is huge, like, yes, money is a lot for sure for pharmacists for sure. As far as jobs, like, the right hand side, every year the demand is increasing. Supply, as I said, like, there's just 20,000 people, 20,000, like, pharmacists in the country every year. Senior people are retiring. Whatever people, seniority people, they are retiring. Retirement age is 64 here. A lot of people are retiring. Some people, like, if you're working as a pharmacist, if you're working as a doctor, you work for 10, 15 years, अच्छा बैलेंस हो जाता है बैंक में. Lot of people, like, my staff pharmacist, जो एक गोरी pharmacist थी, I have a picture with her in next few slide. She is probably like 47 years old. 47 years old. She's retired. Why? Huge money. So, like, every year, like, she have saved up a lot. So every year, 6%, say 6 to 10%, like, job raise हो रही है every year. Like, कि आप यहाँ पे आते हो, जॉब मिलने के चांसेस बहुत ज़्यादा है, बहुत ज़्यादा. Right now, like, I don't know if anybody follows me on LinkedIn. I was looking for, like, pharmacist. I'm willing to pay. Okay, if you come to me, I'm willing to pay $50,000 bonus if you come and work for me. So I'm giving them, like, before even you start and work for me, here's the money. And you have to stay with me for 3 years. So yeah, like, a lot of money. So all your wishes can come true for sure. So far, I've traveled 18 countries thanks to this job for sure. Okay. So, like, journey to become a licensed pharmacist, it was tough. It was tough. Nothing is easy, as I said before, like, but you have to work towards whatever your dream goal is. So the picture you guys are seeing on the right-hand side, that's the most, like, beginning picture. When I moved to States, I was working as a technician, probably $10 an hour, very basic job. Very basic job, like, technician, assist pharmacists, pharmacy managers, like, filling prescription, or, like, dispense medication. So technicians assist pharmacists basically. So that's the job, like, I took because I wanted to practice as a pharmacist in future. Like, whenever I meet all the requirements, I wanted to practice as a pharmacist. So that's the basic job. Like, I don't, at that time, I didn't care about money. Whatever money I'm getting, perfectly fine. I took a job, started working as a tech. Uh, People used to like me, like, since day one, like, with all the service I used to provide them. We Indians, like, that's the best thing. Like, we think from heart first, and then we think from brain. We think from heart. So, like, koi buda jata hai, even right now, like, if they don't have money, I'll pay for their medications. So, like, we always care. That's, like, God has created us, like, all the Indian people. So yeah, like, since beginning, like, people loves me for sure. What are the steps? These are the steps, okay? If somebody of you are planning to work as a pharmacist, take down the notes for sure. This slide, I can share it with you in future if you guys miss. Uh, but yeah, like, these are the steps if you want to work as a pharmacist. First, you have to apply for FPGEE. It's the Foreign Pharmacy Graduate Equivalency Exam. You have to have like 5 years bachelor's program done in India or any foreign country. Then only you are eligible to take this FPGEE exam. You pass the test. TOEFL. TOEFL, I'm sure a lot of people have heard. Test of English as a foreign language. I'll tell you, like, that test is very easy. The problem with us, because English is not our primary language, majority of the people, like, के problem होता है, like, speaking. When I was new, I had like heavy accent. People would not understand, and they will ask me, like, could you repeat, like what you just said? Could you please repeat it? So, like, you have to score certain, like, points in each category, like, listening, reading, writing, and speaking. Like, you have to score like certain points. यहाँ पे बच्चे जो रहते हैं, like, they go to libraries, like, at a very young age. We don't have, like, that much libraries back in India. Like, here, every city, every town will have so many libraries, so many books. People, like, kids at young age, like, 5, 6 years, and they will have laptop, and they will have, like, iPad. They will be reading, like, they will be listening. So, like, yeah, like, it was tough for me. Initially, it was tough, but I cleared. When you clear this two test, the first and second one, you get FPGEC. That stands for Foreign Pharmacy Graduate Examination Committee certification. You get the certificates. Yeah, that you meet all the National Board of Pharmacy. In America, you meet their requirements. Now, your education from India and education in US, once you clear this two tests, 100% same. So PharmD, doctorate in pharmacy, bachelor's in pharmacy in India, or MPharm or whatever, PhD, whatever you have studied, it's equivalent once you clear this two test. And then you submit that. But again, like, after this, still, like, it's a long journey. Like, you have to work in like hospital pharmacy or retail pharmacy for 1 year, so that you get like familiar with, like, how the pharmacy functions. how the pharmacy runs in US. So yeah, you have to send those hours to the board, and then that, like, pharmacist or pharmacy manager will sign it, and then they can approve your exam. Last two exams, NAPLEX, वो है लाइसेंस की एग्जाम बेसिकली, लाइक which is valid in entire US. It's valid entire US. If you are planning, I'm based in Chicago, Illinois, but if I want to go and practice in Florida, if I want to go and practice in New York or California, wherever, NAPLEX will take care for it. GPAT, I think that's valid. If I'm not wrong, it's valid for entire India. So similar exam, NAPLEX, is valid throughout the US. But the last test is the law exam. Law exam is different. 52 states, different law exam. So right now I'm practicing in Chicago, Illinois. If I want to go in different states, I have to take their exam as well, because every state has their own laws, especially narcotics. People in America, narcotics, so very strict laws, DEA, FDA, a lot of things, so yeah, law exam is very important about that. Once you are done with all these 5 tests, officially a pharmacist. Okay, I've taken a video so you guys can see the pharmacy setting. It's going to be just 9 minutes video. I'm going to be playing that really quick so you get an idea. Almost 1 लाख स्क्वायर फीट की फार्मेसीज रहती है हमारे वहाँ इंडिया में। कैसे छोटी छोटी फार्मेसीज रहती है, वैसा नहीं रहता है यहाँ पे। बड़ी बड़ी फार्मेसीज रहती है, ऑलमोस्ट 100,000 स्क्वायर फीट, 1 लाख स्क्वायर फीट की फार्मेसीज। हियर इज सम वीडियो फॉर एवरीबॉडी।
Hi, how are you? I just got a call. My medicines are ready. So, sure, no? दोस्तों, हमारे यहाँ इंडिया में आपने देखा होगा जो मेडिकल स्टोर होते हैं वो अपने घरों के आसपास या फिर किसी चौराहों पर और एक बहुत ही छोटी सी दुकानों में होते हैं। लेकिन आज मैं आपको दिखाने वाला हूँ अमेरिका का मेडिकल स्टोर, और ये स्टोर है, इसका नाम है CVS फार्मेसी। अगर आप गूगल पर सर्च करेंगे तो ये एक बहुत बड़ी फार्मेसी है अमेरिका में, और यहाँ पर मेडिकल स्टोर कैसा होता है वो पूरा जानकारी हम इस वीडियो में देखेंगे। तो वीडियो बहुत ही इंटरेस्टिंग है, इसलिए आप इसे आखिरी तक देखें। और अगर वीडियो पसंद आए तो इसे लाइक और शेयर जरूर करें, ताकि बाकी के लोग भी जान सके कि भाई अमेरिका के मेडिकल स्टोर कैसे होते हैं। तो जैसा कि आपने देखा पीछे साइड में जो वाइन के कूलर वगैरह लगे हुए थे, फ्रिज वगैरह, उसमें बियर वगैरह भी मिल रही थी, वाइन भी रखी हुई है वहां पर। और ये क्या है, फोटो निकालने के लिए एक प्रिंटिंग मशीन रखी हुई है। यहाँ पर क्या है, अलग से कोई एक स्टूडियो नहीं होता है कि भाई आपको अपनी कोई पासपोर्ट साइज की कॉपी फोटो निकालनी है, तो उसके लिए अलग से नहीं होता है। ऐसे ही मतलब कि फार्मेसी या फिर सुपर स्टोर जो होता है उन्हीं के अंदर ये अलग से लगा के रखते हैं, जहाँ से आप फोटो कॉपी ले सकते हैं या फिर फोटो प्रिंट करा सकते हैं। तो सामने आपने देखा होगा कि ये फार्मेसी है मेन, जो जिस चीज के लिए वीडियो में बना रहा हूँ वो। और मुझे क्या है, यहाँ से अपनी मेडिसिन कलेक्ट करनी है, वो सब पूरा प्रोसेस में आपको समझाऊंगा कि कैसे मतलब कि मेडिसिन आप यहाँ से कलेक्ट कर सकते हैं। तो सबसे पहले आप पहले वहाँ जो इन्होंने डिस्टेंस का लगा रखा है 6 फीट का, तो यहाँ मैं वेट कर रहा हूँ अपनी बारी का, और फिर मेडिसिन पिकअप करते हैं। टेक योर टाइम, थैंक यू सो मच हेल्पिंग अस। तो जैसा कि उन्होंने बताया कि आपकी मेडिसिन रेडी होने में थोड़ा टाइम लगेगा, तो मैं उसके लिए वेट करूंगा। और ये जो आप देख रहे हैं काउंटर के पीछे जो एरिया है, वहां पर मेडिकल स्टाफ और पूरी मेडिसिन वहीं मिलती हैं। और मेडिसिन लेने का यहाँ पर एक ही तरीका है कि सबसे पहले आप डॉक्टर के पास जाओ, और अगर डॉक्टर को लगता है कि भाई आपको रियल में मेडिसिन की जरूरत है तो ही वो प्रेस्क्राइब करेंगे। और आपको मतलब कि वो कुछ लेटर या प्रेस्क्रिप्शन लेटर नहीं देंगे, वो डायरेक्ट पूछेंगे कि आपके घर के पास कोई मेडिसिन है का स्टोर है क्या, या कोई फार्मेसी का स्टोर है क्या। तो मेरे घर के पास में था, तो उन्होंने डायरेक्ट यहाँ पर वो ईमेल कर दिया है इन लोगों के पास, और अब मैं सिर्फ यहाँ पर आया हूँ और मुझे वो पिकअप करना है अपना आईडी दिखा करके। तो ये देख सकते हैं आप, मतलब कि अभी क्रिसमस का टाइम चल रहा है, तो ये पूरा वाइन वगैरह भी रखी हुई है और कार्ड, क्रिसमस के कार्ड वगैरह यहाँ रखे हुए हैं। तो ये आप देख सकते हैं दोस्तों, कितना मतलब कि ये स्टोर यहाँ पर इतना बड़ा है कि यहाँ पर हर चीज मिलती है, XYZ कोई भी आप चीज ले सकते हैं यहाँ पर, जैसे कि ये ब्यूटी से रिलेटेड जो है, फैशन वगैरह से रिलेटेड नेल पॉलिश और नेल पेंट वगैरह ये सब यहाँ रखे हुए हैं। तो मतलब कि मैंने कभी सोचा नहीं था, जब पहली बार मैं यहाँ आया था तो मैंने देखा इस CVS फार्मेसी के अंदर आकर के देखा कि कितना बड़ा मतलब कि मेडिकल स्टोर होता है, कि जो अपन रेगुलर डेली लाइफ में चीजें यूज करते हैं वो सब भी यहाँ पर मिलती हैं। जो सामने आपने देखा होगा कि कूलर में पानी वगैरह भी है, दूध वगैरह भी है, और स्नैक्स भी आप ले सकते हैं वहां पर, और बाकी की जो गिफ्ट आइटम होते हैं, बच्चों के आइटम होते हैं, वो सारे मतलब कि यहाँ पर अवेलेबल है। आप देख सकते हैं लोशन वगैरह ये सब। तो कहना मतलब कहीं ना कहीं जैसे अपने इंडिया में होता है ना कि मेडिकल स्टोर मतलब कि एक छोटी सी दुकान होती है, उसके अंदर दवाइयाँ होती है और अंदर भी नहीं जाने देते कई जगहों पर के, तो ऐसा मेडिकल स्टोर होता है। लेकिन यहाँ पर ऐसा कुछ भी नहीं है। आप यहाँ आओ, कोई भी सामान जो आपको चाहिए वो उठाओ और अपना लेकर के सीधा बिलिंग करा सकते हैं अपन। बिलिंग काउंटर दो अलग अलग है, जहाँ पर एक जगह मेडिसिन का काउंटर है और दूसरी जगह जनरल जो चीजें होते हैं उनका काउंटर है। वहां आकर के आप बिलिंग
आपको ये कर दो, क्योंकि और कोई दूसरा आदमी इसके ऊपर
तो जस्ट अभी मुझे कॉल आया कि भाई आपकी मेडिसिन रेडी है, आप आकर के पिकअप कर सकते हैं। तो अभी मैं वहाँ काउंटर पे जाऊंगा अपनी मेडिसिन को पिकअप करने के लिए, और आप भी पूरा देखिए आगे कि वो कैसे मैं अपनी मेडिसिन पिकअप करता हूँ वहाँ पे या बिलिंग कराता हूँ, वो सब आप डिटेल में देख पाएंगे। तो यहाँ पर क्या है कि कोविड-19 की ड्राइव थ्रू के जरिए टेस्टिंग चल रही है, तो ये लोग क्या है उस चीज के बारे में और बिजी है। तो अभी इन्होंने मेरी मेडिसिन निकाल ली है और अब मैं उसकी पेमेंट कराऊंगा। नथिंग। What is that? My mobile number? Yeah. No. No. Yeah. Thank you. Have a good day. तो पहले जो लेडी खड़ी हुई थी वो किसी और काम में बिजी हो गई, तो उसको कुछ समझ में भी नहीं आया। तो ये भाई साहब आए और उन्होंने वो मेरी मेडिसिन मुझे दी, और जैसा कि आपने देखा कि $32.99 की वो मेडिसिन मुझे पड़ी है। और ये है दोस्तों CVS फार्मेसी। और ये मतलब कि क्या है, अमेरिका में गिनी चुनी ही ऐसी फार्मेसी है जिनके अलग अलग स्टोर पूरे ऑल ओवर अमेरिका में है। उनके मैं आपको नाम बता देता हूँ, जैसे कि वॉलग्रीन है, CVS फार्मेसी है, और रेडिएंट करके एक फार्मेसी होती है। और वॉलमार्ट के अंदर भी मतलब कि जो वॉलमार्ट के स्टोर होते हैं उनके अंदर भी वॉलमार्ट की फार्मेसी होती है। तो यही गिने चुने यहाँ पे फार्मेसी के स्टोर, और जो कि ऑल ओवर अमेरिका में फैले हुए हैं। तो आप देख सकते हैं कि यहाँ पर अभी क्रिसमस का टाइम चल रहा है, तो क्रिसमस के गिफ्ट वगैरह भी इन्होंने लगाए हुए हैं अलग अलग।
Okay, so that was just a small introduction I wanted to give. I don't want to spend too much time either for sure, because I know there's more lecturers coming there. Now the next slide we are going to be moving would be like different areas where anybody, not just Indian pharmacists. I give the example of Indian pharmacists because like that's what we all want to know, different areas where we can work. As you can see the pie chart here, 40% people either works in retail or hospital pharmacy, okay? Because hospital and retail pays more basically. 25% people goes in research and development, just like India. Research and development, we need it like, everywhere, no matter where we go. 10% clinical development, pharmacovigilance, 3% sales and marketing, regulatory affairs, and medical science division. So these are the areas like you guys can work. Like if you become a pharmacist here, or if you could not become a pharmacist, these are the areas you can work at. Now, like what are my duties? Like working as a pharmacy. I know somebody I saw like the question popping up, like what does the pharmacist does actually? Patient care is like what basically I do. In America, the pharmacist provides pharmaceutical care to patient as well as basic primary healthcare services such as immunizations. Using knowledge of mechanism of action of the drugs, the pharmacist understands how they should be used to achieve maximum benefit, minimum side effect, and to avoid any kind of drug interaction. Whatever we learned in pharmacology, we are actually implementing in America, like, about the medication. That's what we do. Services, like, we provide as a pharmacist. We extend a prescription. We can write a lab order for a patient. We immunize, we vaccinate the humans, and we administer a medication to demonstrate how to properly use. Like, a puffer is there, nebulizer machine or whatever. We explain them because not everybody knows how to use it. This thing, like, I have explained in little detail, like, for example, extend the prescription. Pharmacists can extend certain prescription. अगर समझो किसी ने prescribe किया है like diabetic medication. America, like medication has to come from doctor. They have to write and then only pharmacist can dispense that. Without prescription, you cannot get any medication. You cannot for sure. But again, like there is chances like that doctor might be out of the office, he is visiting, traveling, meeting friends. At that time, like you have a right. You can write the prescription for that patient. You can dispense emergency supplies, like if not the narcotics, but any other medication, like you can dispense that as an emergency supply. It's called extension. Okay. But again, end of the day, you have to notify to the doctor that, yeah, like, in your absence, you have prescribed the medication to that patient. Okay. You can prescribe lab test. Yes, you can prescribe lab test. Which kind? Bone density, INR, to check like warfarin therapy, liver function test. अगर किसी को टीबी हो गया है, ट्यूबरकुलोसिस, ट्रीटमेंट में लाइक लीवर फंक्शन टेस्ट भी चेक, और मिथोट्रैक्सिट थेरेपी, उसमें लीवर फंक्शंस बिगड़ जाते हैं तो लाइक वी कैन प्रिस्क्राइब लाइक लीवर फंक्शन. EKG for like QT interval, we can prescribe that. Chest X-ray, okay? If somebody is on amiodarone for their heart, their chest X-ray, we have to monitor for sure. A1C test for like diabetes, then thyroid function test, like you can prescribe. You have to follow, as I said, like every state has different law. Certain state let you like prescribe like the laboratory test. You have to have the prescription no matter what. Whatever you do, you have to have the prescription for everything, so it gets like recorded in an individual's profile. So you can do all these things back here. What other things you can do? Prescribe a medication when no diagnosis is needed. Like a person is a like chain smoker, you can prescribe them nicotine patches, like smoking chewing gum, like there's chewing gum like nicotine patches, then chewing gum comes as well. Then morning after, like birth control pills, after sexual activity, whatever, like you can prescribe that too to the like women's. You are pregnant and you suffer from nausea and vomiting, like which medication, so like maclizine, ondansetron, you can just prescribe that to them. You don't have to visit the doctor. They are very open. They will tell you like this is what is going on. I need this pill. Can you prescribe? Yes, you can. You are trying to get pregnant. Which medication? Yeah, you can. Then certain kind of like folic acid and vitamins you can prescribe. You or your child is traveling to the area where you could get diarrhea, malaria, acute like mountain sickness, you can prescribe the medications for that. Lice treatment, you can prescribe the medication for that as well. For that, like, you don't want to go to the doctor's office and sit in the queue, spend 2 hours. Pharmacist, easy to approach. You don't have to wait. Like, pharmacist, that's why like people prefers talk to the pharmacist better than like they prefer to talk to the doctors. So that's why like we have good relations to my patients compared to my patients have relation with their doctors. For sure, I have good relation. Okay. Prescribe medication for certain minor health problems like acne ho gaya hai. So acne में like which medication, minocycline, doxycycline, you can prescribe. Mouth ulcers, allergic conjunctivitis, menstrual pain, बहुत common है, people don't talk about it. There it's opened, you can prescribe them the medication. Eczema, like mild to moderate phase. Kids, like I have a 40 days old daughter. She wears the diapers. Fortunately, she does not get diaper rash, but you can prescribe like medication for diaper rash, hemorrhoids, cold sores, all these things. Like see like example, I have like taken out where you don't have to go and see the doctor. Vaginal infection, you don't want to tell a lot of things for sure. You can prescribe it. Okay. Next thing is like adjust a prescription. Your pharmacist can adjust your prescription in order to reduce drug side effect. Okay. Basic example, atorvastatin or any cholesterol medication. Most common side effect, I'm sure Dr. Angadi sir have taught you all कि muscle pain बहुत होता है उसमें. So like as a pharmacist, you can decrease the dose or you can change the medication. Hypertension medication, diabetes medication. But again, end of the day, you have to explain the same thing, like why you did that. It gets documented for sure. So you have to tell that to the patient's doctor. So that way, like multiple drug interaction and a lot of things does not happen. You can substitute a medication in the event of inventory shortage. Okay, if certain medications are not coming, like 80% of the medications comes to US from India because India is the biggest market for all the generic drugs. Generic drugs way cheaper. You can substitute like whichever is the safest possible alternative is, whichever cheapest alternative you can substitute. Then next thing, administer a medication to demonstrate its proper use. For example, a lot of people like they don't know how to use the injection. If they're diabetic, they don't know how to use like the syringes, needles, or like the pens. Lentus, Solostar, Humalog. I know that's a very common brand back in India as well. We use the same brand here. So like they don't know because like first time diabetic patient, they don't know. So you can teach them like it's like subcutaneous route or like which way you inject. You explain them. So that's your responsibility. So I hope like whoever raised their question, these are like duties like we perform. This is another like small slide. The पहला जो picture था, पहला जो video था, like that was the outside of the pharmacy. This is like the back door. Like हम लोग अंदर क्या करते हैं, different roles of different people, like technicians, pharmacists. This is that video.
Welcome to the pharmacy. Filling prescriptions isn't the only thing pharmacists do. Today we will be exploring the prescription filling process and discovering ways you can speed up the procedure. The first station is the drop-off station. This is where you drop off your prescription and give it to a pharmacy team member. It is also where they gather important information from you, so don't just leave a prescription on the counter and walk away. Well, hello. To drop off, here we need your name, your date of birth, your address, possible drug allergies, we don't want you to die, other medications you're currently taking, your insurance information, and any other medical condition that you may have. Also, make sure you let us know if you have your insurance card or if it's changed, because we want to make sure that you get the best price. The next station is the RX filling station. This is where we process your prescription. I am going to talk to you about prescriptions. Things that that we may like run into a problem that may take longer to fill the prescription is the doctor may not put their name on it. I don't know why they do that. They may forget like the quantity or like the strength of the medication. I don't know how they expect us to fill it. It's like a guessing game. I guess I didn't take the mind reading class that they wanted us to take. Anyway, without that proper information, we cannot fill a prescription because it's technically illegal. Depends who you are. Maybe we'll make an exception. Another thing that may happen is the insurance may not want to pay for the medication. So what that means, guys, is that the doctor's office, insurance company, they have a fight. And whoever wins, basically they eventually let us know and we'll run the prescription or they'll change it to something else. In the life of the pharmacy. Have a great day. The next station is the fill station, where we assemble your prescription. At this station, we take the label that printed off at the previous station and assemble the prescription. We're all assigned a name tag so they can track down to see who assembled the prescription. So we scan our name badge, we scan the label, and then we scan the prescription bottle. If you scan the wrong one, it'll make it sound like this, letting you know you selected the incorrect drug. So then you'll have to redo it. So you scan your name badge, you scan the label, and then you scan the drug. And that's to let you know that you selected the right drug. So then we'll count by 5. And this is so we can count a larger quantity more efficiently. And then we'll put it in a bottle. Put the label on there, and then the pharmacist will now verify the prescription. The next station is the pharmacist verification station.
You made it to the final step of the prescription filling process. This is where I check for accuracy, correct dosing. I also check for drug interactions and allergies, all to make sure the prescription is safe for you. Now, next, I'll show you a couple of other things that I do here in the pharmacy. Oh, hi there. I was just logging in my controlled substance order for today. This is just one of many things that we do to keep track of controlled substances and prevent theft here in the pharmacy. We also perform a monthly inventory on these drugs, and only the pharmacy manager can order them. So all of this is very time consuming and can take away from my other responsibilities. I have all these books around the pharmacy, but I don't need them because I know everything. Just kidding. The world of pharmacy changes very rapidly, so there are times when I have to look up information. We mostly use an up-to-date database like facts and comparisons, but I have these other references around as well, especially the Texas Pharmacy Law Book. Here in the pharmacy, our phone takes up a lot of our time during the day. Comunicamos con doctores, pacientes, otras farmacias, y compañías de seguranza. And doctors oftentimes leave prescriptions on our voicemail for patients. Let's listen to one now.
My name is Dr. For prescription patient, the name is Vicky. And I put the bars, 2 milligrams, 2 bars. I think I put 4 times a day, and I want to give them 0. The next one is for the higher cortisol, 13 times. The nurse is day. I give them the patient only wants the other one. Prescription of promethazine with codeine, 2 tablespoons 3 times a day. It has to be the purple brand. Patients allergic to the red kind. 36412. Good. Phone number is 413-888. Patient is on our way, so please take one. We also don't have insurance.
That sounds shady. So let's see. Doctors calling in 3 controlled substances, using street names for drugs, asking for specific colors and brands, mispronouncing things, and the DEA number isn't valid. See, when we get suspicious prescriptions like this, we have to get the doctor involved and sometimes the police. So as you can imagine, this takes up a lot of our time.
The next station is the pickup station. This is where you pick up your prescription and pay for it. Pickup, we ask for identifying factors when the guest comes up, such as a name, date of birth, phone number, and an address. Did you catch that? Let's listen to it again. At pickup, we ask for identifying factors when the guest comes up, such as a name, date of birth, phone number, and an address. And this is also where we collect the payment for their medication. Lastly, if anybody wants to purchase any products containing pseudoephedrine or Sudafed, they would ask us what they want, and then we get it here behind the counter. Lastly, we'll send a guest that has any questions over to counseling. The last station is a counseling station. This is where the pharmacist provides important information about your medications, as well as answer any of your questions.
Okay, there's 3 steps to remember when you're at the patient consultation. First, stay engaged with the pharmacist because we're focusing all on you. Secondly, let the pharmacist know if you're confused about it because remember, we're talking about your healthcare. And finally, don't be shy. There are no dumb questions, so ask away.
That's all, folks. Thank you for exploring the pharmacy with us today.
Okay, so that was like, that's what we do. Like, that was the back part of pharmacy. What do you get? First of all, Jope sir, really sorry. Like, I know I'm taking too much of your time. I'm almost done. Last thing what we do is like, patient satisfaction. After you do all this thing, yeah, your 95% of the people would be very much satisfied for sure. It's really important to treat each patient best way every day. Patient satisfaction and experience must be the priority for sure. From the moment they check in to the moment they check out, friendly faces will calm them down. Everybody's sick. They are not coming to buy burgers or chips or whatever. They're all sick people. They want to see like happy faces. You cannot be like, mad. You cannot get mad at them for sure, for whatever reason. Okay, this thing I don't want to explain. Like, this was what I did, like, to reach wherever I am. Lot of things. Like, I personally have given, like, in last 2 years, 30,000 people, like, COVID boosters, COVID immunizations, and me and my team have given at least 1,20,000 से ज़्यादा लोगों को immunization दिया है. I have really good relation with state of Illinois people. Like, so I was involved, like, together, at least 2,000 people, and we give everybody, like, in one day, the booster shots. I was the one who was involved drive-through pharmacy में. So like, and I also help like, Walgreens in buying out, like, कि किसी को अगर business बेचना है, so I help like, Walgreens to buy out, and I work in hospitals as well, or set up like, different locations. That's what I do. What it has given me, I was there in news media, like, this was the lady I'm talking about. She was working with me. She's just 48 years old. She's already retired. She's a pharmacist. So we did like, a clinic together, and that's why, like, I think I was in the news media as well. This is the drive-through pharmacy, like, I was talking about. Like, I was involved in a lot of drive-through. Like, people just sit in their car, they don't have to get out of their car, and we will swab their nose, we'll give them the swab, and they will swab on their own. and we will be doing the test. So that's my role as well. Like, I'm a director for like, all the COVID testing center as well. Like, at my place, I'm the director there. So I have to sign. Like, if somebody's positive, without my signature, it's not gonna get reported. It gets to the state, and that's how like state and federal government used to keep a track on all the numbers. So I was taking care for my store, not the entire nation, but the place I am the director at. So I was taking care on that as well. That was my last slide. I'm sorry if it was too boring. But yeah, like, this is my information. You can reach me out. My email is bamik_thacker, bamik_thacker at hotmail.com. I'm there on WhatsApp. But as I said before, like, I might not be responding you right away. I'll try my best to respond you whenever, like, I have chance. But you can note down my number as well. +1 is the country code 630-677-9006. I'm also there on LinkedIn as well, so you can reach me out there as well. Alternative resources, doctors, nurse practitioner, Dr. Angari, like, all the professors, you can reach them out for any other questions. So that is all about me. Any questions? Anybody? I'm ready to take the questions.
Yes, sir. Thank you so much. Thank you, sir. Now we will start. Am I audible to you?
Sir,
Yeah, thank you,
can
sir.
hear you.
Now we will go ahead and address the questions in the chat box. And there are few questions asked by the participants. First question is from Pawan Patidar. Question is, how much budget we have to start a pharmacy shop in America? Thank you, sir.
We cannot. Like, this big company, Walgreens, CVS, they are big giant companies. Like, the business I take care, at least बहुत ज़्यादा investment है. And it's like 3 or 4 pharmacies are there, as like, the first picture said. So like, all our pharmacies are owned by those companies, Walgreens, Walmart. We cannot start our own pharmacy, unfortunately. Hope that answers your question.
Sir, we have another question from Dinesh. How an Indian pharmacist get entry in US as a pharmacist? What is the procedure exams to get entry in US?
Exams, like, I already showed in the previous slide, like, 5 different tests you have to take. To enter, like, you have to just apply for the visa. We do not sponsor anybody. Like, you have to apply for the visa. If you get approved, yeah, welcome. I'm personally hiring a lot of people, so approach me. I'll hire you for sure, guarantee.
Yes, sure, sir. So we have another question from Dr. Anuj. Elaborate the role of community pharmacist.
There was like, a couple slides I showed, like, community pharmacist, hospital pharmacist is the other term, retail pharmacist is the other term. The role of my pharmacy, like, it was explained. So I can forward the slides just so I don't take, like, too much time. I know I've spent a lot of time and there's like more lectures to come. And I'm sure like he's preparing, he's thinking. I will forward, like, all the information which I do, all my duties, I will share it with you guys for sure.
Okay, thank you, sir. There's another question asked by Jay Pagare. What is the procedure after B Farm in India, and what will be financial requirement? Thank you, sir.
Financial requirement, there is none. Like, we Indians, we are smart people. This is what is needed. Like, just the brain is needed. Financial requirement, none. Just like, and America is very welcoming country. And we have way better scopes, like, here for pharmacists than any other country, whether it's like Australia, New Zealand, Germany, Canada, Mexico. Pharmacy in America is very much advanced. You get more money. And we are hiring for sure. Like, that's what I can tell you. No financial need for sure. But yeah, just talent.
Sir, we have next question from Ankita Pure. Sir, is there no facility like OTC medicine in US?
There is, like, a lot of medications, like, whatever diagnosis मैंने एक स्लाइड दिखाया था जहाँ पे डायग्नोसिस की ज़रूरत नहीं होती है, तो like, कि those medications are available without prescriptions. those medications we can recommend because a lot of people they don't know which one to use. So those all category, like I shared in previous slide, you can all prescribe those medications to them. They cannot buy. You have to explain basically which one is better over which one. So you have to explain and they will buy it from you.
Sir, another question is from Sakshi. Could I apply through SAT for the further studies in pharmacy?
To be honest, I don't know what is that. If you guys have any information you can provide me, and probably I will try my best to answer that question. I don't know what is that.
Okay.
Okay. Sir, another question is from Nisha Shrikoli, sir.
Any age limit to give these equivalent exams?
No age limit. Like, I have a lot of friends, like, they became pharmacists. Like one of my Italian friend, she moved from Italy. She is 52 years of age, and she was studying. As I said, like, there's very few people, like, who becomes like, pharmacist and practices. She became pharmacist at the age of like 52. She was studying with me. She just recently, I've been practicing pharmacy for almost 4 or 5 years now. She just recently became pharmacist. So there's no age limit. Even 60 years old can, like, study and can work as a pharmacist.
Okay, thank you, sir. It looks like we have covered most of the questions now. And
Mr. Host, this is Santosh. Can I ask question to Mr. Bhomik?
yes, sir, you can ask, please.
Yeah, Mr. Bhomik, good evening. This is Santosh from a student of Yash Institute of Pharmacy. Currently, I have engaged with the research and development with Torrent Pharmaceuticals, Gujarat, India. As I seen your complete session, it is really so nice and helpful to the students as well as the practitioners engaged with the different organizations. So I would like to ask two questions. First, I seen in the video that the doctor directly prescribed or mail to the pharmacy that medicine how to prescribe to the from the pharmacist. So I little bit confused whether doctor prescribe write diagnostic notes or the directly prescription to the pharmacy through the email which is that the patient has received.
Can you repeat your question? You can repeat in Hindi as well, perfectly fine. So I understand your question. I want to make sure I answer your question correctly.
Yeah.
Can I ask a little bit?
I seen that the video the doctor has the email to the pharmacy.
Mm-hmm.
That email was the prescription or the diagnostic notes to the pharmacy.
Okay, so like whatever I've understood your question, yeah, like prescription will have what the patient has been diagnosed with. For example,
Yeah.
if a patient had like
Okay.
fracture, and they will
Okay,
prescribe like drug.
I got my answer. I got my answer. Yeah.
Mm-hmm.
Second thing, I engaged to the develop the dosage forms with the multiple units like thousands counts, 500 counts, and I seen the video that the pharmacy has dispensed the medicines from the large bottle to the required amount. In between, if you are dispensing the two or more than medicines, what care you are taking to avoid the cross contaminations? I think that is the procedure written here, but I still not understand while developing and researching with that department.
Contamination, to be honest, I don't know, like how many medications are gonna get contaminated. They
No,
all come
while
from India.
dispensing the multiple dosage,
Uh-huh. What's your question on that?
while dispensing the multiple medicines, example, you have medicine, three medicine prescription in the
Mhm.
medicine, three medicine in the prescription. You dispense first medicine from the 1,000 tablet bottle, and second from the another 1,000 tablet, and third another thousand.
Mhm.
As I see, you have that pharmacies have dispensed 2 or 3 times. In between the dispensing, there should be the some care to avoid the cross contamination. So I would like to understand from you.
Which kind of contamination?
Bhaumik, can I intervene and make the question simpler for you?
Okay, thank you. You can proceed.
So, Bhaumik, can I make a question simpler for you?
Sir.
Same questions what the candidate is asking. So the question is that if you have the container and which contains the multiple tablets, say, and it is written in your pharmacy shop, and if patient, suppose say 1,000 tablets are there in single container. So if the patients want 10 tablets, okay, and you are opening that container and then taking the 10 tablet out and providing to the patient, something like that. So during this process, so you are opening the container and you are taking it out and something like that. So environment your tablet get affected by the environment. So the question is that how that cross contamination is being taken care. So the question is right.
Correct.
Yeah. So Bhaumik, is a
Like,
very simple question.
yeah.
Yeah.
So like, see, the thing is like we never like I will answer the first question. We never dispense 1,000 pills at the same time. Maximum quantity we dispense is 90 days supply, 3 months supply. So prescription is gonna be either 30 pills, 60 pills, or 90. When we are done filling, like one medication, for example, atorvastatin, we will close the lead, like we'll put it back, and then only like we seal it, we put the label and everything, then add another medication. So that way, like lot of open bottles are not there basically.
Yeah.
Make sense? Chances are
The
very
answer
less,
is,
like
so the answer
contamination.
is that there is no any practice in the media. There is no such type of practice in the shop where the bottles are get opened and prescribed to the patient. So that is right. So there are
Very few people will do it, but again, we are not dispensing sterile drugs. These are
yeah,
all like oral pills.
yeah,
Like sterile like happens at the hospital pharmacy. They
right,
use different practice. So like that question will be implemented at hospital for sure. Like sterility will be a huge role. But at
Correct.
retail,
yeah.
mainly like injections, they are pre-boxed. Like insulins, they are pre-boxed. We don't have to open that. Pills, like they are not sterile products up to
So
certain
that could,
extent.
yeah, so I can assist to answer this question again. That what you exactly said is correct. So in case of the sterile preparation, so there is no chance or there is no way to open that particular container. However, in case of the oral, yes, in up to certain time or certain extent, that could be get open, but that are likely get affected in a very less extent
That is true.
because of the environment, and even the oral prescriptions are very less likely affected by the microbial contamination. When you
That is
see
correct.
the microbial contamination in case of the sterile preparations, the chances are very high. So there is no scope in case of the sterile, but in case of oral, the chances are very less. So
Absolutely
if
right.
yeah, so that is the answer for this. Yeah, Dr. Abdul is wanted to say something. Yeah,
Can
Dr.
you hear
Abdul, go
me?
ahead.
Hi, can you hear me?
Can hear
Yeah,
you.
very well.
Great. Uh, the speaker's name is Tucker. Is it okay if I call you Tucker?
Exactly fine. I was embarrassed, like whenever everybody was calling me sir. I prefer you guys to call me by my name.
Yeah, that's good. So the question which has been asked is about there is any contamination. So what sort of contamination are we looking at? That's a rightly put question by Tucker. There's no contamination as such when you're using an oral preparations.
Right.
Depending upon the type of medication, there are certain medications where the atmosphere, exposing to the atmosphere becomes an issue. And the second point is it is dispensed in a way you wear gloves and all, and probably the ones which do have an effect by the atmosphere or put in a container where they're not exposed to the light or atmosphere. When it comes to the hospital services, as Tucker rightly said, the sterile preparations are prepared under the wood. And most of the doses that are supplied to the patients are unit doses, which are wrapped in a automated mission. So probably that doesn't arise. So this is all policy driven. So every hospital has a policy where when you are dispensing all these medications that need to go to the sterile, if it is an oral. And let me emphasize on oral liquids, which are dispensed in hospitals and inpatient. What happens is we develop a policy based on the evidence that is available. So if you open the oral liquids, you should not keep it for more than a month. Means the expiry for an oral liquid would be for 1 month. So these are all policy driven. They're all taken care. Is that okay?
Yes, sir. 100%.
Thank you very much.
Thank you so much, sir. It looks like we have covered most of the questions now. There are many more questions, but due to the time limits, we will not able to address them. So once again, thank you so much, sir. I would like to thank Mr. Thakkar, sir, for this informative session. Now I would like to invite Prasanna to propose the vote of thanks. Over to you, Prasanna.
Thank you, Arad. I deem it a great honor and privilege to propose a vote of thanks. Let me first give a glory to Almighty for making the first session of webinar a grand success. I thank our special guest, Mr. Bhaumik Thakkar, sir, pharmacy manager in Walgreens, Illinois, United States of America, for spending his valuable time with us. We are grateful for our principal, Dr. Sachinanand Angadi, sir. His able guidance has always encouraged us. I thank him for his continued support. My heartfelt thanks to all the faculty, organization committee members, and technical committee members for their contribution, guidance, and encouragement in all our efforts. I thank all the distinguished invitees present here and accepting our invitation. Last but not the least, I thank you all, my dear friends, for your cooperation in making this function a resounding success. Once again, thank you all your attention. Thank you. Over to you, Varun.
Thank you so much, Prasanna. As a token of appreciation, sir, I request you to accept the appreciation certificate on behalf of Yash Institute of Pharmacy, Aurangabad. Thank you, sir.
Thank you again. If any question, don't hesitate to reach out.
Now, I would like to request Aniket Pawar to start the next session. Over to you, Aniket. Thank you.
Thank you, Varad. May I audible?
Yes.
Okay, thank you.
Yes, you are audible.
I am extremely delighted to introduce our next resource person, Dr. Murlidhar Zope, sir, Associate Vice President of R&D Analytical Development, Sun Pharma, Vadodara, India. Myself, Aniket Pawar, studying in the third year B Pharm, Yash Institute of Pharmacy. Now it's time to introduce our honorable guest, Dr. Murlidhar Zope, sir. Sir has extensive experience of 25 years in the pharmaceutical industry, working with the prominent organization in the research area like a Sun Pharmaceuticals, Glenmark Pharmaceuticals, and the Fen Care Pharma Limited. His core expertise area of research in the analytical research of novel drug delivery system, specialty of conventional formulation of various types of the pharmaceutical dosage forms to support a new clinical entity, that is NCE, new drug applications, that is NDA, and abbreviated new drug applications, ANDA. Sir has completed his B Pharm and M Pharm from the Pune University and awarded a PhD for his work in the area of impurity profiling from the various complex drug products. Sir conducts research in the area of various nanotech platforms and complex specialty formulations from proof of concept to functional characterization
and a quality speciation include the microbiological properties. Dr. Muralidhar Zope sir lead the various analytical teams for characterization of the novel drug delivery system product and especially formulation in the area of analytical method development and validations, setting speciations, in vitro bioequivalence, non-clinical pharmacokinetic studies, microbiological evolution, along with the extractable and leachable studies as per the regulatory agencies across the world. Now, I would like to request Dr. Muralidhar Zope sir to start the session.
Yeah, so thank you very much, Aniket, for your exciting introduction for me. First of all, yeah, I would like to highlight some of the points which I have taken from Bhomik presentation. Okay, so Aniket, are you able to see my screen?
Sir, yes, sir.
Very good. Okay, so very good session by Mr. Bhomik on what the pharmacy practices and all this is happening in the US, and people who are on the call for providing them the opportunities and finding the success in a few of the areas. So I would like to highlight some of the areas. Actually, you might have seen the rules and regulations. So up to what extent the rules and regulations are there for prescribing the medicines. And one of the best quote from Mr. Bhomik is that 80% of the medicines are coming from the India. So that is a very big a note for the India. And you believe that if 80% of the medicines is coming from the India to the going to the US, so our responsibility um, is to get this medicine in a quality way. If you are violating such type of medicines in manufacturing area or quality area, so the rules and regulations in America are very tight. So you can consider this topic is very important for you guys, dealing with impurities. So how you will deal these impurities if certain impurities are beyond their controls or something like that in the pharmaceuticals. So what regulatory is nothing but in USA, the US FDA, that is United States Food and Drug Administration. So how they are taking the actions on that. So we will come to this in this session. So what we are going to see in these sessions are what are impurities, how these impurities are getting generated. So from where these impurities are getting generated. Then why there is a need for identifications and qualifications of these impurities. So what is the structure of detection, this identification, quantification, and qualification of these impurities? And finally, the regulatory expectations and implications. So first of all, I will go through the regulatory implications of violating this presence of the impurity in the drug product. So you will come to know that means how we have to apply the rules and regulations on that particular impurities. So as a quality perspective, FDA inspect the drug substance and drug product. So what I am saying is FDA. FDA means any regulatory agency in the world. So there are different names for different regulatory agencies in the world. For example, in case of America, it is US FDA. In case of Europe, it is EMEA. In case of Japan, it is a PMDA. In case of India, here we can say FDA again, Indian FDA, something like that. So these are the different agencies having the different names, and they are inspecting your drug substance, means API manufacturing or drug product manufacturing facilities. So what they are doing, so they are evaluating the quality of the products. So and during evaluation, the most critical at. So anybody want to ask anything? Nikhil, Sonone. Hello.
Sir, your voice is not coming.
Yeah. No. I have disturbed if there is something questions for me or something like that, because someone is talking in between.
No, I'll just mute. I'll just mute everyone.
Yeah. Okay. So the most, thank you. Most critical attributes of the inspection is related to the impurities only, because impurities are very, very hot topic in the industries. Insufficient response if agencies are finding any inspection, any point during the inspections, and they are, in case of the America, so they are prescribing your that questions in the form of 483. So in the form 483, you will find most of the questions. So you have to answer when the facility get inspected, and that leading to the warning letter. And if you are not providing sufficient answer to that warning letter, so your manufacturing unit get shut down. So that is the ultimate effect. Why? I mean, that is the ultimate effect of how the regulatory agencies are putting their efforts on the inspecting the facilities. So what are these different major deficiencies that agencies are getting observed? So absence or deficient discussion on the risk of having potential mutagenic impurities in the final product. So for example, there are certain anti-cancer drug or any immunosuppressant drug and any other heart disease drugs on the heart disease or something like that. So if you are not putting your discussions, or you are not putting your explanation in the filing on the mutagenic impurity, so that could be the one of the major deficiency in the regulatory submission. Then absence or insufficient discussion on fate and carryover of related substances of starting materials to the final substance. So they, when you are manufacturing, say, the paracetamol or any drug, so there are many materials which are acting as a starting material. So there are certain impurities which are present in the starting material. So you should have discussion on that particular things in the submission. Then non-adequate or poorly justified specification in place to control of the quality of the starting material. Since just what we are seeing as the starting material is very, very important. So the quality or specification, so you must have something specifications in hand to control your starting materials. Then non-adequate or missing specification for reagents and solvents, so that are being used to manufacture the drug substances from the introduction of starting material. Again, this is the reagents and solvent. These are various reagents and solvents which are making the processes. So you need to have control on that reagents and processes also. Then non-adequate or missing discussions on carry forward of reagents and elemental impurities to the final substance. So it is also very important. So during the processes, you are taking the help of equipments which contain the different type of materials. So you need to assess the elemental impurities in your drug product. Absence or insufficient discussion on fate and carry forward of impurities from synthetic intermediate to the final substance. So there are certain cases or reactions in your manufacturing of the drug substance or drug product, where up to certain level of the synthetic impurities are getting formed. But if you are not getting remove all the impurities from your drug substance or drug product, so that, and if you are not adding this as a discussion or writing the justification to the regulatory agencies, so that could be one of the potential risk for the observations. Then again, this is the type of question, just I am reading and then giving the explanation that what is this question actually. We acknowledge your proposal to establish limit for newly identified impurities, including proposed structure identification. Please be aware, we will not grant this CBE30 until you have provided a QSAR bacterial mutagenicity assessment in line with ICH M7 principle, which demonstrate unequivocal negative bacterial mutagenicity, which we consider critical to establishing the risk of newly identified impurities. Please provide this information in support of your proposal. So what do you mean by this CBE30? CBE30 nothing but a change being effective 30. 30 means 30 days. So this is the product. This may be the one of the product which has been marketed in the United States of America, and some organization has been filed is CB30 that something is getting introduced on a product and we wanted to establish their structural identifications and qualification. But what agency is asking that you please provide a QSAR, that is quality structure activity relationship bacterial mutagenicity assessment. So there are different ways that how you can able to assess this mutagenicity of particular impurities. So that need to be demonstrated along with this CBE30 form. Then only the FDA get grant your application to monitor these impurities. Then the next is the company lack to establish and document the accuracy, sensitivity, specificity, and reproducibility of the test method. So test method is also very, very important in identifying and qualifying qualifications of your impurities. So if you see, this is the trend of the last 3 years, where most of the organizations has got deficiencies from the agencies. So you can see that impurity qualification is the major area, what I told you earlier, where there are a lot of observations. So even at the first observation, you see 199 observations are there. Even the organizations are trying to answer the 199 observation till 97 observations are repeat and not doable. So in such cases, so there may be a chance that the organization may get a warning from the FDA that your company does not have sufficient standard to qualify these impurities or something like that. So you can take the note of this, that how serious this impurity qualifications is there related to the API and the drug products. So what is the need of studying impurities? So there are two type of need. One is a regulatory requirement and second is the scientific and technical requirement. So what are the components of the regulatory requirement? That you need to establish the quality and safety of the product. Then acceptance criteria determination. So whatever you are determining the impurities, what could be my acceptance criteria? Then expiry date, retest date, and shelf life evaluation of the drug product. So that is also again one of the criteria need to be met for the as a regulatory requirement. Then stability and storage conditions should be well defined for the drug product. Threshold limits evaluation should be well justified. Then method validations. There are a lot of parameters we will see in the next slides, and that what parameter need to be followed for as a regulatory requirement. Looking at the technicals and scientific requirement, the synthetic and production process optimization. So that need to be explained very well during your manufacturing of the API and drug product. Then efficacy improvement, that how you are improving your product while decreasing the impurity content or something like that. Then ADME and toxicology study need to be properly justified in the submission. Then stability improvement, and that how you are improving the stability once if you find that there are a number of impurities which are generated in your drug product. So how you can able to improve your stability of the drug product. DRIs and pathway predictions. So there are different drug interactions and different formations of your impurities are there. So their pathways need to be predicted properly, and how you will control that pathways. You control the processes in that pathways that need to be understand, and that need to be taken into account in the scientific rationale to the any agencies. Then what are these different impurities? So major categories of impurities are the organic impurities, process and drug related impurities, inorganic impurities, residual solvents. And what are the other type of impurities are polymeric form of the impurities, then enantiomeric impurities, leachable impurities, nitrosamine impurities, and extraneous contaminant. So all these impurities that nothing but they are not having the any therapeutic value. And you everyone in the call actually have very well definitions of this impurity that I'm not going to repeat it here. Then these are the sources of impurities. So from where in the processes how these impurities are getting generated. So coming from the API starting material, then your reaction to the reaction intermediate. During this process, so these reagents, ligands, solvents, and catalyst, then starting material impurities and byproducts. So these categories of impurities are coming from this stage. Then during the next stage to the final API, so some of the byproducts and the reagent, solvent, and catalyst here also. So in this stage are getting introduced in your API. And once the API is getting formed, so their inherent impurities as a degradation impurities, as well as the polymeric form of the impurities are present in this API substance itself. From the API, when you try to make the formulations, the API, you need to add the excipients in the processes. So excipient and API interactions is taking place from where the impurities are getting generated. And during from this API plus excipient with the different processes, you are forming the drug product. So in the while formation of these drug products, so these processes excipient impurities, because excipient API interactions may get happened. So excipient impurities are getting generated along with this solvents. Solvents are also being utilized in the different processes of formation of the drug product. So these solvents are also acting as impurities in your drug product. So final outcome is a drug product which we are selling in the market. So and again in the drug product also depending on the processes, so how much temperature, how much humidity, temperature, pH, something. There are different parameters so that are being taken place during formation of your during processes from the API to the drug product. So in that processes, your drug API in the drug product get degraded as inherent impurities. And along with this, once your product is get in the drug in the container. So what is meant by CCS? CCS is nothing but a container closure system. So drug product in the case of the packaging material, the packaging material get also impurities from the packaging material get introduced in your drug product also. So from starting of the API material, so starting material of the API to the drug product, so all these type of impurities may get introduced in your drug product itself. And we should be cautious and curious, and we should have a lot of effort to get nullify these impurities in below their threshold limits. So we will see in the next slides actually how these impurities get controlled, what are the control limits, reporting limits, thresholds, or something like that. Then these are again some of the impurities which are being classified on their types basically. So there are organic, inorganic, and solvent-based impurities are there. Yeah, again, inorganic degradation impurities, process-related impurities. So basically, we have seen these impurities in previous slides. Inorganic impurities are heavy metals, reagent, catalyst, and other impurities are there. And the solvent-based impurities are organic solvents and inorganic solvents. So somewhere I will take it fast because I don't know whether time will get permitted for me to complete all the presentations. So I would like to have some interactions with you guys. So in order to get or enhance your knowledge on this particular topic. Then coming to the organic impurities, when you see here, these are getting arise during the synthesis, purification, and storage of the drug substance. Then residual unreacted starting material, and that is also carry forward impurities as organic impurities. Synthetic reactions like hydrolysis, acetylation, and decarboxylations are being taken place. So during this unreacted materials getting carry forward, then can generate during storage of the product due to different interactions also what we have discussed. Then some of the enantiomeric impurities, genotoxic impurities, and nitrosamines, and these are also impurities as a categorized as a organic impurities, where you can able to see in case from this is the starting material for paracetamol, para-aminophenol is the starting material, then it is getting formed as a paracetamol. So if this group is getting reacted here, and this is not getting completed to the paracetamol, this group is nothing but an impurity in your drug product as categorized impurity as a byproduct without and having the therapeutic value. coming to next organic impurities, so it is very interesting one. So this impurity is already a impurity in one of the impurity in your drug product, but in certain cases you might require the residual solvent to be utilized in your manufacturing process, or this residual solvent may be a contaminant from the environment. So these residuals as a so for example, this is a formaldehyde. If formaldehyde is attacking on the amine group of your impurities, so there is a formation of different type of impurity, which is amine impurity. So from amine it is getting formed to the amine impurity, and this impurity is very toxic in nature. So we need to control these impurities by bacterial immunogenicity test, and that need to be assessed properly and need to be submit in the regulatory dossier. So these are the challenges. Some of the this is some of the challenges which you may encounter during your drug product stability also. So these are the inorganic impurities. These are very well established in the literature. So some of the pharmacopoeia has taken these impurities in different classes, that class 1, class 2, class 3, and other inorganic impurities. So there are certain limits also for inorganic impurities, how this has been controlled in your drug product. So examples of the class 1s are arsenic, cadmium, mercury, and lead. And then class 2 example is the cobalt, nickel, vanadium, silver, etc. Then class 3 is the copper, lithium, barium, and other impurities are aluminum, boron, and calcium, etc. So when you see that how these are being classified as a significant safety concern to the low inherent toxicity. So accordingly, they have the pharmacopoeias and different guidelines have prescribed certain limit for controlling these impurities in the drug product. These are the residual solvents. Most of the residual solvents are being utilized in the manufacturing of the APIs, and some of them are also required to be seen or in the different processes while manufacturing of the drug product. So again, they are classified as a class 1, class 2, and class 3 solvents. So class 1s are known human carcinogens, toxic and environmental hazard. So like the benzene, carbon tetrachloride, and 1,2-dichloroethane, 1,2-dichloro, 1,1-dichloroethane, and 1,1,1-trichloroethane. These are the different solvents that are human carcinogens. Then class 2 is a non-genotoxic but need to be controlled with their PD limits, which are prescribed in different guidelines and pharmacopoeia. PD nothing but as this permitted daily exposure. So for the according to that and taking the different formulation maximum daily dose, so the limit is being prescribed in different guidelines and the pharmacopoeia. So class 3 is then acetic acid solvent. So there is one limit for the class is the 0.5%, or you can say the 5,000 ppm in any of the drug product or in API. So that need to be monitored as a residual solvents. Then these are the enantiomeric impurities. So when you see this structure, so it is a mirror. Enantiomeric is nothing but a mirror image of each other. So if one drug is more effective, however, the mirror image of that particular drug is less effective, even though their elemental composition is same. What I mean to say, the elemental composition is carbon, hydrogen, nitrogen, oxygen. That if everything is same and even only the mirror image is changed, then you need to monitor their presence in the drug product. For example, in case of ibuprofen, the S plus form is the more therapeutically pharmacological active, and R minus form has no inflammatory effect. So the agency will ask you to control this R minus form in your drug product. So in case of dextroamphetamine exhibits more pronounced effect on the CNS than levoamphetamine. So the content of the levoamphetamine need to be monitored in your drug product. These are the just examples I am giving you in front of you people, but this is the story for each and every product. So the systematically you need to monitor all type of impurities. These are the genotoxic impurities. These are very, very important impurities for regulatory agencies, because these impurities may get affected and the form the mutations, therefore these potentially causing the cancers. So these impurity, how these are being monitored in the drug product. So if your drug product is for say per se, it is more than 10 year to more than 10 years. Suppose the patient has to take the medicine more than 10 years. So you only permit 1.5 microgram per day. Even though if you are taking the 10 tablet, only 1.5 microgram per day is permitted. So if your daily dose is 1 gram, and your daily intake is 1.5 microgram, so you calculate 1.5 microgram divided by 1 gram and multiply by 100, so that much percentage may be coming 0.00 or something like that. So that much amount of the percentage to be monitored in your drug product, that is the requirement from the agency. So however, if your medicine is within the 1 to 10 year of use, then the intake is increased, because it is permitted to 10 years only. Then again it is 1 to 20, then it is 20. Then if it is less than 1 month, then it is 120 microgram per day. Even though it is 120 microgram versus the your drug therapy may be 1 gram, so you can calculate, and that much percent only allowed in your drug product. So we need to take care of all these facts. And if it is crossing more than this, so there are various structural tools which are available. That is called as a QSAR, DIREC, and TopCat. So these are the models where you can try to establish their safety potential, depending on their whether they will be more mutagenic or less mutagenic, or depending on their main molecule. Then this is the call that we need to take from the structures, whether my drug is there, and if something is getting formed from that drug and having such type of groups. So this type of groups are normally giving the alert for the genotoxicity. So we should be curious about if my compounds are having such type of structures in it or not. Then nitrosamine, so these are the compounds, the structural groups of very potency mutagenic carcinogens. So I will show you that what are these different structures, and if these compounds are present, so they are always present in the group, then classified as a probable or possible human carcinogens. So that's why this need to be monitored in your drug product. Then again, nitrosamines are formed by chemical reactions of secondary or tertiary amines with nitrites under acidic condition. So if during your process, if you have such type of likelihood process, then you need to take care of this. So these are the class of nitrosamine impurities which are really need to be monitored in your drug product. Then leachable, this is very interesting example. When you are suppose say your drug product is there in the packaging material, and your drug product contains different preservative. Here in case, here you can say this M cresol as a preservative. If you have used this M cresol as a preservative in one of the medicine, and that medicine is in the glass vials with the rubber stopper. However, in the rubber stopper, there is a bromobutyl group which is present in the rubber stopper, that is getting reacted with your preservative, that is memcresol, is forming the 2,4,6 tribromo 3 methyl phenol as an impurity. And this impurity is of major concern, and such type of impurities are also getting monitored in your drug product. So even this is not from your drug substance itself. If your drug substance is something here as you can say cefuroxime or something like that, so this impurity is from the excipient, not from the drug substance. So such type of impurities are also very, very important of concern that need to be monitored in your drug product. So these are the some of the areas where how you predict impurities. For example, these trace and stability studies of the drug products, you can come to know that something is coming in my product. Then degradant related impurities are also there in the drug product. So you can see on the different processes. So where my processes are getting affected, where I am getting this type of impurities. Again, degradation impurities. So these are the tools where the acid hydrolysis, base hydrolysis, different type of hydrolysis are there. So you can take the help of these impurities in finding out the degradation pattern of your drug substance. Then excipients, what we have seen the interactions, and how you will identify these impurities. So there are different tools that we are going to cover in the next slides. Then this is very important slides, these 3 to 4 important slides. So if what is the reporting threshold? So how what we have discussed till now, that is the nature of the impurities, what type of impurities are present in the drug substance or drug product, and now we are talking about how you have to deal these impurities, means how you have to control, means what are the control limit for drug substance and drug products. When everything is in control, then regulatory agency does not ask you much more questions on this. If it is beyond this control, then that is our role, that how we can reduce it to safety concerns, or do you have any explanation for this? So the first threshold for this impurities is the reporting threshold. Suppose, for example, if my this is for the drug substance and this is for the drug product. So if my drug substance is less than 2 gram and more than 2 gram per day, then you can report this is more than, if your impurities are getting more than 0.05%, then you have to report this impurity in your certificate of analysis. And if it is more than 0.2 more than 2 gram, then more than 0.03% need to be report on your certificate of analysis. So in case of the drug product, so this is the drug substance. Say I can put examples here, simple example, what I can say, paracetamol, paracetamol API. So here paracetamol tablet, you can say, for if it is less than 1 gram per day, then 0.1 more than 0.1% need to be addressed on the certificate of analysis. If it is more than 1 gram per day, then more than 0.05% need to be monitored in your drug product. Coming to the identification threshold. So what we have seen is the reporting threshold. Reporting threshold मतलब आपको खाली उसको रिपोर्ट करना है सीओए के ऊपर. While identification threshold means इसके ऊपर सपोज जाता है तो आपको वो क्या है, what is that chemical moiety, that call need to be taken here. If it is less than 2 gram per day, then if it is more than 0.1% or 1 milligram total daily intake, so that is the call we need to take, that what is that chemical moiety which is coming more than 0.1%. So if it is more than 2 gram per day, then more than 0.05% you need to identify it. So likewise, if you see in the drug product, so if it is less than 1 milligram up to 1% you have permitted, or 5 microgram per day it is permitted, then these are the different limited you might have seen, you will seen these impurity, these limits in the different guidelines also. Just to understand you guys, that I just highlighted you, these are the different ranges and these are the different limits, and these are the different components. Means this is, suppose if your drug product is per se the drug substance in the drug product is within this range, then more than 0.5% you need to identify this particular impurity, or 5 microgram per total daily intake. So whichever is lower, मतलब ये इसके ऊपर ये 0.5 कंसीडर करना है, और इसके ऊपर से जो परसेंट आता है उसमें से दोनों में से जो परसेंट कम आता है उसके ऊपर सपोज आपकी इम्प्यूरिटी रहती है, तो उसको आपको आइडेंटिफाई करना है. So how you will identify this impurity? On this different, with the help of these different techniques, LC, UVPDA, LC, MSMS, and GCMS. So there are different hyphenated techniques which are available to identify this impurity. And these are the limits where if यहाँ तो आपने आइडेंटिफाई कर लिया कि भाई वो स्ट्रक्चर क्या है, लेकिन वो स्ट्रक्चर आपने आइडेंटिफाई करने के बाद भी वो सपोज इसके ऊपर जाता है, then आपको क्या करना है? So these are the limits where if your identified impurities are crossing this limits, then you need to qualify this qualify that particular impurities. For example, less than 2 gram 0.15, यहाँ पे आप देखेंगे 0.10 था, यहाँ पे आप देखेंगे 0.15 है, थोड़ा सा लिबर्टी आपको क्वालिफिकेशन के लिए दिया हुआ है. So these are the limits where these are also the limits which are available in the guidelines. You can study afterward. So इसके ऊपर जाने के बाद suppose 0.15 के ऊपर जाने के बाद मैं क्या करूं? So you have to qualify these impurities. So what are the different ways of qualification? So qualifications is nothing but comparative studies you need to do. Suppose if you are making the generic drug, what in the previous lecture, what Bhomik was talking about, 80% of the drug we are selling to America from India. So what are these 80% of the drug? Out of 80%, I can say there are 75% of the drugs are the generic in nature. So what are the generic drug? Generic drug are nothing but a drug you are making or copying something else. So that from where you are copying from the RLD. RLD is nothing but a reference listed drug. So if you can able to qualify your impurities as the same impurities and up to the same extent in the RLD, then the agencies may agree your proposals. Otherwise, you need to take other routes for the qualifications. So this is the active metabolite. If that impurity is active metabolite, then again you can give the explanation. There are certain scientific literature. If there are certain scientific literatures are available, then you can take the help of that also. Otherwise, toxicity studies are the mandatory for the qualification of such type of impurities. Then this is a decision tree. If something is getting higher in your chromatography or analytical techniques, if for example, is impurity more than the identification threshold, what identification threshold what we have seen so far, then if it is yes, then if then it is a structure identify. If it is no, then no action you have to take. Means what does it mean? It is impurity more than identification threshold. If it is not more than identification threshold, no action to be taken. If it is yes, then structure to be identified. What we have seen with the particular limit. Then once your structure has been identified, and then you need to take it to reduce to the not more than its identification threshold. If you are not able to, if you will able to do this, there's no further actions is required. If no, then what we have seen is the toxicity studies are the ways to qualify these impurities on the any clinical routes. Even though even in clinical routes, if you are not able to justify this impurity, if then then you need to reduce this impurity to the self level, and if it is qualified very well, then there no need to do anything on that. So coming to this, if your structure is getting identified, and then if the human risk is there, then you have to assess the human risk. If human risk is there, then you have to reduce to it self level, and if it is not there, then the greater than the qualification threshold, if it is a greater than qualification threshold, again you have to reduce it to the qualification thresholds, and again the same cycle is there. So this is the decision tree actually, what how you can able to qualify your impurity which is present in your drug product. Then this is an example where I have tried to put it in a simple ways. So if your maximum daily dose of any drug substance is 500 milligram, so it is well within the 2 gram. So what will be the reporting threshold? It will be 0.05%. मतलब इसके ऊपर suppose आता है तो आपको COA के ऊपर आपको उसको लिखना है, and ये जो values है इसके ऊपर आता है तो then you have to identify the structure, and this is the values here you have to qualify this particular moiety in case of the drug substance. So likewise these limits are there for the drug product. So here you see for 500 milligram it is a 0.05 as a reporting threshold. Identification threshold is 0.10 or 1 milligram, whichever is lower. What does it means? This ये 1 milligram divided by 500 आप करोगे और multiply by 100 करेंगे, तो वो लगभग आता है आपका 0.5%. So 0.5% and 0.1%, whichever is lower. So 0.1% is lower. So you have to take this limit in consideration. So however, in this case also the same calculation will be applied. However, for if you take the drug product and your dose is 500 milligrams, so the limit get increase, because the agencies have given certain liberty depending on the processes and the interactions which may happen from drug substance to drug product. So it is from 0.1 to 0.2 and 0.15 to 0.2%. So likewise, again, you have to consider this 2 milligram depending on the maximum daily dose, and you have to calculate the amount. And once that amount is less, whatever the amount is lesser, that need to be considered for your qualification or identifications and qualification threshold respectively. So take an examples of the drug substance. So above table, this above table shows in the drug substance, while in the below table is showing in the drug product. So if it is your 0.044% is the impurity which is getting in your chromatography, and your limit is 0.05%, so no need to report on the certificate of analysis. However, if it is a 0.0963, and if your reporting threshold is 0.05, then you need to report report on the COA as a 0.10 as a round-up figure. However, in case of the known impurities, so this is identification impurities and qualification impurities are nothing but a known impurities. So this is, suppose impurity A, if it is 0.12 and your limit is 0.15. So this is the limit for your known impurities. However, for unknown, in case of this unknown, if suppose if it is 0.11, then you need to identify. However, for 0.15% is a known impurity, and you are getting 0.12%, so you need to report it. But action, what is the action? You have identified it, leave it. But if the impurity is 0.25% and which is more than your 0.15% limit, so you need to identify it. Is okay, you have identified it, but in addition to that, you have to qualify this impurity with respect to the 4 parameter what we have discussed as a comparative evaluation, toxicity, active metabolite, scientific literature, and toxicity studies. Then coming to the drug product, it is the same case depending on these values. So I am not taking too much time here to describe the drug product. I believe if anyone want to understand, then I can take it after having this session. Then how you control these impurities in your drug product. So these are the different controls you have to take. That input control, that you need to control these impurities at the quality of starting material, reagents, solvents, and excipient. Then process control, the different reaction conditions are there, purification points are there, critical process parameters are there, then intermediate controls, productions and storage condition, then process analytical technology. So the different processes are there where you can see from where these impurities are coming, then accordingly you can put your control so far. Then verification of quality. So from the specification, so there are certain specifications which are already in place. So you can refer that specification for the quality perspective as a verification point. So API and drug product quality specification, you can check whether how much impurities are putting in the specifications or not. Then process and degradation chemistry knowledge indicate necessary control. So what we are seeing here, all the input material, process, and the specification, we can able to control all the impurities in the drug product. So this is the next topic. So to control all the impurities, so you will require the analytical methods, the good analytical methods. So while developing the very good analytical method, so what different factors you need to consider during your development? So you need to consider this factor: method type, API and DP. API nothing but a active pharmaceutical ingredient, and then drug product. Analyte chemistry, what you want to quantify and up to what extent you want to quantify. Then what are the analytical techniques, then sample selections and preparation, specifications and regulatory requirements. So all these factors you need to take into consideration while developing the analytical methods. So we'll take one by one that factor. So so method type. So what type of method you are going to develop? So there are a lot of methods which are need to be taken for the development. But here, as a perspective of the impurities, related substance method is the one that you can take into consideration. So you have to develop the related substance method here. So API and DF. So what are the different type of sample matrix? So there are certain API. In case of the API, so whether it is a peptide, whether it is a protein, whether it is a normal chemical moiety, whether it is a chromospheric, whether it is a non-chromospheric, so all type of matrix you need to take into account for the API. And for the drug product, different dosage forms, so whether it is a tablet, whether it is a capsule, suspension, injection, what type of injection, there are different type of injections. So lot of information is available, so you need to gather all the information here for as a dosage form. Then composition again. So composition again, you need to take all the factors again. The surfactant, where there is a polymer is there, surfactant is there, preservative is there. So what type of pHs and all this you need to take into consideration. Then analyte chemistry. So there are certain parameters of the analyte, that is the whether it is a polar, non-polar, chromospheric groups are present or not, volatile, non-volatile. Then whether it is a I need to quantify anions or cations. So all type of these attributes of the analyte you need to taken into consideration. Then analytical techniques. So lot of analytical techniques are there. Here I have just a brief certain only. So apart from this, there are different hyphenated techniques are also available to quantify this impurity based on analytical chemistry. Then HPLC, GC, ICL, CMS, these are normally being utilized. Then selection of detectors. So different type of detectors are there. So which detectors you will take into consideration, that also need to be established. Then sample selections and preparations is a very important stage, because a lot of different type of formulations are there in the market, and depending on their different composition, so you need to have the sample preparation accordingly. So samples with multiple impurities as a worst case scenario. So you need to choose that the samples where the lot of impurities are present in the that particular sample. Then sample preparation depend on complexity of dosage form. So I can say there are different formulations are there, but depending on their composition, so again the complexity will get increase or decrease. So that type of dosage forms also you need to taken into account. So this is the criteria, that what is the sample preparations you are making should be simple as possible as it is. That anyone can reproduce the result, whatever the methods you are going to develop. Then sample cleanup without affecting recovery. So whatever the impurities, during the validations, whatever the impurities you are putting in the recovery studies, so that need to be established at different locations. So that also taken into account while developing the methodology. The specifications, coming to the specification, the standards and sample concentration depends on the specification itself. So if your specification are too tight, what is meant by tight? Tight means if it is less than 0.05%. Okay, so in that case, you need to calculate your analyte concentration, that is the main drug concentration, and depending on the specification, it will be get calculated. Then LOD and LOQ shall be established according to the specification only. So your method should be sensitive enough. Then regulatory requirement, that system suitability criteria, the resolutions for adjacent peak, tailing factor, reproducibility and precision. So many others are there that you need to taken into account. So regulatory expectations for method during the method development, as it should be sensitive enough, then it should be stability indicating, then it should be according to the specification, that reproducible or transferable. It is very important. In research, if you are developing the methodology, then it need to be transferred properly at different locations of any of the organization. Then method validation. So these all are the parameters which are to be required once you develop the method, that it should be specific, linear, accurate, precise, sensitive, robust, and robust. So we will discuss. So anyone has any questions on this? Then we will take during the interactions. Then quantification. So these are the instrumentations and these are the different processes where you need to apply during your quantifications of the impurities. The different techniques used on the nature of molecule. That LC with ultraviolet detector, PDA, photodiode array detector, then FLD fluorescence detector and conductivity detector. So these are different detectors to be utilized with liquid chromatography module. Then mass detectors are also there. And then gas chromatography for volatile components, semi-volatile component with the FID detector, GC with MS detector. So all these need to be taken into account while developing the method at first instance. However, for the as a procedure part, so these are the different procedure need to be taken into account for quantification of these impurities. Either it is by external standard or relative response factor or using the reference standard materials or area normalizations. These are the different ways of. So when you look this chromatography as an industrial perspective, I can say this is the simple chromatography where only one impurity is present in the drug product. This is at a 5. You can see you can able to see the 5 minute. This is only one impurity present, and this is the chromatography normally employed. So this is a second example I'm going to show you. So this is you can see here ample of impurities are present. Here I can say 8 to 9 impurities are there, and they are well separated from the chromatography. So this is the expectations where each and every peak should get separated and quantified during the chromatography. Another example in case of complex formulation, you can see here to 90, there are 90 to 95 number of impurities are there, and see here the chromatography is up to 120 minutes, and each and every peak is get very well separated. So this is the requirement, and this is the one should able to separate such type of impurities and quantify such type of impurities with your validation parameters and to satisfy the requirement from the different regulatory agencies. These are the just examples I have shown to you, and these are the challenges. No doubt certain challenges are there, and you need to have certain knowledge and experience to overcome these challenges. So what are these challenges are nothing but stringent limits, sensitivity, low concentration, the nature of analyte, light sensitive, thermolabile, detection and quantification, sensitivity again coming in picture. Then non-chromatophoric molecules you need to quantify. Then what could be the resources for detections and quantification of particular impurities. Then storage condition, that maintaining the reactions and storage conditions during the maybe testing. Then environmental conditions, what we have discussed in the next previous one, as the temperature, humidity, and working on different climatic zone worldwide. That is again one of the challenges. So here different zones have different conditions. So you should get qualify your impurities in that particular product, and accordingly you need to establish that yes, in that particular zone my impurities are getting very well tested. And this is the last slide actually, where you can see there are most of the guidelines which are available. So one can follow all the guidelines for different countries in the world on the impurities, how to deal the impurities on even your drug products. Keep learning on this. There is no end for such type of topics. So there are always stringent criteria which is upcoming year by year. So that is from my side. Hello.
Just a minute, sir.
Yeah.
Thank you for the session, sir.
Thank you, sir, for giving your valuable insight on the dealing with the impurities in the pharmaceutical regulatory perspective.
Yeah.
Now, I would like to request.
Just a minute, Aniket. Just a minute.
Yes, sir.
Zope sir, we are extremely sorry. We were late in our schedule, and you had to curtail your time.
Yeah, I have finished actually, but if there are any questions from the audience, then I can take it up for 5 minutes, something like that.
Yes, 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 sir. Sure, sir.
Yeah. So anyone has any question, though I can give the clarification. So whatever the presentation is there actually these are very superficial presentation, but this need to be understand first, and then there are different topics or subtopic in the presentation. So there might be a lot of questions to the audience that what type of related substances. So there are different. This is a very vast topic. But is the topic just covered to have the insight that what is the seriousness to the regulatory agencies as per the regulatory agencies, and how we should be able to put up the practices in our drug product development program actually. So I would like to take some of the questions. So if it is there, then everyone is welcome to have questions on that.
Yes, sir. There is one question from Pawan Pajidar.
Okay.
Question is that what different types of the sterile water used in the pharmaceutical industries are regulated by the which department, and what is the main process?
So see, this is what we have talked about the analytical methodologies and controlling the impurities in the drug product. But looking at the different type of sterile water, so sterile water for injection is mostly being utilized for your manufacturing of the sterile dosage form. That is the first source. And what is the main process? The process is there are different processes to produce the sterile water for injection. So reverse osmosis and distillations and osmosis process are there. And again, once it is getting calm, so there are different attributes which are being assessed for producing such type of water. For example, there are the chemical testings as well as the microbial testing, which are being taken into account to qualify this water for injections and sterile water which is being utilized in the pharmaceutical industries. Next. I think I have given the correct answer. That question was from Pawan, right? Hello, you are not audible, Aniket.
Yes, sir.
Yeah.
Okay, sir. Thank you for your guidance. Next question from the Shweta. The question is that what about the impurities in herbal products or in the phyto compounds?
Yeah, so whatever I have covered is for the allopathic medicines only. But in case of your herbal product, the same case is there. But these are herbal products will be more challenging, or phyto compounds are more challenging. We need to see the what are our intention. Means what type of impurities. If you have prior knowledge of herbal products and what impurities are coming depending on your chemical constituents, and if you know the targeted impurities, so you can very well understand their chemistry, and you can able to find out the different ways for its quantifications. So,
Yes, sir. Thank you.
yeah.
There is a next question, I think, from Rushikesh Patil. They raise their hand.
Yeah.
Yeah, Rushikesh, please.
Hello, sir. This is Rishikesh. I want to ask you a question that while working on ANDA products, what is the way or how we can decide the specs? That is
Exactly.
particular
Yeah.
specification
So
part.
yeah.
Can you just elaborate more on that?
Exactly. So I have shown you one table that if your maximum daily dose is say 500 milligram. So what is meant by 500 milligram? Say you have the paracetamol tablet. and paracetamol tablet contained 500 milligram of the paracetamol. So and it is to be taken once in a day. So your maximum daily dose is 500 milligram in that case, and there is a guidance.
Yes.
And so there is a guidance. So when it is a 500 milligram, so according to the guidance, you have to see their range. So where my maximum daily dose fits into, and once you see that range, specified range, so there are different type of thresholds. So three type of thresholds I have explained you. One is reporting
Yeah,
threshold,
reporting,
identification threshold,
qualification.
and qualification threshold. So depending on that, if it is identification threshold crossing that particular limit, so that need to be indicate on the certificate of analysis. Means whoever have given you the tablet for testing, so you have to say that this is the impurities which are coming in your drug product. That is number one. If that impurity is crossing your identification threshold, say 0.1%, so in that case your responsibility is that you need to identify the impurity. पहले तो इतना आता है, दूसरा थ्रेशोल्ड जो है वो क्या है, मतलब 0.1 के ऊपर सपोज आता है या 0.12 आता है, तो वो क्या है, मतलब वो क्या है, मतलब आइबुप्रोफेन की कौन सी impurity है, आइबुप्रोफेन की है कि एक्सिपिएंट की कौन सी है, तो उसका पर्टिकुलर लिमिट जो है वो क्वालिफिकेशन थ्रेशोल्ड में आएगा. सपोज 0.2% तुम्हारा क्वालिफिकेशन लिमिट है, तो 0.15 और 0.1 एंड 0.15 सॉरी 0.2 के बीच में आता है, तो तुमको कुछ करने की जरूरत नहीं. इफ इट इज क्रॉसिंग 0.2%, इफ इट इज कमिंग इन 0.25% इन योर ड्रग प्रोडक्ट, देन इसका इम्पैक्ट क्या है, सेफ्टी कंसर्न क्या है, उसका जेनोटॉक्स, ये इम्प्योरिटी जेनोटॉक्सिक तो नहीं है, ये इम्प्योरिटी कोई साइड इफेक्ट तो नहीं दे रही है ना, ये इम्प्योरिटी मैं कैसा क्वालिफाई करूं, मतलब if you do not have any answer for that qualification, then toxicity studies need to be performed. Okay. So this is the cycle, this is the chain actually where you need to define, and these are the specific in your all three. Hello. Yeah, Rushikesh, I think I have given answer to your questions.
Yes, sir. Yes, absolutely.
Okay.
But
Yeah.
one more thing I want to ask you that as of you said that working in ANDA is we are copying to NDA molecule. Like we are,
ANDA is totally different. ANDA is that you are copying to someone else.
Yes,
Okay.
yes, yes. But as of we are working in ANDA, then there is RLD, which is already present with
Market.
FDA and all in market. So FDA must have correlative data for RLD, and we also have correlative data with our in-house product. So
Right.
if there is any unmatching criteria with our in-house product with respect to RLD, then what are the scenarios?
Right.
Then
So if
how
it is
to
unmatching,
deal with
yeah.
it?
If it is unmatching, the first scenario is why it is unmatching. So what is there? Your processes are different, your molecule chemical entity is different. So are you using something excipient which are different in nature? So you need to understand your chemistry first, processes and chemistry on your product, and you need to have the yeah. So that is our call. So may I audible to you now, Rushikesh? Yeah. So you need to see that what is there, what is differences are there with respect to reference listed drug. Whether I'm using the excipient or different quality, processes are different, or environment is different. So by which nature I'm getting different profile in my testing methodology. If it is something you could able to diagnose it, then it is our call that we need to rectify on that particular topic. Hello. Is it okay, Rishikesh? Yeah, you have to unmute yourself. So Aniket, you please unmute to the Rishikesh. He is not able to speak.
Yeah. Sir, can you just have just a little focus on reverse engineering?
Question.
As of I
I
know that it is done with RLD itself, no?
Yes, reverse engineering, of course, it is to be done on the RLD itself. So there are
Yeah.
certain polymers, say, for example, polymers are there. So in case of the polymer, there are different molecular weights are present. Okay, and suppose there are X polymer which is belonging to the 40,000 to 1 lakh polymer is there. So which one? And there is no any clear-cut information which is provided on the RLD, whether which type of polymer is present, which molecular weight of the polymer is present in your drug product. So in that case, you need to develop a methodology it should be accurate and precise in such a way that you should able to determine the correct molecular weight of your polymer. If you are not able to do that, so that will,
sir, let us stop now.
so that will, yeah, so that will appear, that may appear the impurities in your drug product or something like that, and you will not able to put the correct molecular weight polymer in your drug product. So that is the simple case I am just highlighting in front of you now.
Thank you, sir.
Any
It
questions?
looks like we have covered most of the questions from the participants.
Okay, thank you.
I would like to thank Dr. Muralidhar Zope sir for the valuable insights. Now I invite Ms. Sukanya Suryavanshi to propose the vote of thanks.
Good morning. बोलो ना. हाँ ठीक है मैं करता हूँ बाद में. हाँ कॉलेज में हो. काय झालं सर? तिला ऍडमिट कर सांग सुकन्याला. दीपाली.
Appreciation certificate on the behalf of Yash Institute
आवाज
of
किती कमी तिचा
Pharmacy
आवाज.
and Indian Pharmaceutical Association.
तिचा आवाज कमी येतोय तिचा. खूपच कमी आहे बारीक.
A very good morning, distinguished guest, Dr. Muralidhar Zope sir, Associate Vice President, Research and Development in the Analytical Development, Sun Pharma, Vadodara, India. Respected Principal, Dr. Sachinanda Angdi sir, Yash Institute of Pharmacy. Aurangabad, teachers and my dear friends, on the behalf of all the dignitaries present here, I assure you that this wonderful session will surely help us to build concrete concepts for dealing with impurities in pharmaceutical, a regulatory perspective. I extend my gratitude to our today's distinguished guest, Dr. Muralidhar sir, to take out time from his busy schedule for gracing and sharing us with your finding and opinions today. It was really interesting session. Thank you, sir.
Yeah, thanks a lot for giving me opportunity to express my thoughts in front of you guys. Thank you.
Yes, thank you, sir. I would like to invite Preeti Ingle for the next session. Over to you, Preeti.
Thank you. Good morning, everyone. Honorable Principal Sir, management, speakers, delegates, faculty members, research scholars, students, industry persons, and my dear friends. Welcome you all for our third session of Global Pharmaceutical Trends International Webinar. We have Dr. Abdul Rauf sir, who is going to enlighten us on topic hospital pharmacy practice research. Before moving to that, I would like to request our principal sir to speak words.
Thank you. Good afternoon, all the participants. Shri Muralidhar Zope sir, our friend Shri Paresh Wani, because of whom Shri Muralidhar sir could present his session today. He had presented his consent, and I can see the enthusiasm that he was present right from the start of the session till now. The next session of Dr. Abdul Rauf sir, one of our good friend and an all-round excellent person. Before, am I audible?
Very much.
Okay. Thank you, doctor. So, before the professional session begins and you are introduced professionally, I would just like to bring this to the notice of all of the participants that Dr. Abdul Rauf is a very nice gentleman. He's an all-rounder personality. He still plays cricket. He is a very good dancer. He's a very good singer. And really, this is a great opportunity to have you here, sir. So on behalf of the management, all the faculty organizing committee, I welcome you heartily. Thank you.
Thank you so much, sir, for your humble words. I would humbly again like to remind everyone, if you have any question during a session, please type them in chat box. We will bring them at the time of question and answer at the end of the session. Now, I would like to request Ms. Pratiksha Harshil to introduce Dr. Abdul Rauf, sir. Over to you, Pratiksha.
Thank you, Preeti. It's my great pleasure to introduce the speaker of third technical session in this webinar, Dr. Abdul Rauf sir. Sir is the Assistant Director of Pharmacy Department, Corporate Pharmacy Services of Hamad Medical Corporation. Sir received both B.Pharm and M.Pharm degree from KLE Dharwad University, India, and MSc Clinical Pharmacology and PhD from the University of Aberdeen, UK, and receive hands-on training in pharmacovigilance from Upashala Monitoring Center of WHO, Sweden. Sir underwent certification programs related to epidemiology and biostatics UK, research to publication BMJ UK, and drug information to toxicology USA. In addition to his extended role, Sir specializes in medication safety practices, including adverse drug reaction, medication error. Sir has authored in several publications and served as receiver for the numerous international high-impact journals. Sir served as a speaker, presenter, and scientific committee member in several conferences. He also supervises PhD students based in both India and UK. Currently, he is involved in research related to pharmacy prescribing, antimicrobial stewardship, patient reporting areas, medication waste, and COVID-19 vaccination, advocacy, pregnancy registry related to COVID-19 formulary, adherence toxicology, and poison epidemiology registries few quality projects looking at medication safety. Sir is having membership of various reputed professional bodies like Registered Pharmacies Qatar, Registered Pharmacy India, and member of Community Pharmacy Accreditation Standard Workshop Group of SCH, and many others. Thank you for accepting our invitation and welcome on the behalf of all the audience and organizing community. With this short introduction, I hand over to Preeti. Over to you, Preeti. Thanks.
Thank you, Pratiksha. Now I would like to request Dr. Rauf sir to please start the session. Over to you, sir.
Thank you. Thank you very much. Let me share my slides. Right. Right. Thank you very much for the introduction. And non-professional curricular activities have been introduced by Angadi, my very good friend, who was my classmate in my master's. We were together there and we had very good fun there. And the introduction wasn't a short one. It's a quite long introduction. I really appreciate that. So without further delay, let me start my session. So the initial session was about pharmacy practices in states. They really have a very good pharmacy practices where they look into the community pharmacy practices, hospital pharmacy practices, more patient-centered care and all. So my topic would be more about scope of hospital pharmacy practice research from our perspective. When I say our perspective, it is Hamad Medical Corporation perspective. And I'm Dr. Abdul Rauf, and I take up the responsibility of being an assistant director of pharmacy at the corporate level, where we oversee 13 hospitals with more than or almost equal to 1,000 pharmacists, including clinical pharmacists with different services. So today my presentation would take you through different objectives which need to be achieved by end of this project, and haven't put the slide which talks about I have no conflict of interest. Yes, my presentation doesn't have any conflict of interest. I'm free from any conflicts. This is purely educational session which we are looking at. So I would take you through different definitions of what you mean by pharmacy practice, what do you mean by research, what do you mean by pharmacy practice research, because understanding the definitions are quite important to understand what is the scope of that particular aspect of any practices. So then we would go through the scope of pharmacy practice, the scope of pharmacy practice research. Then I wouldn't go into the details of methodologies because it is not within within this presentation. So probably if I have to take methodology, that would take another 4 to 5 hours to talk about pharmacy practice researchers. Then we would talk about the challenges in pharmacy practice research, which is quite important. The experience comes from the literature as well as our personal experience. Then evidence and impact of pharmacy practice research. When you say evidence, the evidence to drive pharmacy practice research and the impact of this pharmacy practice research on the patients. Then we also look into the scope of hospital pharmacy practice research from our context, and we would give you some examples from our pharmacy practice research center. So let's start this. Am I clear to everyone? You guys can hear me?
Yes, sir. Yes, sir.
Okay. Well, so about the definitions of pharmacy practice. So always I used to have this in my mind, a big dilemma. What is the definition of pharmacy practice? Because different people think it in a different way, because different countries have different practices. So I would go with the definition that has been put forward by FIP, International Pharmaceutical Federation, which is quite simple, but it encompasses everything what is there in a pharmacy practice, which says responding to the needs of people who use the pharmacy services to provide optimal and evidence-based care. There is a lot into it. When you say optimal and evidence-based care, you need to think from two perspectives, from the patient perspective and from the pharmacist perspective. Optimal in terms of safe, effective, and cost-effective medications for the patients, whereas the evidence that drives these practices that shows an impact on patient outcomes. So this clearly talks about the patient's perspective as well as the pharmacy perspective. So you need to be very clear. Any need of a patient from pharmacy perspective that can be provided by a pharmacist with optimal and evidence-based care, that can come under pharmacy practice. These definitions vary. Some definitions are there, but these definitions have been taken from one of the publications that has been published recently in 2019, where they looked at different papers. It's not a systematic review paper. It's just a review paper where they looked into different studies and they extracted the definitions and they extracted the pharmacy research definitions as well. And where they found out, what they found out is there are 13 definitions in that clearly says that the practices are different. Why are the practices different? The practice is different based on the country and the setting that you work in, as well as the budget that is available to provide these practices. So you would have different definitions for the for pharmacy practice. Let's say there is one definition which talks more about research, which says pharmacy practice is a core component of pharmacy and encompasses the development of pharmacy roles and responsibility of a pharmacist, more of a research component. But the one which I liked is from Baba, who is a professor in UK, who talks about pharmaceutical practice. What's the difference between pharmacy practice and a pharmaceutical practice? This is just inclusion of a pharmaceutical science into it, where you talk about the industries, where you develop, research, distribute, looking into all aspects, right from the industry to the monitoring into the hospital. So included everything into the definition. So the basic thing about this is pharmacy practices do not have one standard definition, which need to be developed looking at a real model of it. Now, to understand this, how would you combine a research to pharmacy practice? We need to understand what is a definition of research. For me, it is quite simple because we do a lot. It is systematic investigation of any problems, any concerns, any unknown uncertainties using a rigorous research methods that would generate a valuable conclusions which can be used to change practices, to change whatever context it is in. So simply we need to add this research aspect to the pharmacy practices. Now we know pharmacy practices, which includes everything, but within that you have to incorporate this research component, which is more of a systematic investigation of unknown uncertainties, problems, the rigorous research designs or methods that would produce a valuable conclusion. Now, the definition of pharmacy practice research, what have we done? We have combined the pharmacy practice research with the research. But even these definitions, this slide is very busy. I kept it busy purposely because we don't have a right definition for the pharmacy practice research, because the pharmacy practice itself has a different definition. Then that would vary depending upon the settings, depending upon the country. So the one which I like the most is the pharmacy practice research looks at the impact of practice of pharmacists that had an impact on healthcare systems, medication use, and patient care. So you have a pharmacy practice research. What does it include? The definition says it looks at the impact of these practices on healthcare system. When you talk about the healthcare system, it looks at the workforce of healthcare system and what is the cost of pharmacy practice. Is it useful? Is it beneficial? What are the different services that are provided? Are they beneficial compared to other services? The pharmacist can have or can serve similar to a doctor in certain areas. Is that more valuable than the other healthcare professional services that are provided? Comparison of that. And second impact that a pharmacy practice would look at is medications use or medicines use. What is it? It's about the safety, effectiveness, and cost effectiveness of medications. So there is a lot of research that is done on that. For example, you can talk about medication errors, you can talk about adverse drug reactions, you can talk about the effectiveness of these medications based on the context where you are looking, what you're looking. So the third one is about the patient care. This is the clinical outcomes of the patients. the definitive outcomes you're looking at when you provide them with the pharmacotherapy, when you're looking at the medication reconciliation, when you're looking at different aspects of a patient care, optimizing the medication care. So this definition is quite important to understand the pharmacy practice impact on these three different domains. Then the same paper has published more than 9 pharmacy practice definitions. It is understood this is because as I told you earlier, the settings are different, the practices would be different in different countries and different hospitals. So another definition which talks about is that pharmacy practice research is a component of health service research. When you talk about health service research, the definition of health service research is more of looking at patients accessing healthcare system. Why do they access? How do they access? How does it cost? This is more about healthcare research, health services research. So they say pharmacy practice is a part of it. Why do they say a part of it? It's a component of it because they would like to compare the impact of pharmacy services on healthcare service. So in terms of the new roles the pharmacists could have. So there are a variety of roles which are really growing day by day. Pharmaceutical care, you have many clinical roles, clinical pharmacy. So all these roles, what impact does it have on health services? That is what about the pharmacy practice. So this is one of the definitions they talk, considering the pharmacy practice research. So let's straight away go into the scope of pharmacy services, which is a conceptual model where they talk about community pharmacy, pharmaceutical services, clinical pharmacies, and social and administrative. When you talk about social and administrative, it's more of the behavior of patients, why they do not take medications, why they delay, what is their understanding of medication, what's their knowledge about the medications. So that is one of the practices which are quite coming up, but we don't have that, but we do bits and pieces of it. Then community pharmacy services, which are a lot. Then pharmaceutical services and clinical. This is the basic scope which they try to have a scope of pharmacy practice looking at this conceptual model. Then, just to come out of this dilemma with different definitions, they have developed 3 domains where a pharmacy practice research can be looked at. The 3 domains is product related, where you can talk about medicines use, where they look at the safety, effectiveness, optimization of medications, and all that. It's more of a product related, where is the patient-centered care. That means what does a pharmacy services would have the impact on the patient outcome? How would they have a pharmacotherapy and what impact does it have? How does they look at the definitive outcomes? How would they improve the quality of life? So different aspects to it. That is from the patient point of view. And where the first one, medication use, is more of a product oriented. And the third one is the healthcare services. This is the third domain where there could be a pharmacy practice services, which looks at what impact the pharmacy services, which have defined before. The services impact on it. How could what cost is there for these services? How to sustain these services in healthcare profession. So as I told you, I wouldn't go into the details of the methodologies because it is not within the scope of this presentation. As you can see, it would take a long time. I would just explain about the methodologies. The methodologies, pharmacy practice research, are similar to any other research that are done in medical research or clinical research or anywhere. There's the similar, but there are certain novel methodologies which may not be novel to other fields, but it could be novel to pharmacy field or there, which people have to understand. So the methodologies can be divided based on the type of questions which you want to answer. The first thing, if you're looking at the time orientation, where you look at the safety of medications. When you talk about the safety of medications, probably you wouldn't capture in your pre-licensed trials the effects of medications which are rare, So probably you want to know the safety aspect of that, which is time oriented. So where you can look at retrospectives as well as prospective study. When you talk about retrospective study, what you do is you look at the outcome. You have seen the outcome, means they
Doctor, you are not audible. Hello. Hello.
I think there is a range problem, sir.
Okay.
Just wait for a few minutes.
Hello.
Hello. Hello. Hello. Am I audible?
Yes, sir.
Abdul sir?
सर जॉईन करतायत परत.
Yes, sir.
हो. त्यांना पुन्हा जॉईन करायला सांगितलंय. झाले नाही ना आणखी. Well, audience, there is some connection problem. Dr. Abdul Rauf will join immediately. All participants are requested to wait. Dr. Abdul will join shortly.
Okay, sir. Welcome, sir.
Sorry, guys, because there is a technical problem. In fact, there is an issue with the internet here. So we are trying to overcome that issue. Sorry for delay. And I'm not sure which slide I am on. So I'm not sure on which slide I am on. So let me, if I get an answer from someone, so probably I can just continue from there. Hello?
I think you are on scope, sir. Scope of pharmacy. Yes, sir.
Pharmacy practice research?
Yes.
Okay, right. The scope of pharmacy practice research, again, these three domains have been extracted from the same paper which they spoke about the definitions of pharmacy practice research, and they have made three domains. One is medicines use, another patient-centered care, and the health services provision. So the medicines use more of the product-oriented, whereas the patient-centered care is more of a patient-oriented, and health services looking at the impact of this pharmacy services in terms of budget, in terms of time, in terms of cost, in terms of its impact on the services itself. So these are the three domains which they looked at. And for me, the review could have done and should have added another domain, which is more about professional development, which I'll be talking when we go to our settings. Am I clear now? Hello?
Yes, sir.
Okay.
Yes, sir. You are very clear.
Okay. Thank you very much. So we were talking about, I have even completed this slide. Sorry, so the methodologies, I will not be going into the details of the methodology. Let me go through little faster so that we can catch up with the time. The methodologies depends upon the type of questions you are asking. If it is a time-oriented, which is related to the safety aspect, where you can see some rare side effects, some rare safety issues which cannot be identified in pre-licensing clinical trials, you would go for time-oriented, where it could be a retrospective. You look at the outcome. The outcome is nothing but the adverse effect which is in the context, and you go back and look at the exposure. When you want to look at as a cohort prospective, then you have an exposure, you use the medications, and you're looking over a year what impact and what side effects that medications could have. And certain analyses are done to associate those. And the second one is about study purpose. This is more about the prevalence of studies, more of a cross-sectional studies where you describe the situation. And you describe the situation, and the description should lead to hypothesis. So if you have an analysis within that study, probably it is an analytical designs which you look at, where you have inferential, but it doesn't give you a strong association of causality, but it just says that this is related to this may be. So generating the hypothesis. Another one is investigation oriented. It's more about effectiveness of medications, where you have experimental studies, you have randomized controlled studies looking at the effectiveness or efficacy of medications. So that is more about investigational orientation. When you say investigators orientation, the whole experiment is within his control. He can change what he wants to do. You would randomize that. You would look at the effect. So these are other types of experimental methods that are used in pharmacy practice. These are the general ones which are used worldwide. Then based on the question oriented, we do a lot of work which is about social pharmacy, where you use questionnaires, a lot of questionnaires. When you talk about qualitative and quantitative and a mixed method, there are two things that you need to remember. One is inductive, another one is deductive. When you say inductive, you know nothing about the subject. So you want to know what people know about this subject. So you go through focus group, through interviews to get in-depth knowledge of that particular concept, a particular phenomenon. So you take that and you develop a questionnaire based on that, which is a deductive way. and you have service based on it. Sometimes you use mixed methods. You go with focus groups, then you mix with quantitative surveys, and you come up with a more valid data that can be utilized. And the last but not least is based on pharmacoepidemiology studies design, which you look at different risk factors, associations, and different prevalences and all that. And these studies are more of an observational studies, again, which could be case controls, cohorts, case series, and all those. Am I clear? Can you hear me? Yep.
Yes, yes, yes.
I'm a bit scared because, I may get disconnected and I will be talking. Thank you very much.
No, you are, the moment you get disconnected, we'll let you know.
Okay, thank you very much. Now, what is the impact and the evidence? There are two things to this heading. One is evidence, the evidence that drives pharmacy practice research, and the impact of this pharmacy practice research on patients, on healthcare systems, on medications. So let's talk about a general dilemma about the evidence. What happens? Is it a right evidence? Can you really utilize this evidence? Then we would go into the pharmacy practice research. When you talk about the evidence, there is a multiplicity research that has been published. There is a lot of research. Does it answer your question, what you're looking for? That's another question mark. Because all this research, you have preclinical, pre-registration research, which usually are not published, but they are done in a very rigorous way. And you have individual randomized studies. But all this, you have a lot of research that has been published. But how would you select the research that answers your particular question? That is a real dilemma, because most of this research answers a big question. A big question means which are more prevalent. They ignore the things which are less prevalent, which are very important for a healthcare system when you combine together. So, for example, this is one. So they just ignore the prevalent. Another thing, even this big question that has been answered, they cannot be generalized. Just take an example, a research study that has been conducted in North America on Caucasians who are 50 years old about a certain impact of a medication or effect of it. Can you generalize that to the population in Australia, which different ethnicities? No, you cannot. So there is a lot of dilemma looking at a lot of research that has been published. Second, the quality. Always you can see the quality of research has become a big issue. Thrash in, thrash out. If you don't do it properly, then you wouldn't get a good results. And if you don't get a good results, if you try to adopt that results, probably you are definitely harming the whole system. So the quality of research is quite important. So looking into these aspects, the new drive is come that is about evidence-based practices, where you have a Cochrane reviews, which look at a very systematic way of research that has been published worldwide, and they combine it and they increase the population size, sample size, reduce the confidence intervals of an effectiveness of a medication, which would definitely give a valuable results that can be utilized to change the practices. Another one, if you look at this, in UK, they have the NICE guidelines, which commission the same reviews, and Center for Dissemination, Reviews and Dissemination would help in synthesizing the reviews for the NICE guidelines in the UK. So other countries are also developing the same thing. They wanted to have a collaboration, Cochrane organizations, where can they look at the evidence. Now, another aspect to it is the systematic reviews and critical appraisal. As I told you, the quality of studies makes a big difference in generating a good results and a good conclusion, and having conclusion that can affect the practices. So the systematic reviews, what it does, basically it is conducted in a very systematic way, pulling in all the research, looking into the quality of this research and synthesizing the research, and finally coming out with the effect size. Now, when I say that, if you have a question, very crisp question, then you can use something called as a PICO. That is patient, interventions, comparisons, and outcome. So it is a very crisp way. We have different terms within that. So use these terms, you combine them, then it gives you how you have to search different databases. Now, I would whenever I search for a systematic review, I would take a parent database such as PubMed such as Embase, and I would combine with another small databases which are very much towards this particular population. And if you do a statistical analysis within the systematic reviews, then probably that is a meta-analysis. And if you don't do it, then it's a systematic review which with a narrative outcomes of it. So this becomes very important. And the grades are based on the Cochrane criteria, which tells you that, you know, the bias, the limitations, based on that the grading is given in terms of the evidence which can be utilized in clinical practices. Then all this evidence through all the Cochrane systematic reviews that comes out will influence your practices. Even in our organizations, we develop a lot of clinical guidelines. So initially we do a systematic review to look at the impact or the effect of the medications, and we look what are the safety issues, and we come up with our own guidelines that definitely informs our practitioners. Am I clear? Hello?
Yes, sir.
Okay, that's good. Then let's talk about the evidence that is used in the pharmacies. Now, most of the publications that we have is about the medications, to choose one particular medication compared to another one in terms of safety and effectiveness. But just imagine these methodologies can be applied towards pharmacy practice research, which has an impact on health services. And now, when I say that, you need to understand these type of methodologies which are important to be applied in this pharmacy practice research. Again, that becomes a big dilemma, because most of the publications that come out says that pharmacy practice research lack quality of research. When I say quality, they are small in numbers, they have not chosen a right design to answer a right question, and the quality is quite low. So that is one about different dilemmas that can impact the pharmacy practice research. Then you look at systematic reviews of pharmacy. Again, there is a lot of literature on systematic reviews of pharmacy practices research, which says we are still immature in terms of research. Pharmacists are still immature in terms of research. That is something we need to improve. So this is about the evidence that drive the pharmacy practice research, which clearly says in a nutshell that we need to have a high quality research that need to be conducted to change your practices, as well as whatever the small studies, pull them in, look at the quality, and try to get the conclusions out of which can be informed to the policymakers to upgrade our profession and our practices. Now, another aspect to this is the outcomes. You have to choose a right outcome in the research to show that our practices are quite valuable. Simple example of this is that there has been a study that has been conducted to look at the pharmacy services and the other healthcare professional services given to cardiovascular disease outcomes. The education and cardio, they did not find any difference. It is the same. In fact, the other professions, they've been providing education on cardiovascular diseases, their impact on the patients is much better than the pharmacy. But one important thing that they found out in the study is the patient satisfaction. The patient satisfaction was more in pharmacists delivered education compared to others. So we have to look at the right outcome that needs to be captured and identified. Now, the pharmacy practice research, the policy, practice, and research triangulation, the pharmacy practice research can be categorized into 4 categories based on its relation to policies decisions. Now, these are four different types of research that can be conducted to the pharmacy practice research based on its policy links. Number 1 is research-informed policies. The best example for that is smoking cessation, where they looked at the impact of pharmacists who are trained in smoking cessation training compared to the pharmacists who are not being trained, and their impact in rates of smoking cessation when they have counseled, educated the patients. physicians, and they had really found that the training and the education that received by the pharmacists who have provided this education has really reduced the smoking rates, and there has been a high rates of cessation. So these type of research, they inform the policies. The policies take up this type of valid information, valid evidence to develop the policies where the privileges and practices are given to the pharmacists. And another one is about research to support a planned policy. Probably you wouldn't have big research, just a bits and pieces of research which you combine together to support a planned policy. And another one is research to confirm their approaches or implemented. For example, one of the good examples which I'll be talking about, which we have done in our setting, is pharmacist prescribing. So this type of research would look into already established pharmacy prescribing through other channels of research. Then they will look at what impact it had, it has been properly implemented or not. So you can see different levels of research that informs the policy in different ways. One directly informing the policy to change the practices, another one supporting it, which may change the policies later, another one where you have established a service, so you're looking at the service, whether it has been implemented. And the last but not least, that will look at the improvements that are required to sustain these services. That is about research inform future services. Then what are the challenges? The challenges from my personal experience is the time. But from the literature's point of view, which you can see the expertise, the people who conduct this research need to be expertise. They need to be experts in the designs. They need to be experts how to collect the data. So the expertise is required. If you don't have expertise, probably we may not be motivated in this. And the time, you need to finish this in time frame, commitment towards the time. And the important factor that is a challenge is the money. That needs to be funded. Now, when I say that needs to be funded, don't think it needs to be funded for some other reasons. No, maybe some of the researchers within the research require some sort of a training, which we do. In focus groups, we train our researchers. There you require money to pay for the courses and all. And you would pay this funding for research assistance for your research. So funding becomes quite important in research. This is one of the challenges which you can see. The engaging colleagues. How do you engage colleagues? Very difficult to engage colleagues. They require a commitment. Some of the colleagues say that I'm not an expert, so you need to train them. You have to monitor, you have to train them in the way they have to collect the data. You need them to be committed towards the research. So engaging them through different ways, motivating them in different ways to get engaged, that becomes another challenge in research. We had seen many researchers which have been submitted, they have never seen the end. So they're all stuck there, not conducted or still going on, failed to do it because the engagement of the colleagues are quite important. Then changing the status quo. Here, what happens is, whenever a new practices come in, for example, I'm doing a research for a diabetes education for a patient, but you have another group of diabetes nutrition group, where the dietitians would give education to the diabetic patients. They would feel threat from you. So there will be a lot of resistance from there. Probably that would really stop you from doing the research. That is one of the challenges which you find. The negative findings, always remember, the negative findings would be there. We should be brave enough to put this forward and have an explanation for that. And we should have another outcome to explain why is it a negative finding. Then funding, as I told, it is always an important thing, and the duplication of research. Now, most of the people, whenever you talk about research, it is being duplicated, then the questions come out, why are we duplicating this? Can we generalize this in our setting? So you need to be very clear what you're doing, what is the inclusion, what is an exclusion criteria, how you're utilizing that methodology. Then communicating with the policymakers, that's the biggest challenge which you could have once you get your results, convincing them to change your practices. Am I okay on this slide? Can you hear me? Yeah, good. So now let's straight away get into our settings. Till now, I was talking about the pharmacy practice scope, the pharmacy practice research, what is a pharmacy practice research, what is the scope of it, what are the impact, what is the evidence that need to be driven, and all that. Now let's get to our settings. We are a huge organization, you can see, with 13 hospitals and different services, ambulatory services and all. So our pharmacists are scattered all over. We have almost 1,000, that includes pharmacy technicians, and we have clinical pharmacies. You ask me, it is there. We have IT pharmacists, we have drug information pharmacists, we have everyone. So for us, the pharmacy practice would be just stretching the boundaries. Probably it keeps on changing. So we need to look at different aspects. So you can understand what would be our research. Our research covers the whole scope of pharmacy practice. So if you look at the publication that looked systematically at different hospitals, and they have captured different pharmacy practice research and pharmacy practice scope within the hospitals, and they've come up with 3 domains and you can say these are the 3 areas which they say the pharmacy practice goes on. One is pharmaceutical care, and this is a holistic approach. It's not easy to have. Even I would say we would do bits and pieces of this, because the pharmaceutical care is more about a responsible provision of a pharmacist providing medication therapy that would have a definite outcomes on the patient and improved quality of life. So just imagine, when you talk about medication therapy, you talk about medication optimization, right from duplications, adverse effects, identification, alternatives, everything. That is one part. Then you're looking at quality of life. You follow them up and see how is the quality of life. So you are taking the responsibility, accountability of having a definitive outcomes, as well as you're looking at the quality of life. So that is quite difficult. To have this, you need to have a process, a structure in place, which can identify and document all of this. So that is not available in most of the countries. They may be doing a bits and pieces. So this is one of the domains of pharmacy practice. Clinical pharmacies, yes, we do have, but even clinical pharmacy scope differs from country to country. In Poland, probably if you say clinical pharmacy, it is more about preparations, sterile preparations and all that. In other countries, they have a different, it all depends upon what practices you have in that hospital, what budget you have to provide these practices, and how would you substantiate that these practices impact the health system. So for us, clinical pharmacy, it goes right from medication review, medication reconciliation, then pharmacotherapies, provision of pharmacotherapy. Then you look at reporting adverse drug effects, medication errors, rounds with the doctors, providing them the alternatives, education. Everything is included within our pharmacy practice scope here. And the public health is more about looking at vaccination, that is more of immunization, which you can see, and you can smoking cessations, which impact the public, and the role of pharmacists and looking at their impact on the public services. So keeping this in mind, we would go into our pharmacy department services and others. We have clinical pharmacists, hospital pharmacists, drug supply pharmacists, IT services, where you have automation and you have a special pharmacist to provide this sort of services. And you have clinics, which is the patient-centered care, where you can see MTM clinics, anticoagulant, diabetic clinics, lithium clinics, antimicrobial stewardship programs, and medication safety office, which they identify different types of medication errors, more of pharmacovigilance there. All the aspects of medication safety is taken care by the medication safety office, and you have medication safety pharmacists. And you have pharmacy continuous professional education research office, which is not covered within the scope which we are looked at the pharmacy practice research. They talk about medication use, they talk about patient-centered care, they talk about health services, but they don't talk of educational research, which is quite important to upgrade our profession, getting the privileges based on our competencies, looking at the need assessments, what is required to get those privileges, get competent enough in certain areas. Then we have Drug Information and Poison Information Center. We have pharmacists as drug information pharmacists, and we have ASHP residency programs and clinical attachments. The pharmacists come from different countries to do their clinical attachment with us. So these are different services. So our scope of a practice is based on all the services that are being provided. So if you look at that and add the component of research, then we need to look at every aspect of this to provide or to get the privileges of it. And the second thing, to show the impact of our practices on health system or healthcare system. So we have established Pharmacy Practice Research Center. This has been established 4 years ago, where we look into different aspects of pharmacy research and we guide them. The main mission for our Pharmacy Research Center is to build a vibrant pharmacy research capacity. The mission is to have the pharmacists as a good researcher. So we need to build that. If you need to build that, you need to know what are the needs to make an organization where you have a pharmacy services to build a capacity that has research in that. So probably you need to first know what is required. So we have done all that. We have done need assessments. What is lacking in our pharmacists is the motivation, is the knowledge, is the process. They are not knowing about the pharmacy research. So based on that, we wanted to develop a very good capacity, research capacity. That is our mission. And the vision is to be a regional leaders in healthcare research through excellence in pharmacy research, practice research. Why did I say excellent leader in healthcare research? Because pharmacy practice research is a component of healthcare research, which they focus on pharmacy services, their roles, why people access to the pharmacy services, what impact this could have on our healthcare services. So it is a part of healthcare research. So being a leader in healthcare research through excellence in pharmacy practice research is our vision. Am I clear now? Hello?
Yes, sir.
Okay. So now about the structure, you can see we have our own pharmacy research center structure where my boss is on the top, Dr. Muzal Hay, and I'm the head, and we have a co-head, and we have different people under us. We have coordinators. We have research officers from each hospital who would coordinate the type of research that are going in. If they need any help, they would come back to us. They give a feedback about that. And if they require some sort of training and all, we can provide them. So the another part to it is we looked at the areas of interest. So this came up from the need assessment, where this felt that medication safety and quality research is one of the areas, and we have huge number of people who are involved in it. These people meet once in a month to come up with ideas to conduct certain sort of pharmacy practice research, as well as another area is infectious diseases group, and we have that as well, and we have oncology groups. So just a snapshot of what has been happening in our hospital. We have in 2020, around 108 research projects. Now these projects could be ongoing, submitted, or completed. Out of this, or the previous researchers, we have 83 published research articles, 36 poster presentations, 26 drug use evaluation projects, 22 quality improvement, and 7 conference as oral presentations in different conferences, and 6 clinical audits. Now you could question, how would a clinical audit be a research? No, it is not a research, but we want to just identify, because these audits can lead to a research question. The quality improvement projects that are given, they are in a gray area. Is it a quality or a research? Probably that could even generate questions for research. So we have to identify and capture these as well. Now, the COVID research, what we have 6 projects in safety and efficacy. That research studies focusing on safety and efficacy of medications, COVID medications, which we looked into their adverse effects, their drug-drug interactions, and all that. There were 6 studies on that, and we had 6 studies on prevalence and the risk factors of people with the COVID. What is the prevalence and what are the risk factors? And the last 6 projects which we had in 2020, or clinical characteristics and outcome research, we looked at the characteristics of people who had COVID, who have been admitted in ICU, and we looked at their outcomes, what happened, the length of stay, what are the different therapies that have been used, and what is the severity, how severe it was, and all that. Now, just a research output. If you look at this, as I told you, that we have 108 research. Now, if you look at which is the most frequent area that this research has been conducted is in oncology. When you look at the publications, it is published in infectious disease because it is understood because more of a COVID era. Then we have around 38 original research followed by case reports. Whenever you say case reports, it becomes quite important for pharmacists, especially clinical pharmacists, where they see a lot of cases where they need to really assure this side effect is not because of the drug, it may be because of the patient in the patient condition. In this process, they would evaluate, they use different tools to see where this medication, the side effects is, or the safety issues because of this medication, and they publish a lot of case reports. And the case reports mainly used for very rare adverse effects, but they are of great importance because once you pull in as a case series, that could generate a hypothesis which can lead to different study designs. It could be a randomized control trial, or it could be observational study to confirm that there is this sort of adverse reaction or a side effect that can come out with this medications. Now let's look at those 3 themes or 3 domains which they said the pharmacy practice research is incorporated. And the 4th domain I would incorporate is educational research and see how we do in these 3 different domains. This would be quite interesting and motivational for you because we publish a lot of publications, because without a publication, your study is of no good, because it is once you publish, it is peer reviewed and it is a valid information. Again, there could be bias, but it has to be critically appraised when you are using it for your practice. So the first one is of medicine's use. As I told you, it is more of a product related. So you look at the safety, efficacy, and economic aspect of a medicine. So let's look at the first one, the medication use related research, which we do in our organization. The first one is medication errors. There could be an overlap between these themes and the researchers that come out of it. Just take an example of medication errors. The medication errors can be divided into 2 different research aspects. One is to see what type of medication errors you have, how severe are they, then what type of medications are involved in this medication errors. Then you try to mitigate them and come up with a strategy so that they do not take place. Another aspect to it is why people do not report medication errors. That is more about the safety culture, blame-free culture, just culture. So that is another aspect to the medication error research. And the third one is to know how the stakeholders, people in a high position, think about the safety culture, which would give a very good conclusions and very good results. And that can be combined together, which could impact the health services, which could impact an organization in terms of safety culture. So always remember, when they say there are 2 hospitals, one hospitals have reported around 100 medication errors and another reported 0, that doesn't mean that the second hospital doesn't have any incident. They are not reporting. So I would be more scared to go to the other one where they don't report it, rather than going to the one which reports 100 medication errors, because they learn from the mistakes. So always remember, reporting medication errors is quite important. Then drug use evaluation, it's very common for us, and it is very frequent because all the medications that are introduced into a hospital needs to have drug use evaluation within 1 year of introduction. And the main concepts of drug use evaluation, you look at the safety, efficacy, and economical aspect, and the consumption. It has been appropriately consumed, appropriately used or not. So we have a lot of drug use evaluation. Some of them are published, some I'll show that what has been published. Then case reports, as I told you, which makes a lot of difference. And we look at safety of medications during pregnancy. Now, a new aspect, the new FDA report has come up not to use ondansetron in pregnancy. It is based on a systematic review with huge number of people, but their settings are different, but they did not contraindicate. They say avoid. So we have a pile of information on people used ondansetron for the past 5 years. So we are looking at the safety of that, looking at the outcomes with people who have used the ondansetron. This type of research on which are related to the medication. Then I would be emphasizing on the optimization of medication use in terms of social behavior, which we have done on a COVID, which I would explain later. Then we have antimicrobial stewardship, where we look at point prevalence of certain medications and compare it internationally to see whether our usage is appropriate. Are we overusing an antimicrobial, or we are within the power, or we are better than that, which would give more information about it. Then pharmacoeconomic studies, I would explain that as well. We have a lot of pharmacoeconomic studies. All these are related to the medication use research with one of the domains which you have seen. Then, as I told you, the best publications we have, you would see me in all this publication, most of these publications is about hyperkalemia produced by labetalol. We are the first ones to publish this, and this has really made a great impact and informed healthcare professionals looking at hyperkalemia. And if a patient has an hyperkalemia, if he wants to labetalol, he will be a bit careful in doing that. So then we had about isomeprazole having a liver injury. It's one of the case reports that have been published. All these case reports talk about the safety, which are rare. So probably this informs the healthcare professionals to be careful whenever they see patients of this population while prescribing the medications. Then we had other misoprostol for miscarriage management in women with previous 5 cesareans, which is quite rare. So we have done this and we have published, so that clearly says you need to be careful when you use that. Then we have pyridoxine effectiveness and safe for postpartum lactation, because it's been very much used in our hospital. So we did a systematic review to look at it. Is it really effective or not? We found out no, it is not. So probably you need to have reservations on this. Then you have neonatal outcomes with oral. So this is one of the very good examples where a patient cannot take a parental injection of corticosteroid for premature labor. So what is an alternative? We looked into the literature, nothing has been given on that. So I said, why not we try an oral administration? The oral administration doesn't have any evidence. So we tried oral administration because the patient cannot take parenteral, and we looked at the outcomes, and the outcomes were quite good. And retrospective drug evaluation study, as I told you, we have a drug evaluation, which is one of the mandatory things. So we have published that as well. And we have one of the case reports that is being published during COVID, management of pregnant COVID patients, where we are pharmacists involved in therapeutic management, where we looked into different medications, we provided the information, we involved in the case, so we were there in it. Another one is very unique, where mifepristone for the abortion cannot be given orally because the patient is in ECMO. So probably we had to go give through NG tube and crushed NG tube, where there is no evidence, and the effectiveness of the crushed mifepristone through NG tube was quite eminent in our publication. Then, as I told you, I had put a lot of emphasis on studies where funding is quite important and it works and we complete the study. This is, most of the studies we have are collaborative studies internationally with UK, our own Qatar University, national, international, regional collaborations. This study is, as I told you about the medication errors, initially we looked at the previous few years, what are different types of medication errors, and we used James Reason's model where they looked at active and passive reasons for medication errors, and used theoretical domains to come up with what is it, and we used different tools to look at the severity of medication, and we have developed intervention that could mitigate these sort of severity. This is the first one. That followed with exploring the facilitators and barriers to medication reporting, which is quite important. Why healthcare professionals do not report. If they don't report, they don't learn anything from the medication errors, and you keep on repeating the same thing. So there are a lot of things. So we used a theoretical domain framework, which has 33 domains. On different behaviors. The behaviors could be because of lack of education, fear, safety aspect. So these domains are looked at, and the questionnaire is made based on these domains. And once you know which are the most prioritized domain, then you would look at behavioral interventions to overcome this behavioral changes. So it is a very upcoming model in qualitative study. And nowadays, most of the journals wouldn't publish any survey if it doesn't use a theoretical domain framework. So we use that to know why healthcare professionals do not submit medication errors. It's a mixed method. Then we had prospective healthcare professionals knowing what are the causes of medication errors. That's been published in PLOS One. And last but not least, but we looked at the views and experience of the stakeholders or the people at the higher position about the safety culture. We combined all this together and we come up with an interventions that could improve the safety of medication use. So this is a very big project where we had received $1 million for it. And it's a collaboration project with UK and USA. And we had colleagues from Ireland as well. Then the next one is quite important. There's a little story behind this. We went in 2020 to receive a vaccination, and my colleagues who are research colleagues, who we all sat together and we were just having coffee after the vaccination, and something came to my mind saying that today is not my slot. Today I was not supposed to take the vaccination. Why did they call me? So another colleague says, because other pharmacists are not interested to take, then that has stimulated something, that has put something in my mind, saying that if pharmacist being a medication expert and a frontliner, if he doesn't accept taking, then you wouldn't advocate taking vaccination. Then the research came out to know about the behavior using the same TDF, theoretical domain framework, about the behavior, why they do not take. And if you can see the results, it is quite clear. They say it is a responsibility to take the vaccine, but they're not sure whether their advice would help the patients to take. That means there are a lot of things in it. So we have identified those things, and we would have the strategies to change and have their acceptance rates higher and advocacy, and not to have an hesitancy on this. Then the second domain is about the patient-centered care research. It is mainly patient-centered. So we have anticoagulant clinics, diabetic clinics, and MTM clinics. All these are related to patient care. And we have publications from these centers which looked into the impact of pharmacists providing these services and the outcomes of it. The first one with the patients managed anticoagulant clinics in collaboration with other healthcare providers, and having another group, comparative group, where you're looking at only people, other healthcare professionals providing this, and they found out there is a great impact in both anticoagulant as well as the diabetic clinic. And there is another one, evaluation of pharmacist-based a comparison of anticoagulant with another healthcare provider. It's purely pharmacists who run the anticoagulant compared to other groups, and they have seen that there are certain variables like INR and all, they have been properly managed, and people are more satisfied with the pharmacist. This type of research would drive your practices. You would have an evidence. What is the main aim of pharmacy practice research? You have to get that straight into our mind. The main aim is to support evidence-based policy and practice decisions, where to employ a pharmacist and looking into medication use and prescribing. That is the complete medication management. So it should support your practices so that you would get privileges. Your practices would improve. You would have a better practice, better regulatory recommendations, regulatory recommendations for your privileges. So this research is quite important. That is the main aim. What is the second aim, which I could say? Understanding different aspects of clinical humanistic and economical impact to the change of these practices, which clearly shows that the change that is happening in a traditional professional boundary where doctors are everything, probably our services could impact the clinical, economical, and humanistic. When you say clinical, you're looking at the clinical outcomes, how well the blood pressure is controlled, looking at different variables, different biomarkers. When you talk about humanistic, how satisfied a person is with your services. And the third one, you're talking economical. Is it more economical than doctors providing it in terms of services, as well as reducing any sort of errors that come out? And all this put together, there is a lot of research which looked at it. When the pharmacist does a medication review, they could identify more medication errors than a doctor. That means you would have economical impact reduced almost to 50%. So all these drive that. Then the third domain is about the health services, where you have smoking cessation, where pharmacist's role has been very much seen in smoking cessation, and you have pharmacists prescribing, clinical interventions and economic impact, and antibiotic stimulation. And the publications which are available is, we spoke about the patient care. Here we are talking about how you inform the health services about your role and an impact. And one of the papers is about anticoagulant clean drive-up services during COVID-19 pandemic in Qatar, which clearly says it is feasible, which can reduce the burden on healthcare services for the visits, and all this could be a better way of managing the anticoagulant. Another one is about pharmacists deliver smoking cessation program, which clearly says that pharmacists' role can impact in terms of smoking cessation. They can do better than other healthcare professionals. Then we talk about economical impact, which is one of our studies, which we had grants again. So we are looking at clinical pharmacist intervention and how safe is it? How would it impact the economical component of the healthcare services? So they looked at all types of clinical intervention, the characteristic. That was the first phase of the study, and that has been sent for the publication. That is characteristics of interventions, to look at the different characteristics. So the second part is the economical impact and the cost benefit of inpatient clinical pharmacists in general hospital, as well as cancer care units. So this clearly shows that their intervention really has an impact. Having one pharmacist would have a great impact, comparing the salaries that have been provided to them, the budget that has been spent on them, compared to not having a pharmacist and going through these interventions, which could lead to a bad outcome. Then we had another one, which is quite important, and we feel really proud about it. In the Middle East, we are the first ones to get in the pharmacy prescribing proposal submitted to the ministry to get our privileges for pharmacy prescribing. And how did we do that? Again, as I told you, evidence driving your pharmacy research, and evidence showing the impact on the. So we had, I had a student from UK. She has done a lot of work. Initially, what we have done is we did a systematic review and looking at the barriers of pharmacy prescribing around the world. We took that barriers and we looked at the competencies required. We looked at the knowledge required. Then we had a focus group with all the stakeholders to look at what do they think? Do they agree for pharmacists to be prescribed or not? Then the third part to it is based on their recommendations. We have done a Delphi study, which, as I said, an innovative methodology where you have consensus report from different experts to look at whether the pharmacy prescribing for the pharmacist is feasible or not. Then a framework is being developed. Then later we did a study about the readiness of pharmacists within our organization for pharmacy prescribing. We combined all that and we proposed submitted a proposal to the ministry to grant us pharmacy prescribing, where you would have a component from the academic where they would develop a curriculum and they will be having training in hospitals, and finally that would lead to pharmacy prescribing. That's something very interesting for us. Then one thing, as I told you, which we are missing in pharmacy practice research in the work that has been published is educational research. We do a lot of educational research, looking at the impact of educational programs. One of the best examples of educational programs is a smart pharmacy program, where we had 30 pharmacists entering this program, looking at the diabetes management, right from the competencies, knowledges that are required. Then taking the lead with other 10 pharmacists each and educating them and choosing certain diabetic patients and taking care of it and looking at the impact of their education and counseling. So it was a big study and it has really showed a great impact. So that is more of an educational research that we had and that has been published. And we do a lot of need assessments for research, for any clinical aspects, everything. And once we find out that these are the priorities, probably we would find out the gaps and we would find out what is required for them. Is it required conference? Is it general workshops or knowledge-based studies? And we provide accordingly. So if you can see that, we had a lot of publications on this, which you can clearly see that assessing pharmacists' knowledge and attitudes towards a direct oral anticoagulants in Katha. Once you assess that, probably you find the gap and what are the needs, and you develop a program for them to improve their competencies. And we also have done need assessments for the pharmacists in terms of research, what exactly they are lacking and how to fill these gaps. That is another one, advancing the pharmacy practice to build a research capacity. And we had another one where we looked at the whole Katha, where we asked a questionnaire regarding their knowledge and practices about drug use in pregnancies. Once we knew what are the deficiencies, we have incorporated this into our conference where people can attend the workshops on drug use in pregnancy, what needs to be used, what are different considerations, what are different categories, how you can mix match these, and what are the risk benefits and all that. Then these are different sort of things which we do in our educational research. And this is our research team. Am I okay?
Yes.
Yeah, this is our research team. We won a lot of awards nationally, internationally. We have won in Dubai, we won in States, we won in different European countries. So our team, this is all about our team. I thank each and every member of our team. And these are international collaborations that we had with people from ACPE, that is Accreditation Council of Pharmacy Education, ASHP, and thank you. I'm ready to answer any questions.
Thank you, sir. Let us start our question answer session. Sir, question from Saurabh. Sir, please tell some platform for pharmacy students which are helpful for us after or pursuing pharmacy degree. Like IEEE platform are available for engineering student. Any platform for pharmacy student?
That depends, like, you know, what sort of platform. Back in India, the more of the scope is for the industrial pharmacists, right? For me, it all depends upon where you want to get it. We don't have such platforms as such, but we have this sort of a clinical attachment to get a feel of it, where you want to decide to go in. But the research as a pharmacy practice research, this is a personal interest where people can develop by themselves. I hope I answered the question.
One another question from Yuvraj sir. It is possible to set a pilot model for pharmacy practice for student in India?
There is a pharmacy practice that is there in India. If you look at different colleges where they train them for clinical pharmacy, such as colleges in Mysore, where they have clinical pharmacy curriculums, and they do have these students getting into different hospitals where they practice pharmacy practice. So it is already there. So it is how you look at it. That is what. And I'm not sure, Angadi should answer this question. Do you have any clinical pharmacy graduation or graduates or any curriculum related to masters?
We had a few programs, but now the current trend is we are in a situation wherein currently the students, they are more interested in either pursuing pharmaceutics or quality assurance.
Yeah, because that is the
typically clinical pharmacy setup. Typically clinical pharmacy setup, this is fairly good in Manipal.
Yeah, Manipal and Mysore, Uti.
Yeah, Manipal is good. Mysore is
Where
also
it
good.
is attached to the hospitals. So
Yes.
they have already set in, because I have a lot of colleagues who have come from India, and we choose them because they do this clinical pharmacy back in India, and they are very much prone to get a job here because they have this clinical experience. So that is one stepping stone for people who can have these. But, looking at India and the trends in India, more of industrial oriented, probably, as you said, pharmaceutical field is the best for them when they do their masters. Even Angadi, you can see when we joined, we had less members in pharmacology compared to pharmaceutics. So that is one of the reasons. But internationally, if you go to see, pharmacy practices have changed from the product to patient-centered. So, the product, you have a lot of automation, things change. We don't require a pharmacist to dispense medications anymore. So in near future, maybe from 20 years from now, you wouldn't have pharmacists dispensing, but rather they would be on more patient care. To have that more patient care, you need to have a curriculum which could teach you what is a pharmacy practice and how could it impact the patient care.
Okay, sir. So, COVID vaccine not give which disease or health condition?
Sorry?
COVID vaccine not gives which disease or health condition?
So if I have to put this question, where the COVID vaccine contraindicated in which situations, right? COVID vaccine.
Yes, sir.
Contraindications. As we know, there is a lot of literature behind this. The first thing is the people, it is usually not contraindicated unless a person has an allergic reaction, severe anaphylactic allergic reactions to COVID vaccine. Until then, it is not contraindicated. So some people with the mild conditions also take. From my experience in Qatar, everyone is eligible except for people who are severely allergic to vaccination. When I say severely allergic to vaccination, having anaphylactic shock. So other than that, I wouldn't see that, and everybody should receive. I'm an advocate of vaccine because it's very useful for the people who have immune deficiencies. The people who have diabetes, the people who have other comorbidities, because it would protect, because the COVID itself would have an adverse effect in terms of morbidities and mortalities.
I would just like to add here, doctor.
Yeah.
With your permission.
Sure.
It is not the question of who should take the dose vaccine. It is the question that a person should take the vaccine for how many times.
That's the question?
A person should take vaccine for how many? A person should take how many doses of vaccine? That is the question now.
Okay, if that is the question, then usually now even the fourth dose is ready to be taken. So if you have a gap between a COVID vaccine for 9 months, you are advised to take another dose. Back in Qatar, what we do, I have a booster dose. The third dose has been finished. Then I could wait for minimum 9 months to get my next dose of COVID. So usually it is 9. This information comes out again from the evidence that has been given. So initially they said 3 months because you had data only for 3 months showing the efficacy of this particular COVID vaccine. Then you had a data for 6 months, then you had a data for 9 months. So for 9 months, it still protects the person from the COVID. So probably it is every 9 months once it's finished. Now we have another memo recently saying that we can go for the fourth dose if we have completed 9 months gap after the third dose. So that is what it is. If that's a question, this is an answer for it.
So, question from Rohit. So, what the role of emergency drug in a treatment and how we take care of dispensing that type of drug as a clinical pharmacist?
This question is more for people who are practicing, right? So what is the role of a pharmacist in emergency medicine? Sorry, can I get that question again, please?
What the role of emergency drug in a treatment and how we take care for dispensing that type of drug as a clinical pharmacist?
For dispensing that type of a drug.
Yes, sir.
This cannot be addressed generally, I believe. That the question has to be specific.
So are we talking from the community point of view or from the hospital point of view? Because emergency medications usually you don't find in the community, it is in the hospital. I can answer this question based on my experience and our policies. We have our own policies in our hospital, what needs to be dispensed, what not to be dispensed. For example, if you are an inpatient pharmacist in emergency medicine, in an emergency care, probably you would suggest or recommend the doctor about the emergency medications that need to be taken. You don't dispense them, right? And if you're an outpatient pharmacist, the emergency medications are not dispensed from the outpatient pharmacist. It is emergency, the name itself, it is an emergency medication. This is not dispensed. In fact, it would be there in a setting where you have different healthcare providers who takes the responsibility of dispensing it.
Yes, sir. So, what is the scope for hospital and clinical pharmacy service in developing nation?
This is a very difficult question. The first thing is, you know, the hierarchy about the doctors taking the complete responsibility of a patient care. First thing, they wouldn't accept. There will be a lot of resistance from them, but things are happening in India. A lot of things are happening in the developing countries. The first thing is the curriculum. Now, as Dr. Angadi said, it would be very difficult to have a clinical pharmacy in a hospital because you require, in our organization, because you require to have a hospital attached with the academic, where you can have your students going for the practicals, for the clinical rounds with it. And now the second part to that is accepting this type of curriculum by the doctors. And you must have heard about doctors, they would never even accept the pharmacy. So there are a lot of resistance, but over the time it would change. The only people who can change is the ones who are experienced from outside, who can really produce evidence to show that pharmacists can do a difference, because you are not only a medication experts, but we can even provide a patient care. So it is all about evidence. You produce evidence, you show that what impact this could have, then definitely things change. And it's already been changed. As I told you, in other settings, we do have this, but it has not been practiced as it is practiced in other countries. Because other countries like us, a doctor cannot question us if we ask for some information. A simple example of that. When we call a doctor saying that the medication that is being prescribed by you has a contraindication for his condition, or the other medications he's been taking, or it is not appropriate, can you change it? So earlier, the doctor used to never answer it. He would say, this is my clinical decision, just dispense, who are you to tell me? So that is what it is about the pharmacy profession. Now things have changed here, it is not that way. Now, when we ask, they have to answer. If you don't have answer, they are accountable for that, because the policies are made in such a way that you are accountable if you don't answer what has been asked. From the pharmacy perspective, we should be ready when he says that, okay, if I cannot give it, what should I give then? What is an alternative? So earlier pharmacists used to be stuck because they don't know what is an alternative. They only know what is contraindicated, but they don't know the alternative. So now the things have changed with these type of clinical pharmacy, pharmaceutical care, where they're ready with the knowledge, which they can provide that, use this particular medication. That really puts a mutual understanding of respect between these two. So for how long it will take to develop this in India, I am not sure.
Thank you a lot, sir. Your response to each and every questions which is asked by participants. Thank you for your response. Now I invite Ms. Kartiki Zadav and Varsha Zadav to propose vote of thanks. Over to you, Pratik. Over to you, Varsha and Kartiki.
I feel privileged to get the opportunity to propose vote of thanks for today's plenary session. Very good afternoon, all. I am Varsha Zadav. Along with Kartiki Zadav, we want to thank our versatile and dynamic guest, Dr. Abdul Rauf sir, for sparing his valuable time from his busiest schedule to grace this occasion of international webinar and guided us on hospital pharmacy practice research. On behalf of audience, I assure you that, sir, you have very nicely enlightened the entire topic for us. It was really wonderful session. Now I call upon Ms. Kartiki Zadav to conclude.
Especially we like scope of pharmacy practices researches. There are scopes such as medicines use, practice-centered care, health services provisions. You have told us about methodologies in pharmacy practice researches. It includes classification based on orientation, based on studies, etc. Evidence and impact on pharmacy practices research using evidence to influences practices. Challenges in pharmacy practice and researches. Again, I would like to convey my sincere thanks to Dr. Abdul Rauf sir for amazing session and to our beloved principal, Dr. S.S. Angdi sir, for organizing such a knowledgeable feast for us. I also extend my grateful thanks to all the delegates, including industry personals, principals, teachers, students, and other participants across the globe for their presence and wonderful questions and answer session. And last but not the least, I'm also thankful for organizing committee, all teachers, support staff, and students. Thanks again, and over to you, Priti. Thank you, Kartiki and Varsha. Sir, as a token of appreciation, I request you to accept the appreciation certificate on the behalf of Yash Institute of Pharmacy, Aurangabad. Thank you, sir.
Thank you very much. Thank you.
Now, with this, we will take a lunch break and again join for a last session at 2 p.m. by Dr. Bakshi from Germany. Your feedback link will also send in a last session. Thank you, sir.
Thank you.
Good afternoon, everyone.
Good afternoon, Mayur sir.
Good afternoon.
Okay,
Shall I try to share my?
Yes, sir.
Shall I try to share my screen, or you're still?
Just wait for 2 minutes.
Okay.
We'll start.
Good afternoon, sir. The
Good
future
afternoon.
belongs to those who believe in the beauty of their dreams. A very good afternoon to our guest of Honorable Dr. Mayur Bakshi sir, Honorable Principal Dr. Angli sir, respected teacher and all dear participants present here. I am Nikita Rinde and it's my great pleasure to welcome you all. Now let's start with our last session on single cell analysis by cytometry and its application in immuno-oncology by the expert talk of Dr. Mayur Bakshi sir. Immuno-oncology is nothing but the cancer treatment using immunotherapy where the use of power of body on immune system to prevent, control, and eliminate cancer. So for that purpose, single cell analysis by the cytometry is the important task and that we will learn from Dr. Mayur Bakshi sir. Now I would like to request Ms. Prachi Sheke to introduce our guest of honor to audience.
Thank you, Nikita. Am I audible?
Yes, you are audible. Yes, yes.
Good afternoon, everyone. I am Prachi Sheke, and it's my great pleasure to introduce our dynamic guest, Dr. Mayur Bakshi sir. Sir has an extensive experience in the field of single cell analysis by cytometry. Sir has worked as Doctor Research Associate, Postdoctor Research Associate, Field Application Scientist, and now sir currently working as Senior Product Specialist in Thermo-Fisher Scientific in Germany. Sir has completed B.Pharm from Yash Institute of Pharmacy, Aurangabad in the year of 2019. He completed his Master of Science in Pharmaceutical Biotechnology in Proteomics from Martin Luther University, Germany. He completed his PhD in Natural Sciences from University of Technology, Germany. Sir has several awards to his credits, like Young Investigator Award Travel, Japan, Scholar in Training Award of Radiation Research Society, USA. Sir has also honored with travel stipend for 11th Human Proteome Organization Annual Conference in Next Generation Proteomics, USA. His expertise are multi-parametric single cell analysis, proteomic data analysis, cryosectioning HPLC, and MS for proteomics analysis, and so on. Sir has numerous publications in reputed journals. Along with the technical skills, Sir has participated in various training programs like project and time management, collaborating in high-performing team and communication skill, etc. So we are very blessed to have you, sir. I welcome you in this international webinar on the behalf of respected principal, sir, and entire Yash Institute of Pharmacy organizing team.
Thank you, Prachi. Now I would like to request to our beloved principal, sir, to say a few words. Please, sir.
Good afternoon, all the audience, all participants, speakers. Today, I welcome our esteemed speaker, Dr. Mayur Bakshi, for delivering his talk. Before actually proceeding to his talk, I would like to welcome him on behalf of all the management, teaching staff, non-teaching staff, the organizing committee, and really it is a proud moment for us. One of our alumni is associated with a Thermo Fisher company and is working in Germany, and he has provided his consent and is available here to provide the presentation. So without taking much of his time, I would like to invite him, thank him for accepting the invitation and making himself available for the presentation. Thank you. Thank you.
Thanks, sir. So now I would like to request our guest of honor, Dr. Mayur Bakshi, sir, to start their session. Please, sir.
Good morning. Thank you very much for your invitation. I'm just gonna share my presentation.
Sure.
Please let me know when you see it.
Yes. Yes. Visible.
In the full slide mode, is it visible?
Not visible, but just wait. Yes. Now it is visible.
Okay.
Okay.
Good.
Okay.
So I can start. Great. So thank you very much again for giving me this opportunity. It's really honor for me to come back and share my some of the knowledge that I have acquired after leaving the Yash Institute of Pharmacy and built really my career based on the pharmacy graduation that I have done at Yash, and I absolutely know how I finished my B Pharmacy. So I think we are all in the same boat. Let's call it like that. Thank you for kind introduction and organizing committee, Dr. Angadi sir, for giving me this opportunity. And today I would like to share with you some of the things that we do in the lab in Thermo Fisher Scientific, generally in the immuno-oncology world, oncology world. Let's say immunology these days is after our friend SARS-CoV-2 has came into the scenario, immunology has been of more and more attention to the world. And cytometry especially is becoming a technology of use or favorite technology amongst the world's class scientists in the immunology. So my presentation, yes.
पाटिल मैडम ला भेटा ना म्हणलं.
Can you please mute everybody so that if you have question, please write it in the chat and I'll try to answer. But please stay on a mute during this session. Yes, so I was talking about the global trends in the cytometry. I will try to give you a small overview what we do or what currently is done in the cytometry world around the world. Before I start my presentation, I would like to give you a short disclaimer that this is just a legal content I need to provide because of the European law, and that the presentation is my personal opinion, is not my professional advice or from my organization, especially Thermo Fisher Scientific that I'm working on. And the content is mostly available publicly. They are taken from the public resources and should be used only for the education purposes. And absolutely, I would like to inform you that this is not a diagnostic or professional medical um, suggestion or anything. So please, if you want to use it in any diagnostic or medical purposes, contact your experts in the field. After saying that, let's come back to the after Dr. Rao's presentation, which was amazing in terms of clinical pharmacy. But as a scientist, as a pharmacist, I always like to enjoy in a lab. And let me bring you back to the labs that we are actually in a pharmacy using regularly as a pharmacist or as an R&D scientist or even a pharmacy students, like pharmacology labs, analytics labs, you can imagine whatever. So the combination of everything would be in my presentation today. So the outline of the presentation is as follows, as you can see on the slide. I would like to give three sections, or I would like to give an outline in three sections. One is the technology overview of a cytometry, and this might be more interesting for the students. I'm sure there are some people here in the audience, they already have experience in the cytometry or the fluorescence cytometry, and for them this is more like a revision, I would call it. In that, we can talk about sample preparation, instrumentation, and some aspects of data analysis. What is more interesting for the audience, which is more advanced in the field, is the current challenges and the cutting edge. What we do in our R&Ds, or most of the companies do in their R&Ds, to develop those instruments to help the scientists to provide these cutting edge technologies to either diagnose or prognose the very difficult diseases as a cancers. So this would be very interesting. And lastly, I would like to show some publications, like recent publications or some old classic publications where the cytometry has been used successfully. I can assure you there are more than 100,000 publications in the world of flow cytometry. Depends on what kind of biological question you're looking at. Either it may be cancer or just the basic studies of immune cells. So the technology is available from 3 or 4 decades now, and it's constantly under development. So I would suggest following. I would go through each section of my talk, and if you have a question, please shoot the question or during the presentation, or if you really have burning question, you can unmute yourself, stop me. I would like to have this session as interactive as possible, and as, let's say, more the more you get from it, the more happy I am, let's say. So if I'm still audible and visible, and if you can see my presentation, you can see. Okay, thank you.
Yes, audible and visible both. Okay,
absolutely,
sir.
thank you. So let's talk about the cytometry technology overview. What is actually cytometry? Do we use that in a pharmacy practice? How many people has used cytometry? Or probably some of the people who studied in Yash already knows the lectures from Dr. Angadi sir from first year of anatomy, that we need to remember the names of the blood cells. And at that point I really didn't like it so much, because you need to remember everything by heart. But now more and more I go into detail, or more see application of those things, I get more interested in it. So what you learn in your pharmacy degree is always very applicable in the future. So basics is very important. And thank you for providing us this great basics, Dr. Angadi sir. So why we need cytometry, why we need to know the cells, especially in terms of the immuno-oncology. This paper in the Nature Reviews of Cancer, published in 2015, I just took a random image of it, and as you can see that the tumor or the cancer is such a complex disease, especially when it comes to the solid tumors. It's a mixture, or it's a cocktail of several different cells, not just the immune cells, but the mutant cells or the carcinogenic, oncogenic cells, which are growing rapidly in your. The cancer cells are secreting some stuff. The immune cells are also secreting some stuff in order to prevent growing those. And cancer cells are very successful in hiding from your immune system. That's why they grow in such a big tumor, or let's say they try to escape the immune checkpoints. But please don't get afraid of this figure. It just I want to show you is there is so much interaction going on around the cancer or any pathology that you're looking. So there are different cells, as I mentioned, cancer cells. There are some dying cancer cells. There are natural killer cells. There are T cells, different T cells. There are dendritic cells and different immune cells and other stimuli such as the anti-cancer drugs that we are giving as a pharmacist, or the ionizing radiation for the radiotherapy patient. So it's a big chaos, and we want to have a global overview what's happening in this tumor microenvironment and how we can efficiently do the diagnosis, prognosis, and treatment of those cancer patients to increase their life expectancy or quality of a life. What is other interesting thing is it's not just about cells. There are different types of cells, but there are cells which have different parameters. So as you know, the immune cells, they have different functions. They are attacking the cancer cells. That means they need to produce certain kind of cytokines. They are dying cells. They are going into the apoptosis. They are proliferating cells. They have different transcription factors. They develop different immune response. They have inflammatory markers and different metabolism and different DNA content based on their size, based on their cell cycle, etc. So it's not just about the different cells. What are the cells um, in my tumor microenvironment, but it's also about what these cells are, in which state they are. So we really want to know that, and we need a very powerful tools to analyze such a complex microenvironment. So again, putting back into the context of the drug development of a pharmacy, here is a classification of some of the immuno-oncology agents that have been used in the cancer treatment, typically in lymphomas, myelomas, which are origin of immune system cancers. So if you can see my screen right, and if you can see that there are several. May I please request somebody to mute this background noise? I'm hearing some background noise. Thank you. So you can see that there is a classification of immuno-oncology agents. There are different cell therapies that have been used. CAR T cell therapy is the future of a cancer technology, or personalized medicine, let's say cancer treatment. Then there is a cytokine treatment. There is a monoclonal antibody treatments that we have been looking at, or recently a lot. There is a cell-induced death inducers. Then there is metabolism inhibitors. There are other medicines like small molecules that we have been standardly classifying as a oncolytic drugs, or let's say the cells death kind of drugs. Then you have the oncolytic viruses. There are anti-cancer vaccines, the chemotherapy, and you can imagine. And now what I can tell you from my experience in the hospitals, the more we develop into the field, the more we understand about how the cancer is progressing, or how it is invading the immune system, or for that matter drugs. We are going towards more and more personalized therapy. That means one size doesn't fit all. So we need to focus on a therapy for one particular patient type. Doesn't matter, it's based on his gender, it's based on his age or her age, it's based on the pathophysiological condition, other comorbidities, if they have some cardiovascular challenges, or so basically what currently tumor panels in the hospitals in Europe, they try to do is they formulate a case study around one patient and try to use combination of all those therapies to um, to enhance the therapy or to kill the tumor effectively, let's say, in these patients. So as a pharmacist perspective, it's very, very interesting for us because that's where our drug development is gonna be focused in the future, especially in the personalized medicine in the difficult diseases such as cancers. So now we know there are cells, there are different cells, there are different functions of those cells, and we need to understand, we need to know why these cells are being killed or why these cells are important to know or important to see. And for pharmacy's perspective or for the drug development or personalized therapy point of view. So here the pharmacists are heavily involved in formulation of those drugs, in formulation of the anti-cancer vaccines or immune effector modulators and cytokines. So these are very, very sensitive substances, and we need to have a very interesting formulations for those. But today is not those. Today is not the topic for that. So now we know what we do, why we need to understand the cells. So I would like to not scare you, but I would like to give you a small example or a holistic overview or holistic picture what a cytometry is. As we know that there are cells, we want to analyze them, and we want to get the data out of them. And how we do that is we make a single cells from the different tissues of origin. I will talk about that in my next slide. We stain them with the antibodies, which are highly specific to the cell types, and either they are mostly conjugated with the fluorophores. That means we can detect them by different lasers of the different capacities, and these lasers are in this some boxes called flow cytometers. So I will talk about the how the flow cytometry works, or how the conjugation of the or how the antibodies are stained to the cells. But this is just the holistic picture that we stain the cells with antibodies containing fluorescence. We put them into the boxes called flow cytometers. They have lasers, and then they spit out this data. So there are these three basic components of any flow cytometry experiment or any single cell cytometry experiment. So again, as I said, I cannot see your questions right now, but if you have one, please feel free to put it in a chat. And there is no stupid question. You know, we all learn, we all are curious about different things, and please feel free to ask the questions. So as we know now, there are these three components of the flow cytometry. What exactly what we can measure with this black box, we can say, or white box called flow cytometer? We measure some cell parameters as the cell size and granularity. It is very, very important parameter because based on their size, we can differentiate between different cell types, such as T cells or B cells, or bigger cells like macrophages. The average size of the T cell is 4 to 5 nanometers, while the average size of bigger cells are 14 to 15 nanometers. So you can see they are double in a size. And based on their size, you can already say, hey, this is not my cell of interest. I'm not looking for bigger cells. I'm just looking for a smaller cell. What we can also see based on the forward and the sideward scatter, I will talk about this in my upcoming slide, is are they granular? This is very important because in a various diseases such as allergies or a cancer for that matter, the cells, they secrete the cytokines stored in the granules of these cells. And once they are degranulated, that means they are activated and they are performing their functions. So sometimes you want to know the cell is granular or non-granular because you want to know this cell is functioning. Very important parameter when you're analyzing the cells is this cell alive? Is this cell dead? So we can analyze using different chemicals such as propydium iodide, which stains the permeable membrane of a cell. As you can see, the cell membrane of the dead cell is permeable, and we can stain it with the propydium iodide and see it on the flow cytometer. The most or the utmost important parameter is the cell surface antigens or cell surface proteins. Every cell, rather immune cells, mesenchymal cells, every different kind of cells, they have their different surface markers. And I think we learned this in immunology, not very intensively in a pharmacy course probably, but this is very, very interesting to analyze what is this CD molecules. They are called cluster of differentiation molecules. So each cell, for example, B cell, will be positive for CD19, while T cell is positive for CD4 and CD8. That means we will use the antibody to detect the B cell, which is CD19 antibody conjugated with the fluorophore. I hope everybody can follow. I know this is the last lecture and you have been active from the morning, but I will try to make my best not to make you fall asleep. So again, coming back, we can measure the cell size and granularity, viability, that is live dead of a cell, cell surface antigens, that is the utmost important factor. Then we can also stain the intracellular proteins, especially transcription factors or phosphorylated proteins or interleukins to check the activation status of the cell and the DNA content of the cell. So just to analyze in which cell cycle the cells are. Are we clear so far? Any comments, questions around this topic in the chat? Am I audible? Am I visible?
Yes, sir, you are audible, but until now, no questions are there.
Okay, perfect. Thank you. So in next slide, or this slide particularly, I would like to show, so what do you think when we are focusing a talk around immuno-oncology, what do you think, which kind of tissues we need to analyze the cancer? What kind of tissues? Absolutely, we can classify roughly into two kind of tissues. One is connective tissue, or one is like non-connective tissue, or let's say like blood and the widely known as leukemias or lymphomas, the blood cancers, or the solid cancer of different organs. So what we want from our for our flow cytometry experiment is single cells, and how we will get the single cells from different tissues I'm going to show you in upcoming slides. So namely, first would be a blood, which is very, very important samples for diagnosis, prognosis, and checking the therapy for the cancer cells. So typically people start with 7 to 10 milliliter of whole blood, and the whole blood is then separated by a Ficol gradient, which is a polymer gradient. You add some Ficol, which is highly dense gradient. You add your diluted or non-diluted blood, typically, as I said, 5 ml or 5 to 10 ml. They centrifuge it and take out the something called a PBMC, which are peripheral blood mononuclear cells, which are the immune fraction of your blood, basically. So PBMCs, if you will ask any immunologist or microbiologist or anybody who is working with the blood, PBMCs are the utmost, the characteristic of your immune system. If you can see the real life example, this in this tube there is a Ficol on the bottom and your blood on the top. You can see altogether it is around 10 ml, so it's a 50-50 ratio of a polymer to the blood. And after centrifugation, you can see that the red blood cells are nicely separated, and this is a Ficol plaque, this is a plasma, and this is a mononuclear cells, that is the PBMCs, that is your immune fraction. This method is a little bit tedious. This has this is error prone method, because it's very difficult to get rid of plasma from your PBMCs or the mononuclear cells or the immune cells fraction. There are different automated platforms available to process the blood, but I'm not talking about the automation especially for the blood. But this is still widely used practice in a lot of clinics or in the research labs who are studying the peripheral blood mononuclear cells for cancer diagnosis, cancer prognosis, and cancer progression, even for that matter. So second most widely available tissue type is obviously the tissues. In different the cytometry is being used in preclinical as well as the clinical models. So these are the mouse tissues. The mice is very standardly used as the experimental animal in the cytometry world or in the cancer world, let's say, the cancer diagnosis or studying or treatment. So you can see this is a liver, which is very tiny liver, which is, you can see each lobe is, there are 3 lobes in the liver, and you can see that each lobe is around 1 centimeter. So you don't have really big tissues that you want to get the cells from. So you're limited with the number of cells always when you're doing such a such a single cell harvesting. So again, let me remind you, our goal is to get the single cell suspensions from this tissue, and I can tell you which is very, very difficult. This is a spleen from mice, from a different mice. Some of them are immune challenged mice, and you can see that very tiny organs. Obviously, they will have millions of cells, but isolating those cells from these tissues is very difficult. You need a precise cuts around the spleen. You don't need to let the mouse bleed into it, and if you, because there is a immediate immune response as soon as you try to sacrifice or attenuate the mice. The spleen gathers a lot of immune signals or immune cells generally. So you need to be very quick when you are harvesting such a tissue, and there is a very high variability between those protocols. So you need to be very precise when handling those animals. Secondly, it is mostly used in the clinics, which is human tumor biopsies. And this is the ultimate difficult samples that you can imagine to get the single cells from. Because the size is never regular. Because tumor, as we know, that they are so heterogeneous, and they are very heterogeneous in a in a size, or even in a composition of immune cell composition. So one tumor can be very, very big, especially like if you're looking at the breast cancers. They they are very a lot in a size. It can be from 20 to 25 centimeter cyst to 5 to 10 centimeters small tumor, which is non-malignant. So, and it also depends on who is the surgeon, because some surgeons, they are very precise cutting around the tumors, or they are sometimes they also take around some tissue to take out, scrap out the residual tissue of a tumor, they call it. And why it is important for cytometrists? Because the composition of cells always change based on when it is taken, by whom it is taken, which surgeon it is, how long was it stored, how long was the operation in the patient. Because the tumor, if you have, for example, this is a glioblastoma, which is the tumor for the brain tumor, basically. And on such a small tumor, if you keep it very long till the surgeon is finished with its surgery, there are already a lot of immune responses happening. So very high heterogeneity of the sample. So we want to make sure that we get a very good single cells from those tissues. And those are very relevant for the cancer treatment, because if we, excuse me, if we find out that there is a huge immune response in the tumor, and we cannot give the how do you say, the PD-1 or the standard immune oncology drugs, because the immune response is just an artifact, because the tumor was just waiting outside till surgeon was finishing the surgery. Is it clear? So what we do in these cases? Thank you. So how we can, so for a blood, it was fairly easy. You put a polymer in it, you separate based on the gradient, just do a small centrifugation step, and that's it. But what we do for such a complex tumors or such a complex tissues is there are a couple of methods used in standardly used in labs where they produce the samples for the flow cytometry. One is typically physical dissociation. You take the tissue and you try to grind it, or you slice it with the glass slides. There are different. The problem with this method is you lose a lot of cells. The cells die, and there is a sometimes if there are a bunch of granulocytes, they activate themselves and they degranulate. There is enzymatic digestion process where the enzymes such as collagenase or pepsin is being used for dissociating this tissue. Nowadays, which is advanced technology, which is called the MAX technology, which is based on the magnetic cell separation, where you label your cells primarily with the magnetic antibodies and separate them based on the magnet. So the challenge in this technology is you already occupy the surface receptor that you want to use later for analysis on the flow cytometer. But it's been widely used, depends on what kind of tumor. So this is how it looks like in the MAX technology. So you have a dissociated tumor or dissociated tissue, we can call it. I don't know if it's a tumor example. And you stain them with these antibodies and with the magnetic coupled antibodies, let's call it like that. These are the columns and this is the magnet. So you put the samples here, flow through, and whatever is not stained for your antibody, which will pass through, and the stained and the cells, which stays here because there is a magnet behind it. And then you can take out this magnetic field and elute them into different tubes. So then you will have pure samples also from the tissues, typically from the tumor tissues. So we have gone through two different sample preparations. One is from blood, whole blood, and second is from the tissues. And this is very important. When I show you the applications later in immuno-oncology, please make sure that we understand what are PBMCs, what are the single cell suspension from the tumors. These are two different things. The second important thing is the sample preparation is the choosing the antibodies. And by choosing the antibodies, I mean which cells you want to look at. When you are doing the cancer treatment, for example, you are doing the CAR T cell therapy, you want to look at the CDT or T cell population of the patients, not the B cell or even B cell populations of the patient or the dendritic cell populations. So as I said, we can analyze the surface markers of the cells, which are called CD molecules. And those CD molecules can be detected and can be combined in a different antibody mixtures and can be analyzed. So based on the okay, thank you. So based on the biological or the immunological question that you're asking, you have to choose your antibodies very, very carefully. What is the second important parameter is in choosing the antibody is which fluorophore or what kind of fluorophore is conjugated with such antibodies. Do we have these antibodies commercially available? And this is very important and interesting question, and this is where the cutting edge technology is going towards, and I will show you this in my second part of the presentation. And this is a typical example for antibodies for flow cytometry validation. So these are the CD molecules which detect the different. Can I request everybody to stay on a mute, please? Thank you. So this is typical antibody list. These are the surface molecules that you want to look at, which are detecting different cell types. These are the fluorophores conjugated to them. These are the companies providing them catalog numbers. And what you do basically is you make a mixture of the antibodies and you analyze your single cell suspensions either from tumors or solid tissues or from connective tissues such as cancer or bone marrow. Blood or bone marrow, pardon me. So the sample preparation, as we said, is pretty simple. We have the mixture of antibody, we call it antibody cocktail, antibody panel, whatever you can see it, with the dyes which are staining either DNA or looking for the dead cells. So what basically we do is different steps. We do the viability staining, that is first if the cell is live or dead, then extracellular staining, which is on the surface. Then we do some fixation and permeabilization step, which is PFA, paraformaldehyde. I don't want to go into much detail here. Then we do the intracellular staining and the nuclear stainings in the cell suspension, and a very simple procedure, it's not. So we talked about why we need the flow cytometer. We talked about what kind of tissues we are looking at. We talked about how to get a single cell suspension, how to make antibody cocktails or antibody mixtures, and how to stain your cells. And now the most, or one of the most important parameter of flow cytometry is these boxes. So there are different companies providing wide variety of flow cytometers. Namely, like my company is Thermo Fisher Scientific. We are constantly developing our technology. These are two products from our company, which is called Bigfoot, or SitePix or Attune SitePix. I don't want to go in. I don't want to sell you instruments, so no commercial things here. But also different companies produce those instruments. So it's widely known, different companies. Beckman Coulter, for example, probably you heard the instrument called Coulter Coulter. Sorry, I'm just cannot pronounce it right now. Coulter Coulter, whatever it is. So basically, these black boxes, what they contain? They contain 2 or 3 different elements, which are very important. One is a source of light, which is a laser. One is a flow chamber. And these are our cells stained with the antibody conjugated to the fluorophore. And very important, what is most important in the flow cytometer is they should pass one by one from this laser. So they should not be like clocked here, like hundreds of cells once at a laser. So single cell or going, that's why it's called a single cell cytometry, because one cell has to pass through laser at once. And that means the system has to be very efficient and very fast to detect all those cells. And these cells are driven by something, or focused, or they are driven by something called a sheath fluid, which is a small or a buffer like a PBS, or you can also, some people also use water for driving those cells into it. So it's very important to understand, because as you can see here, this is a laser and the cell is passing through it. For example, this is a cell. We can see that a laser beam is passing through the cell, and it reflects the lights, and that's how we can see the size of cell based on those based on those reflections. We can also see the granularity of a cell, and also based on those reflection we can see which fluorophore is attached to this cell. So we can get a lot of information from just passing single cells through the lasers. So going a little bit more in details, flow cytometers, they have different lasers, from 1 laser to 8 or 9 lasers, but this is a basic 3 laser flow cytometer. As I said, there are different wavelengths of the lasers where the cells are passed through in a single cell format. Then, as you know, this is very wide range of scattering. We need to narrow down the signal. They are passed through some mirrors called dichotic mirrors. I don't want to go into these technical details. You can read it in any analytical books that you have, or you can look some nice tutorials on the YouTube available. And then they are passed through the something called PMT, which is photon multiplier detectors, or PMT tubes, photon multiplier tubes. And the signal is obtained for size of a cell, for granularity, for different fluorophores, and yes, and on a single cell level. Secondly, I would like to show you how the data is generated. So this is our flow cytometer. The cell is passing through the laser, and the voltage is created because the photon is passing always going to the detector. And this is a dynode detectors, as you can see. Again, I don't want to go too much into details of this. This is your signal coming in as a voltage or as a photon, and the signal is detected for a single cell. So FITS is like a color or a fluorophore that is conjugated with the antibody, and you can see that There are some cells positive to this and some cells. But what we see in a flow cytometry, we don't see this kind of plots. We see various different things. And this is where I want to introduce you to the flow cytometry data. Imagine we are analyzing 10 different cells at a different times in a seconds. They have forward scatter and sideward scatter. That means they have different values for the size and for granularity. And this is the fluorophore that they are attached to, and this is converted into the dot. So each single cell on this plot is a dot from these numbers. Maybe let it make it a little bit more obvious to you. So if you look at this dot plot, look at this biaxial plot, we can call it, or dot plot. So if you are looking at the two different markers such as CD4 or CD8, so in this quadrant, the cell is not positive for both of those markers. That means this is not T cell. It can be B cell, it can be any other cell which is not positive for both of those markers. On this left hand side up quadrant, it is maybe a CD4 T cell because it's a positive, it's a higher for the CD4 cells. On the right hand side, this is CD8 because you can see we plotted the CD8 on the x-axis, and on the upper right corner, which is double positive cell, which is higher expression of both these markers. So I hope I managed to give you some overview. Each dot on this plot is one cell, and this is how you have to look at the plots when you look at the flow cytometry data. So how we look at the data again. So for example, we want to look at the live and the dead cells. First, we would look at the size of the cell. Are they big cells or small cell based on the forward or the sideward scatter, or let's say granularity or the size of a cell. And then we will plot on x-axis our die, which tells us if it's a dead cell or not. So as I mentioned before, each dot on this plot is a cell. So for example, in this experiment, they might have acquired 100,000 events, or many, or 100,000 cells, or 1 lakh cell, just to translate it into 100,000 or 200,000 cells, and out of which 73% of them are alive because they are negative for our marker that we use for the dead cells. Am I clear? So these are the dead cells because they are positive for our dead cell markers. Looking at the bigger perspective, again, please don't get confused here. This is just an example. When you look at the complex thank you. So if you look at the complex tissues such as lymphocytes or PBMCs, there is a complex gating strategy, we call it, or selection strategy in a simple words. First, you want to look at your bigger cells, then you want to look at which cells are live in your population, then you want to look at the T cells or B cells, lymphocytes, and on and on and on. You can go forever with the selection of this population and select the subpopulations or subcell compartment of those cells. I know this is very difficult to see if you have, for example, if you are doing a clinical study of a monoclonal antibody where you want to look at different time points of a patient, which is 4 hours, 8 hours, 20 hours, 24 hours, and you have multiple patients, these plots may be just too much to look at. So what bioinformaticians has found out, there are ways to merge this data together and make such a figures out of it, called the dimensionality reduction tools. And again, this to students, my advice is this is a future where the data analysis of the drug testing is going on. So bioinformatics, when you have interest in the coding and programming, understanding statistics, please look at these opportunities where, because there are a lot of clinical trials, drug testing, because of the poor statistics they didn't go through, and the bioinformaticians are always in such a high demand in every kind of organization that I have been in. So you can look at different cell types. You can see the colors. You just take this picture and say, oh, I have so many macrophages, so many B cells, so many NK cells, and so many neutrophils in my tumor tissue. You don't need to look at these crazy plots all the time. So just to review the cytometry workflow, what we have done is we designed the experiment. We took the sample of interest, either blood or tissue, depends on your which drug you are testing for, which therapy you are testing for. We built the panel using the fluorescence antibodies. We made a cocktail, or you can call it a mixture of antibodies. We stain those cells. We acquire it on our flow cytometer instrument. Again, I didn't go into much detail because a lot of information is available online for this. And then you analyze the data using a lot of softwares, commercially or even free available. And as I said, the softwares are always in demand. The different data analysis tools are always in demand. And if you are in those development, you are absolutely really highly required in the field. So this was the end of my first section. If anybody has a question, please please write it in a chat box. If I have to go somewhere in some section again, if you don't understand why we need at all flow cytometry in a pharmacy, I'm doing my formulation. I don't care how the cells looks like. I can explain you. Or I don't know, I'm just synthesizing my small molecule, but why I need a flow cytometry? So this is a really the biology of everything is run around biology, or the human biology or pharmacology, let's say. And ultimate goal for us pharmacists are to improve the patient health conditions, or like life expectancy. So this is where you test your hypothesis if your drug is working. If you've done a QSAR, the model of a drug, is it, maybe it works very good in your modeling software, but does it really work in the model system, such as mice or even cell culture? You need to know that, and this instrument will give you all that information. Am I clear so far? Am I audible? Any questions?
No, sir. You are audible, sir.
Absolutely. Thank you. Let's go into the non-boring part of the cytometry. Let's say, what are current challenges and what scientists are actually looking for in the future? So probably you have seen this picture. I have seen it very often since I came to Europe, especially in the scientific community, and in a clinician community, where people are so deeply involved in their research areas. They are focused, but they don't see the whole picture. They are like scientist and the blind scientist and the elephant. Some is just seeing, oh, this is a part of my experiment. I'm just looking at the drug delivery. And this guy is just saying, hey, this is not my field. This is pharmacist job. And so what we need to see, or why, where is future going of the cytometry, is in a future to have this global picture of what is happening, especially in the tumor microenvironment, especially in the oncology field, where we are talking about the personalized medicine, where, like, from basic biologists to microbiologists, immunologists, pharmacists, and clinicians and clinical trial scientists are involved heavily together. And we want to build this whole picture together, and the cytometry in the future is in going in that direction, to build this holistic picture to make a personalized therapy for a patient. So what are challenges and what are possible solutions? And absolutely, you can imagine number of parameters. There are many cells, different types of cells, and different types of functionalities of those cells, and they behave differently to the drug candidate you are testing, or in radiation therapy you are doing. Currently, in a practice, they are using 10 to 18 antibodies, or panels of antibodies parameters we can analyze once on a cocktail of cells. But what is the future is people want more and more. The scientists, the more deeply they dive in cancer research, they want to analyze more and more parameters, because we have such a rare cancers like glioblastoma. You do not have so much sample of that. You have a very tiny tumor sample and you want to know as much as possible to develop a efficient therapy for these kind of patients. And then there is a spectral technology, which I'm not talking about, but I also talk about mass cytometry. There is a difficult sample such as brain tissue. They clog because you remember, you want to pass the samples one by one on a single cell level from your in your flow cytometer. That means you need to have these single cell suspensions. And if you have this such a complex tissue as a brain, you have residues. The instrument is always clogged because of these very difficult issues. The possible solution is filtration, where you lose the sample. Again, it is very difficult to lose these rare cells. What we need in the future is clog resistant technology, and this is a positive displacement fluidics. I will not talk about that as well. But what also is important, two different things, is speed and automation. As we know that there can be a lot of human errors when each day is different for us. When we are working in a lab, you can pipette a little bit more, a little bit less antibody, you stain differently, you process the tissues differently. And currently, because of the limitations of automation or auto preparation, sample preparation, there are limitations how many samples we can do per day. And you can imagine during the COVID time, where the hospitals were absolutely full and limited capacity, this has been the biggest challenge of the community in the flow cytometry. And the possible solutions, I'm going to show you a few possible solutions. What are the cutting edge in the field? So let's talk about the number of parameters. Why it is difficult in fluorescence based technology to have more parameters? Because you can see this is, for example, 5 laser system, and the more antibodies you add to your panel, the more they cluster together. So you have a limited amount of spectrum that you can use over the fluorescence channels. And for example, if you go like more than 15 to 20 parameters with the different laser types, they try to mix with each other. That means you have a lot of background signal. You will have overlap between the different cell types, and we absolutely do not want that. So what could be the possible solution? So maybe if you have interest, or if you have any suggestion, please type it in the chat box. But I will show you also what scientists across the globe are thinking around. How can we solve this problem? Can we use different fluorophores? Is there different things that we can use? And absolutely yes. So scientists have been coming up with the idea not to use the fluorescence marker, but to use the metals to conjugate the antibodies to analyze the cells. So as you can see, the metals has a very different intensities, and so they do not overlap because their atomic masses are very different. They are not like a fluorophore, they get excited, or they get emission on the different wavelengths. But the metals have unique features as atomic mass, and we can measure the cells based on the atomic masses, and we can use multiple parameters to analyze those instruments or those cells of interest. What is challenge here to analyze the metals is they are not fluorophores, so we don't need lasers anymore. We need some other kind of instrument to analyze such a tiny masses based on you see, there is a only difference of one Dalton between all those metals, or let's say one atomic unit. So we really need a sensitive instrument, which is called the mass cytometer. And this is the cutting edge of the cytometry world. I have been talking with some scientists in India. There are currently 5 such instruments in India, or 6. Now 6th is coming up in Hyderabad. But some of the IITs or ICGP, they have this instrument around India, and I have been supporting some of the scientists there. So what you do is basically, instead of using the fluorophore conjugated antibodies, you use the metal conjugated antibodies. And again, the principle is the same, but the instrument behind is a mass spectrometer. It doesn't analyze the photon anymore, which is emitted from a fluorophore, Rather, it analyzes the ions which are generated by the masses which are conjugated with the antibody. I really don't want to go too much in detail about this technology, but this is, again, as I said, the cutting edge of one of the cutting edge of the technologies. And using mass cytometry, one can detect up to 50 or 60 parameters these days. It's just that the biggest limitation here is the experiment gets very expensive. But if you have very sophisticated clinical trial, this is the technology of choice for the scientists currently. So clearly, we have some other challenges as we talked about. That is a speed, and I want to introduce you to the acoustic flow cytometry, where the year-old technology is being used to enhance the speed of of an instrument. So you can see this is a organ which you find in Europe in different churches. They play like a music. The principle is when you put the sound waves in a pipe, they the particles they gather in the center of this pipe and they create a vibration, thereby a sound. And it's been used really in the centuries. Um, it's also used in the scientific world, such as ultrasounds, which is used in the in vitro fetus detection or visualization, and which is very safe technology. So let me introduce you again back to our flow cytometry. What we have seen before is our cells. They go through the laser one by one by using a sheet fluid, which is called hydrodynamic core, which is a liquid, a water, a solvent, whatever you can say. That's no problem as long as you have a low speed limit, such as 10 to 20 microliter per minute. That means not many cells you can pass through once. What happened when you pass many cells just by using this hydrodynamic core, that means only a seed fluid or a solvent, you will have a lot of cells gathering around your detection point, which is laser, and you will have a spreading of a data. That means against overlap, against you will have detect a lot of doublets or many dead cells already gathering together in one. If you increase such a high speed, which is 100 to 1000 microliter, which is 1 milliliter per minute, which is absolutely a lot. So if you use the acoustic focusing, that means you have a small acoustic field which is producing a small tiny sound wave around this flow chamber. Then with the low flow rates, it's not a problem. It stays the same. But when you have a very high flow rate, such as 1 milliliter per minute and millions of cells per second, this technology helps to align them in the center and acquire the data of a cells in an efficient manner. I can give you, so this is the example how data spreading is being stopped by acoustic focusing. And this is our company's technology, the company I'm working for, Thermo Fisher Scientific, which is actually 10 times faster than any other cytometer on the market. So the advantage of such a technology is, for example, this is the publication from Professor Costariza from Italy. He is the head of pathology and the head of European Cytometry Society. You can see that, I don't want to go in too much into the data, but you can see that he acquired only in 9 minutes 18 million cells. Can you imagine? With so much data in it, 18 million cells, that's 1.8 crore cells in only 9 minutes. And that very efficiently the data. So, and also found out some rare population. So this is very important in a cancer prognosis, especially when you look at the CTCs, which is circulatory tumor cells in the blood. They are, so if you have 1 million lymphocytes or 1 million immune cells, they are 10 or 15 in total. So you really need to acquire millions of cells to acquire such a rare tiny populations or rare tiny cell number when you are looking at the rare diseases or rare cancers. Another benefit of acoustic technology is now the imaging can be combined with the flow cytometry. So we saw these dot plots, we saw these dots for each cell, but is it convincing? Is it really convincing? It's a electronically data converted. It's not so smart system. But in traditionally, if you want to acquire the data with such a high speed, with the hydrodynamic focusing, because you need to have a very efficient camera, you cannot detect the cells because they just move out of focus. But thanks to acoustic focusing now, because of the sound wave, they really stay in the center of this tube, and you can take a very nice pictures of your cells. That means you look at this, your dot plots. The dots are cells, but you can click on each dot and say, oh, and by the way, these are millions of dots. And you can say, hey, I want to look at this population. What is this? What is these cells? Are they active? Are they dying? You can even how do you say, analyze their area. You can make a circle or different shapes around them and see what kind of cells they are. And again, this is a cutting edge, because lot of scientists, they were reluctant using only these dot plots to see if the cells are what they are looking for, especially in case of the rare cells. I hope I managed to convince you there is a lot of things going on in the field of flow cytometry, especially on the technology side. I am heavily involved in developing such a technology, or let's say, in my company, I have really like responsibility to gather these information from different types of clinicians, scientists, clinical trial people, and really give the feedback to our instrument development team. What should be the future technology of future of this technology? Clear so far? Any questions about the cutting edge, where we are going in the future?
Yes, understood, sir. No questions so far.
All right. Okay. I hope I didn't make you asleep. I know this is Sunday afternoon for everyone, and cytometry is probably not the most favorite subject of the pharmacy community currently.
It is
I would
nice
like,
session.
but I would like to now put your concentration more into this last section, because this is more interesting for us as a pharmacist. Why we need this technology at all? And I have been like talking about this a lot. Okay, we have blood, we have different tissues. We can analyze really lot of cells together with the lot of parameters together. Even we can see the pictures of these cells. But how does it help us? Again, I want to repeat this slide. As a pharmacist, we are interested in the personalized therapy. And if you have carefully listened to the talk before from Dr. Rauf, he was talking about the need of research in a pharmacy. Maybe the rare cases or the rare events, the rare allergies, the rare contraindications. And this is very, very relevant in this cancer therapy, especially because the oncolytic drugs, because of the immune therapy, they are not very similar. It's not one size fit all. Everybody reacts differently, somehow differently, and we need to know how a person is reacting to this therapy. So absolutely very important technology as a pharmacist when you are developing a drug, especially for complex diseases such as cancer. Sorry. So one example, maybe the people who are interested in pharmaceutics or development of the drug delivery system. So this is example how a flow cytometry has been used in the testing the nanoparticle drug delivery. As you can see, so this is two different drugs, CPT and Ps, which is Campo, yeah, whatever it is, I cannot really pronounce it. Pharmacy is a long gone for me. Then there is a doxorubicin with the nanoparticles, and they have been like formulated with the nanoparticle. And this experiment shows you how the drug is released over time in a different cell types, or in a cellular uptake of the drug. using the nanoparticles. So you can see this is a forward scatter area, that means the size of a cells, and this is a fluorescence intensity, which is a fluorescence produced by these nanoparticles. And you can see that over the time how two different anti-cancer drugs are taken up by the cells or by the tissue of interest in this case. And this is published in recently in Pharmaceutics last year, actually, by a Spanish group. And you can effectively see that, okay, 98% of the doxorubicin has been taken by the cells by using such a nanoparticles for the delivery system. So, and you can see, you can also create a sustained release system. You can create a different delivery systems for your drug, and you can test using very simple experiment. Just put the formulation on a cell and see how the cell is taking up the drug. Very classic but very beautiful example how cytometry can be used in such a pharmaceutical context. Second example, which is more a little bit, let's say, a standard guideline from the European Research Initiative on chronic lymphocytic leukemia. This is diagnostic panels. This is not related to the therapy, but more rather a diagnosis of cells, which is published 2 years ago in a cytometry journal. And this is to identify certain types of B cells from the patient PBMCs that we have seen how to isolate PBMCs from blood. And you can see that there are particular populations we need to identify to make sure that before we start the therapy to the patient, to see they have this population of the cells in their blood to start the therapy. Talking about the immune therapy, this is an example showing the immune therapy response. This is when you're giving the immune therapy. Again, this is done from the PBMCs for the anti-PD-1 immunotherapy initiation, and then 40 patients has been used to do this trial, and they have looked at the monocyte frequencies using those populations. And you can clearly see this has been published by the Craig Lab from University of Zurich in Switzerland. And we can see that there are different patients and survival rates of a patients after this therapy. And this has been used by the flow cytometry, has been used as a data analysis tool. And when we are talking about the cutting edge technologies such as imaging flow cytometry or imaging assisted flow cytometry, you can see that the CAR T cell therapy, which is becoming a paradigm shift in the cancer therapy, which is you take your own T cells, you activate them against the cancer, and you give it to the patient. Basically, it's like autologous treatment that you just take your own cells, you activate them, and you use them to kill the tumor tissue. And what is important to see is the T cells that should be activated against tumor cells. Before that, without the imaging, you could just see like that. You were seeing some T cells, you were seeing some cancer cells, and you were seeing the double populations before. But thanks to the imaging technology, now we can really see that the CAR T cells or the T cells which are activated against the tumors, they attack effectively the tumor cells and kill the tumor cells at the end of the experiment. So very interesting phenomenon, very interesting. There are a lot of clinical trials or a lot of preclinical or clinical phase 3, phase 4 trials are going on for the CAR T cell therapy. In some cancer, they have been already approved. So this is again a future of a personalized therapy for cancer, especially for the blood cancers such as lymphomas. Lastly, we cannot escape without our best friend here, COVID-19, which is bothering us, which is why I couldn't come to Aurangabad and Yash for a couple of years now. I have not been in India for like 2-3 years since pandemic started, and I was talking to Angadi sir yesterday about it. So you can see here the very simple flow cytometry experiment from control and COVID-19 patient. This is been study from Italy. Probably you know, in Europe, the first country hit by the COVID-19 was Italy, and they have started these initial trials of analyzing the blood of patients, basically the T cells. And you can see with the COVID-19 patients, the pro-inflammatory interferon gamma has been highly produced by the COVID patient compared to the control donors. And the granzyme B, which is anti-inflammatory molecule, or let's say anti-inflammatory pathway, which has been highly deactivated or highly reduced, because you can see that in a control population, it is the granzyme is highly present, while in COVID-19 late patients, the granzyme is absent, while the interferon gamma is highly present in the COVID-19. So such a basic technology can give you so much systemic information, and this is what I wanted to convey you from the beginning to the end of this lecture. So to end all these three sections, what we have seen so far, we have seen the overview of a technology of a cytometry. We have seen what are the cutting edge. We have seen what are the applications. Generally, I didn't show you a lot. As I told you, there are more than 100,000 of lot of publications have available. If you go to the PubMed and just try cytometry and pharmacy or drug delivery, you can see so many publications. I didn't want to go through all of them. And just few takeaways that I want that you make sure that you have. It is very, very powerful tool to study different cell types for response to drugs, therapy, diseases, functionality, phenotype, that is like what cell is this, DNA content, everything in radiotherapy, monoclonal antibody therapy, or even pathophysiology such as cancer or diabetes. You can name, the list is unlimited here. So you can just name any pathophysiology and you can detect this with the flow cytometry. What we can do is use this data from single cells to understand the therapy or to understand disease progression or even the origin of a disease. Lastly, advances in technology such as mass cytometry, imaging cytometry is helping us diving deep into the molecular targets and identification of subcellular targets in a complex tissue environment. Special for students, as I know I have been a student to on a pharmacy in Yash Institute of Pharmacy, and we are really, we try, just to advise from me, we need really highly skilled scientists, and all the speakers could affirm me this. We always need highly skilled scientists in biology or in pharmacy, who have a pharmacy background or who want to develop their interest in the technology development or application development, clinical trial development, understanding. And always is, I said, bioinformaticians, scientists are always needed in each organization that I have been, and opens you door to many different possibilities career-wise, as well as aspiring your own personal, let's say, possibilities in the future. So look for the opportunities, get experience with advanced technologies, just to understand how it is, how does it look, where I can get the internship, for example. Ask advice from your professors. I have been doing that a lot, talking with the different professors, not just from Yash, also from different colleges, or attended seminars like you do now. Ask your seniors, they have experience, and think in future about future, where you think, because you are a future of a healthcare sector. Pharmacist is such an important piece of puzzle in all this personalized therapy, for any disease, for you name it, cancer or anything. You are a future, and think about future, think in future. And that's my only advice for students, that I can really give it from my heart. And with that, I would like to stop my presentation. I would like to thank you very much to the organizing committee. It's really honor to be back and present some of the work I have been doing during my professional life when I left Yash. Thank you to Angadi sir, Devde Patil sir, Joshi sir, organizing committee, teaching and non-teaching staff of Yash, and Indian Pharmaceutical Association. Thank you very much, and I'm open for questions.
Thank you, sir. Thank you very much, sir, for sharing your incredible knowledge regarding a single cell analysis by cytometry and its application in immunotherapy with the very nice presentation. Thank you, sir. Sir, we would like to start our question answer session.
So,
Yes,
can I
please.
start? So first question is, how can we start the career in this field, and what type of courses do we need to complete?
This is a good question. So you can look at the courses, for example, in India or outside India. I have studied at pharmaceutical biotechnology, so this is more going towards the biotechnology. There are a lot of institutes in India who offer such a courses, even offer internships. Look at the industry partners who are doing the like vaccine research or anything, or for example ICEGB or Indian Institute of Sciences. They offer a lot of courses, not just master's courses, but also like short courses, 4 to 6 weeks. You just go to the website. I can assure you, if you Google flow cytometry India, you will find hundreds of colleges, or not maybe hundreds, but many colleges, many universities offering these courses, and absolutely a lot of industries who are taking up the interns for the special knowledge.
Okay, thank you, sir. And next question is,
So, is this method is affordable for the common people, and is this method is practiced in India nowadays or?
Absolutely. So in India, as I said, the fluorescence flow cytometry you can find almost in each city. I'm pretty sure in Aurangabad, in Government College of Science or something like that, or you will have this in each city. In big institutes, it's been regularly used. I know a lot of colleagues who are working in ICEGB, that is the Center of Genetic Sciences in New Delhi, or in Mumbai, for example, IITs, they are really doing this a lot, or BITS, they are doing this a lot. So it's it's like HPLC or a mass spec. It's the similar range of instrument. It's just about getting a funding and writing a proper grant for your scientific study. I think a lot of agencies are open for giving the grants for such a interesting studies nowadays.
Thank you, sir. So next question was,
Sorry.
yes, sir. Next question was, is this technology or immunotherapy, immuno-oncology therapy show the toxicity, cytotoxicity towards the normal body cells?
Absolutely. So every because the ultimate aim is to kill the cells, to kill the tumor cells, right? And that's why this is very good question. Actually, really appreciate for this question, and that's why I'm talking about the personalized therapy, because every person reacts very differently for the different therapies. So especially when you look at the immunotherapy or the cellular CAR T cell therapy, they absolutely show the side effects or contraindications, and that's why we need a tool like flow cytometry. So if you give, for example, T cells to a patient which has a tumor, and suddenly its immune system starts going down, that means the T cells are not reacting to his or to the patient's tumor on a tumor, rather it's like autoimmune response. So absolutely there are a lot of contraindications, but once we have this understanding of how the patient reacts to different therapy, um, we will able to personalize this for the patient.
thank you, thank you, sir. Next question is, can immunotherapy is effective in every person, or is it vary from person to person?
Yes, this is, I think, again it's linked to my previous answer. Yes, it's effective in every. So if you are looking at the immune therapy, has a vast variety of a portfolio. If you look at the therapies such as monoclonal antibodies, they are so specific to the targets. Uh, they work regularly in different patients. But if you look at the cell therapy, they might be differently working on a different patients or different group of patients or different So if you have, for example, previous conditions such as diabetes or cardiovascular diseases, and if in a hypertension, you have some immune modulators that can also increase the hypertension. So this is very variable from patient to patient, and you need to take in consideration such a patient history before you start immunotherapy for oncology.
Thank you, sir. Sir, the last question is here. How much it is effective therapy from the chemotherapy?
So it's a very difficult question to answer because it's really depend on the type of cancer that you're looking at. Look at the breast cancer, for example. In previous times, before the monoclonal antibodies has been discovered, which is trastuzumab, basically, probably you know, they are targeting the herceptin receptors of a tumor in a breast. Before that, the mortality rate of female patients who had the breast cancer was very, very high. And after discovering the immunotherapy, which is the monoclonal antibody trastuzumab, the mortality rate almost dropped to zero. So it depends. It's really difficult to answer this question. You have some tumor types which are really easily cured by the chemotherapy, but chemotherapy, as you know, small molecules, they have their own side effects. You kill effectively your immune system. You have cases like hair loss. You have nausea, vomiting all the time, and it's a very hard on your general systemic effect. It's very hard systemic effect on the chemotherapy. But the immune therapy, on the other hand, is so targeted that the side effects are minimal when it's studied properly.
Thank you, sir. I think we have covered all the questions. Thank you very much, sir. So now I would like to request Mr. J. Pagare to deliver the vote of thanks. Over to you, Jason.
Very good afternoon to our most versatile and dynamic guest, Dr. Mayur Bakshi, sir, Senior Product Analyst, Senior Product and Specialist, Thermo Fisher Scientific Germany. Respected Principal, sir, Dr. S.S. Angadi, sir, Yash Institute of Pharmacy, Aurangabad, and dear delegates. I am Jay Pagare of final year of our college, taking this privilege to deliver the vote of thanks. I want to thank our esteemed speaker who gave his vital time from his busy schedule to grace this webinar and sharing his knowledge on single cell analysis by cytometry. Sir, your valuable guidance was surely will enlighten to all the participants. I would also like to express my profound gratitude to our principal, sir, Dr. S.S. Angadi, sir, for encouraging us for organizing this event. I also like to thank the organizing committee for this event. Last but not least, I am very much thankful to all participants from different institutes for joining us this international level webinar on global pharmaceutical trends. Once again, I am thank you to all those who directly and indirectly contributed to making this event successful. Sir, as the token of appreciation, please accept this certificate from Yash Institute of Pharmacy, Aurangabad.
Thank you.
Thank you, sir. Now we are at the last step of our international webinar. So I would like to request Mr. Bharat Pandey to conclude the program. Over to you, Bharat.
Thank you so much, Prachi. I hope I am audible to all. A very good afternoon, all. My name is Bharat Pandey. It's a moment of great opportunity for me to extend a hearty thanks on occasion of today's international webinar on global pharmaceutical trends to our chairman, Shri Yashwantrao Gadak Patil sir, founder, president, Mura Education Society and president of Srimati Taisaheb Kadam Seva Bhavi Foundation and Research Center, Sonai. Shri Shankar Rao Yashwantrao Gadak Patil sir, Honorable Cabinet Minister of Soil and Water Conservation of Maharashtra. Shri Prashant Gadak Sir, President, Mula Education Society, Managing Trustee, Shrimati Taisab Katam Seva Bhavi Foundation and Research Center, Sonai. Shri Udan Dada Gadak Patil Sir, Honorable Vice President, Mula Education Society and Trustee of Shrimati Taisab Katam Seva Bhavi Foundation and Research Center, Sonai. Respected Chairperson, Dr. Vinayak Deshmukh Sir, and Honorable Principal, Dr. S.S. Angadi Sir, all the delegates from industry and academia, respected principals, faculty members, research scholars, and dear students from all over the world. I would also like to express heartily thanks to all distinguished personalities in today's webinar. We have privilege to host on today Mr. Bhaumik Thakkar Sir, Pharmacy Manager, Walgreens, Illinois, United States of America. Dr. Muralidhar V. Zope Sir, Associate Vice President, Research and Development, Analytical Development, Sun Pharma, Vadodara, Gujarat, India. Dr. Abdul Rauf Sir, Associate Director, Corporate Pharmacy Services, One Ham Medical Corporation, Doha, Qatar. Dr. Mayur Bakshi Sir, Senior Product Specialist, Central European Region, Thermo Fisher Scientific, Germany. I'd also like to extend vote of thanks to our co-conveners, Shri Girish Hukare Sir, Honorable Joint Commissioner FDA Aurangabad and President of IPA Aurangabad. Shri J.D. Joshi Sir, Honorable Vice President Ajanta Pharma and Vice President IPA Aurangabad. And Dr. S.S. Toshniwal Sir, Honorable Indian Patent Attorney and Secretary IPA Aurangabad. I would also like to thank Dr. Subhash Devde Patil Sir, Founder Director of Yash Institute of Pharmacy. Next, I would also like to thank our IIC and IQAC cell also. We are very much thankful to all the speakers of webinar to spend their valuable time and enlighten all of us for the betterment of pharmacy, personnels and professions. Lastly, my heartfelt thanks to all the faculty and organizing committee and also the technical committee members for their contribution, guidance and encouragement in all the efforts. Thank you. Thank you so much, sir. Thanks to all. The link for feedback form is provided in the chat box, so kindly fill the form. Thank you so much.
So thank you very much to all the participants. Kindly fill the feedback form. The link of feedback form is given into the chat box. And I am announced that officially the program is end now. So you can leave the meeting.